Discuss a plethora of subjects and plethora of studies today. The DNA cell ploidy analysis, some of our faculties in my department have now taken up this project of DNA ploidy analysis, ion flux, cell viability, redox, you name it, there are so many of it, right? So the background information, of course, it's a quickly expanding technology and is increasing in popularity and gives us the ability to analyze many properties of many cells in a very, very short time. I hope you all are understanding. I'll keep it very simple, right? So now let's go to the four main components of a flow cytometer. What happens? What actually happens in the flow cytometer? The cells are in suspensions and they flow in a single file, one cell at a time. Where do the flow pass? They flow past a focused laser, and then they scatter light, and then they emit fluorescence. And this fluorescence is filtered and collected. Collected how? Into a data which is converted into digitized value, and then that can be read by specialized software. It is as simple as that. So fluidics is the cells in suspension which flows in a single line. An interrogation is when the focused laser enters and interrogates each cell, and the cells then scatter light and emit fluorescence, which is filtered and collected into a digitized value which we call electronics, and then we interpret based on our software. So these are the four very, very simple, essential part of a flow cytometer. Boys and girls, are we clear? What we are going to now talk about is fluidics, right? So what happens in a flow? Let's simplify it further. So this is the test tube in which we have the sample, and we got a pressure. We eject the sample out. Where? Into the flow cell. So the cells in suspension flow in a single file. Now you need to have these cells to flow one by one because this is a single cell analysis, and we do it in a multi-parameter. So one cell has got maybe 20 properties. Each cell, 20 properties we analyze simultaneously, and we accomplish through a pressurized laminar flow system. The sample is injected into another sample, and that sample is called a sheath fluid. And then the sample is passing through a small orifice, which is tailor-made by the proprietors, which generally ranges from 50 microns to 300. So here you have a surrounding fluid called the sheath fluid. You have got in that thing a sample fluid, which is being injected through a pressure through a small orifice. Are you all clear? So we need to know what is laminar flow basics. Laminar flow is something which is characterized by fluids moving in a parallel layers. So they don't mix. So if they don't mix, they create a predictable and a consistent flow pattern. Now this is the most important statement, the laminar flow. So in a flow, who does the lamina? The sheath fluid. So the sheath fluid maintains this flow, ensuring that sample stream remains separate and remains focused. Are we clear? Then we come to the fluidic schematic. So you have the sample and you have all sorts of pressures which helps the flow work. The cells from the sample tube are injected into the sheath stream, and the flow is laminar. And then we'll talk about hydrodynamic focusing. This is something which pushes the cells to line up in a single file along a long axis. So here you've got a laminar flow, now you've got a hydrodynamic focusing. The shape of the flow provides the means for the hydrodynamic focusing. So by preventing mixing, the laminar flow ensures individual cells or particles, they pass through the laser beam one cell at a time, allowing precise analysis. Are we clear? Okay. So what is hydrodynamic focusing? Now sheath fluid is a buffered saline. It flows at a higher pressure than the sample fluid. So this differential is very important. The sheath fluid का pressure ज़्यादा है, sample fluid का pressure कम है. So this differential pressure ensures hydrodynamic focusing. This, along with the design of the flow cell, forces the sample to flow in a narrow central core, effectively focusing the cell. आपका उद्देश्य क्या है? लेजर एक एक सेल को हिट करेगा. So the hydrodynamic focusing and the laminar flow ensures that. So the process of focusing the sample into this core is called hydrodynamic focusing. So these two important terms you must know, laminar flow and hydrodynamic focusing. So this is the flow cell, see it carefully. ये देखिए, बाहर वाला fluid, this is sheath fluid where the black arrows are there. Inlet is sample, those cells, those round structures, one by one they are coming from the injector tip. So this is how it happens. This is the sheath, as you are seeing the flow diagram. This is the sample stream and this is the cell. As simple as that. Or though terms, laminar flow and hydrodynamic focusing is ensuring that the cells form one at a time to be hit by the laser. So this is another diagram showing the same thing. So this sample differential should be optimal. That means the sheath fluid minus the sample fluid pressure should be optimal. If the differential is wide, you will never have a single cell suspension. You will have multiple cells coming. That is not the idea. You want single cell. So the proprietors make a very, very optimal pressure of the sheath fluid and the sample fluid, and the differential of which ensures hydrodynamic focusing. Right? Okay. So let us recap. What is fluidics? Purpose: to have flow of cells one by one passing through a light source, which is the laser. The cells move out of the tube because there is a greater pressure on the sample than on the sheath. The cells are focused due to the hydrodynamic focusing and laminar flow. So this covers your fluidics. This is the most important aspect of flow cytometry. Have you all got any doubts? If you have, I'll clarify it in the workshop, not here. Okay? Now we come to the next part, which is interrogation, right? So here we talk about laser. Light source needs to be focused on that point where the cells are focused. So you have ensured the hydrodynamic focusing. Now you want the laser to hit each cell one by one. So on flow lab instrument, we have the lasers. So what is laser? Light amplification by stimulated emission of radiation, right? You must know this full form. You're talking about lasers and you don't know the full form. It is very embarrassing. So they provide a monochromatic wavelength of light. They provide milliwatts to watts of power. They provide coherent light. We will discuss what is coherent light, which means all the emitting photons have the same wavelength, the same phase, and the same direction as the stimulation photons. Are we clear? Recall your class 11-12 physics. So coherent light means the same wavelength. Okay. So here we have the types of lasers. You will see in our instrument we have argon laser, which has a wavelength of 488 nanometers. We have helium neon, we have helium cat, we have red diode, and so on and so forth. So like our instrument has got a 3 laser, 12 color laser. A BD का हमारे instrument है 10, मतलब in one tube you can do 10 antibodies का simultaneous interpretation. So we'll discuss that in the workshop. Okay? Fancy names. Basically, it is what I'm trying to teach you. Now there are two important aspects of flow. Whatever you do, you must know what is light scatter. So when a light from a laser interrupts or interrogates a cell, that cell will scatter light in all directions, right? So this light can travel from the interrogation point to another place where we have kept a detector. For example, a photomultiplier tube or a photodiode. You have read this in class 12? So PMTs are photomultiplier tubes, photodiodes. So these are the detectors which are going to catch hold of that scattered light, which the laser hit on the individual cells. Got it? So this is forward scatter. The direction is along the same axis. So we call it the FSC channel. Now the intensity of the signal is very high, so we have to catch it with a photodiode. And the forward scatter tells us the size of the cell. So for example, a lymphocyte, will it have a high forward scatter or a low forward scatter? Obviously, it will have a low forward scatter. The lower the forward scatter, the smaller the cell. So which is the smallest cell? We are talking about lymphocytes. So which will be the cell with the highest forward scatter? A granulocyte. So this is the basics of flow, right? Okay. So then we have the side scatter. So this is forward scatter. Have a look at this. The laser beam is hitting the single cell suspension, scattering, and then we have this FSC detector, which is a photodiode, which catches hold of this light. Now we are going to analyze that light into a voltage and electronics. That is later. Then we come to side scatter. What is side scatter? That is scattered at 90 degrees to the axis of the laser path. Now we got a side scatter channel which detects this light. Now, what is the meaning of this? The complexity or the granular content of the cell is site scatter. So, which is the cell which has the smallest site scatter? Obviously, it will be a cell without any granules. Which is that cell? Lymphocyte. Have you understood? Similarly, a granulocyte will have a highest site scatter. Why? Because it's complex. Basophils, eosinophils, they will have high site scatter. So, name a cell with the lowest forward scatter and a lowest site scatter. Anyone? Lymphocyte. Name a cell with a very high forward scatter and a very high site scatter. Granulocyte, eosinophil, basophil. Name a leukemia with a very high forward scatter and very high site scatter. Acute promyelocytic leukemia. Are you all understanding? So, this is the fundamentals of flow cytometry. No rocket science. Okay? So, we are going to remove this myth from all of you. You are all MD students or senior residents. We are going to simplify flow cytometry in the workshops in the next 2 days so that you start loving this technology. That's the idea. I hope I'm impressing you all, isn't it? Okay, okay. So, this is site scatter. Now, we have started 30 minutes late. So, I'm going to speak 15 minutes less. And Richa has also promised she's going to speak 15 minutes less. So, we'll skip a few diagrams because these are repetitions. Okay? So, you know all about this. Site scatter, collecting lens, site scatter detector, forward scatter, photodiode. Multiple choice question पूछेगा. Forward scatter is collected by photodiode, PMT, both, none. Answer, photodiode. Strong signals are collected by photodiodes. Site scatter, is it collected or by a photodiode or PMT, either of two or none? PMT. Smaller signals are collected by PMTs. Larger signals are collected by photodiode. Simple questions, ठीक है? जब आप लोग board exam दोगे, ये सब question पूछेगा, ठीक है? So we are not going to repeat. Now see this. Again, the same thing which I've said, lymphocytes, monocytes, granulocytes. Now you all know the fundamentals, ठीक है? Now I am not going to show you any more of the slides, ठीक है? I am now going to stop extempore, ठीक है? और मैं slide नहीं दिखाऊंगा, ठीक है? So now you have done fluidics. You have interrogated cell from laser. Now you are going to capture these through photomultiplier tubes. You are going to convert this into an electronic message. And that electronic message through a software is what your interpretation is. You are all doctors. Okay? So you are going to interpret through this formula कि भाई ये leukemia है कि leukemia नहीं है. Is it chronic lymphoproliferative disorders? For example, if you have a y-axis and an x-axis, y-axis is site scatter and x-axis is CD45. Very small site scatter, extremely bright CD45. What is it? A mature lymphocyte. ठीक है? Which are the conditions which have mature lymphocytes besides normal? Chronic lymphoproliferative disorder. Now come to another question. Site scatter low, CD45 dim. What will it be? Blast. Basic concept yaad hai? So I'm now going to skip 20 slides. Okay? And we'll come to something different. We are not discussing Stokes shift. These are the various fluorescent detectors. We have discussed that. We'll now come to something different so that you all understand. So this is the way we select fluorochromes. We must know what are the lasers in the fluorochrome, in our flow cytometer, what are the filters in the fluorochromes. We have to discuss brightness. Suppose we have a fluorochrome which is very bright, then the emanating antibody should be low emitting. So if we have a very high emitting antibody, we'll select a lesser bright fluorochrome. This is the fundamental. For example, FITC, fluorescent isothiocyanate. It is a green filter. It's very bright. So low emanating surface antigens are selected for a bright fluorescent. For example, CD45 will be collected by FITC. Okay? Low emitting. Now suppose we have PE, phycoerythrin, which is the name of another fluorochrome which is seen in the red corner. So high emitting antibodies will be collected by phycoerythrin. So like that we have various combinations. So our manufacturers combine these fluorochromes and we know exactly which surface antigen can be coupled with this monoclonal antibody into that fluorochrome. So bright fluorochrome, less surface antigen excretion, high surface antigen excretion, low intensity fluorochrome. This is some of the fundamentals, okay? So we'll not go into all this. So we have the types of fluorochromes. These are the common fluorochromes. FITC, PE, PERCP, APC, you'll read all this in the workshop, right? So in the workshop, we are going to discuss all this with you, right? These are the various combinations, spectra. Now we come to a very important aspect, filters. So we have covered fluidics, we have covered the interrogation, we have covered the lasers. We'll talk a little bit of filters. Many wavelengths of light will be scattered from a cell. Now we need to have a procedure to split this light into its specific wavelength so that we detect each of this light independently. Have you all understood? इतने लाइट स्कैटर हो रहे हैं, हर लाइट के एक एक वेवलेंथ को हम लोग को पकड़ना है. कैसे पकड़ते हैं? फिल्टर्स से. सो व्हाट आर फिल्टर्स? ऑप्टिकल फिल्टर्स आर डिजाइन्ड such that they absorb or reflect only some wavelength of light while transmitting the other. Have you understood? So we have three types of filters and these are important for you. LP, long pass filter, SP, short pass filter and BP, band pass filter. ये तीन term क्या है? Nothing to worry. Long pass. They transmit all the wavelengths greater than that specified wavelength. अगर मानो आपका स्पेसिफाइड वेवलेंथ है 500 LP, इसका मतलब है दे विल ट्रांसमिट ऑल वेवलेंथ्स ग्रेटर देन 500. क्लियर? सिमिलरली वी हैव शॉर्ट पास. व्हाट इज शॉर्ट पास फिल्टर? ट्रांसमिट्स ऑल वेवलेंथ लेस देन द स्पेसिफाइड वेवलेंथ. अगर लिखा है 600 SP, सो दे विल ट्रांसमिट ऑल वेवलेंथ लेस देन 600. एंड देन वी हैव एन इंटरेस्टिंग कॉम्बिनेशन कॉल्ड बैंड पास फिल्टर. इसका टर्म ऐसे लिखता है 550/20 बैंड पास. इसका मतलब है 550 - 10 एंड 550 + 10. तो 540 और 560 के बीच में जो वेवलेंथ है इसी को ये लोग ट्रांसमिट करेंगे. हैव यू अंडरस्टूड? तो फिल्टर्स आर ऑफ थ्री टाइप्स. व्हाट आर दे? लॉन्ग पास, शॉर्ट पास, बैंड पास. क्लियर? आई एम जस्ट सिंप्लीफाइंग इट. ओके, इट इज नॉट दैट सिंपल बट आई एम सिंप्लीफाइंग इट. Then we have something called dichroic filters. These are filters which are kept at 45 degrees. Dichroic filters are either a long pass filter or short pass. They are never band pass. So जब आपको पूछते हैं कि dichroic filters क्या है, they can either be a long pass filter or a short pass filter, and they are kept at 45 degrees angle to the incident light. And I'll show you a diagram. See this. This is a dichroic filter. Either a LP or a SP kept at 45 degrees angle. Part of it will be transmitted, part of it will be 90 degrees में reflection होगा. ठीक है? So now with that we have an optical bench layout. We separate, scatter and multiple fluorescence wavelengths simultaneously from each cell. So we have spectral properties, we position the filters and this is how it looks inside the tube. 505 SP, 605 SP, 555 band pass, like that, you know, we have all this. And this is what the machine looks like, a look inside the box. Compensation will not go because now this, in earlier days, Dr. Renu ma'am, Dr. Pati was with us, we used to do it manually. Now everything is automated. We don't want a particular fluorescence with a spectrum of wavelength to overlap with another fluorescence with the same wavelength. So we have to separate that. That is called compensation. अगर दो फ्लोरोकोम का वेवलेंथ एक साथ आ जाएगा, आपका डेटा खराब हो जाएगा. हमें एक एक वेवलेंथ को अलग अलग से लेना है. दैट इज कॉल्ड प्रिवेंटिंग स्पेक्ट्रल ओवरलैप. दैट इज कॉल्ड कॉम्पनसेशन. ओके, सो विद दिस थ्री थिंग्स इन माइंड वी नाउ गो टू द लास्ट पार्ट ऑफ माई लेक्चर विच आर डिटेक्टर्स. फोटोडायोड्स, स्ट्रॉन्ग सिग्नल कौन सा? फॉरवर्ड scatter. Excellent. Photomultiplier tube, side scatter. Lesser signals. Very good. So these are photodiodes and photomultipliers. We have talked everything. Now this is the interrogation recap. A focus light interrogates a cell and scatters light. The scattered light travels down a channel to a detector. Forward side scatter, size and cell membrane shape. Side scatter, internal cytosolic structure. Fluorochromes on the cell or in the cell will become excited by the laser and emit photons. These photons drive down channels and are steered and split by dichroic filters, which could be either long pass or short pass. And the specific wavelengths are then detected by photomultiplier tubes that have a filter in front of them. Clear? Are you all clear? As simple as that. So we have discussed everything. Fluidics, interrogation, electronics, we will not go into details. Now I'll come quickly to the last part, which is what is gating. Okay, this we have discussed. We'll not go into all those details. This will be clarified in the workshop. Okay. Now we come to, once the value of each parameter are listed, specialized software, for example, हमारे जो flow cytometer है उसका software का नाम है FACSuite. ठीक है. So specialized software can graphically represent all your data. Common programs, अगर आपको पूछे, CellQuest is the name of a software. हमारे टाइम में FlowJo होता था. You know who was FlowJo? Anyone? Florence Griffith Jr., 100-meter world champion, who died very early of a heart attack. She used to run with, her long nails and nail polish. Her 100 meters record has not yet been broken. So the Americans, when they introduced BD, they put such a software which was so quick, they named it FlowJo. As quick as Florence Griffith, Olympic 100-meter champion. So these are all jokes, So anyway, so we come to now, sorry for this thing, but I'll just come to the gating part and finish up. These are types of plot which we are going to discuss in the workshop. Single color histogram, 2-color dot plot, 2-color contour plot, 2-color density plot. These are the examples which we'll show you in the workshop. Contour plot, हमारे पास properties है, हम लोग contour करके देखेंगे कि भाई ये दोनों cell एक जगह है कि is it 1 cell population or 2 cell population. So the contours will give you. For example, you want to see is it a lymphocyte or a monocyte. So a contour will show you this is lymphocyte, this is monocyte. If there's one population, the entire contour will come into 1 cell population. So we are going to show you all that thing. Okay? Now gating, the last part. A subset of cells on a plot is identified, allows the ability to look at parameters specific only to that subset, and we use Boolean logic or sequential gating to include or exclude multiple gates. So this is a process of selecting and analyzing specific cell population. It's a very critical step, because if you do the wrong gating, your entire experiment is gone. You will give a wrong diagnosis. So you must know as a morphologist, you cannot enter a flow room without seeing your slide. You are first and foremost a morphologist. After that, you enter the flow cyting. Okay? You must know ontogeny. You must know gating. क्या गेट कर रहे हैं? आपने स्लाइड देखा है. You are pathologists, you are doctors. So you have seen the slide. You have already diagnosed this as APML or a BALL or whatever you want to say. The flow is confirming and prognosticating what you already know, or maybe you may not know. So it helps you. But to enter a flow room without morphology is a cardinal mistake. Please don't do it. Okay? So this is purpose, to isolate and analyze specific cell population, select an area on the scatter plot to include or exclude cells. Cell size, granularity and expression of cell markers is what is based on, and ensures data accuracy by excluding irrelevant events. So जो आपका important है उसी को आप gate करेंगे. Have you all understood? So we have got various types of gate, like B cell, CD19 was a side scatter. Very bright 19, chronic lymphoproliferative disorder or a mature lymphocyte. Dim, blast. Okay. T cell, CD3 was a side scatter. Viable cell, we use actinoamino acid. So AAD we can use, or we can use a 45 side scatter. Blast, CD34, the primitive undifferentiated marker. Side scatter CD34, blast. Lymphocyte gate I have discussed. Monocyte gate, now in monocyte, जहाँ पर monocyte आते हैं वहाँ पर क्या क्या हो सकता है? AML आ सकता है, CML आ सकता है, hairy cell leukemia आ सकता है, myelodysplastic syndromes के cells आ सकते हैं. So they are bright CD45 with a higher side scatter. So all these gates you know because you have done the morphology. You know where the monocytes are going to come. ठीक है? उस monocyte gate में AML, CML, CMML बहुत सारी चीज़ आ सकता है. ठीक है? So ये मत सोचो कि monocyte है तो ये है. Hairy cell leukemia का gate भी वहीं पर आ सकता है. So this knowledge is very important for all of you. ठीक है? Hematogones, you are going to discuss hematogone in MRT lectures, Low side scatter and dimmer expression of CD45 compared to the blast. And they are dim CD10 compared to a blast which is bright CD10. Hematogone is such an important diagnosis where you cannot differentiate it from a blast in morphology. Of course, when I was in AIMS, I was taught how to differentiate and it has really helped. Hematogones never come in sheets. Blasts come in sheets. Hematogones are single. Hematochondria are never in peripheral blood. Hematochondria are in bone marrow. This morphology is so important for you before you come and see the hematocon gating in the flow. So this is what I'm trying to tell you. Without morphology, please don't enter the flow. Plasmacytic gate. Plasma cells here. Are they blast or plasma cells? CD45 negative, right? So these are all the gating strategies, right? We are going to discuss that in the workshop. Okay? I think that's it. We have told you enough. Sample preparation, Dr. Richa is going to tell you in the workshop. So we are not going to do all that. This is the principle of panel selection. Highly expressed antigen, dim fluorochromes. At least one common reagent in each tube, we'll tell that in the workshop, so we can track the population of cells in between the tubes. Antigens for subpopulation identification within a particular lineage to be counted. For example, I have 4 tubes, I'll put my CD34 in all the 4 tubes, so that I know what are my blast gates and what are my non-blast gates. Okay? So this is exactly what happens in a flow cytometry. Fluidics, interrogation, electronics, and interpretation. I think with that, I have come to the end of my lecture. Thank you very much.
So I would like to thank Dr. Chatterjee for such an excellent presentation. You all will agree. It is simplification of such a complex topic that he has taken. And the workshop which is to follow, you can please feel free to clarify all your doubts, because this is a great opportunity to learn small nuances of flow cytometry. So thank you very much, Dr. Chatterjee. And we move on to next. I think Monica's going to.
Yeah. I'll just tell you again in short what ma'am said. Keep it simple. ठीक है? हर चीज़ को complex मत बनाओ. सब कुछ simple रहो और do your morphology before entering the flow. Without that, I think that is the message we are going to give you in 2 days. You all are morphologists first.
They say strong roots make strong trees. I thank you, sir, for simplifying the basic concepts of flow cytometry. I now request Dr. Monica Gupta to introduce our second speaker, Dr. Richa Chauhan.
Okay, good morning everyone. After such a wonderful and comprehensive talk given by Dr. Chatterjee on the overview of flow cytometry, and it's now we are going to move on another topic, that is the application of flow cytometry. So our next session will build up upon this same topic, that is we are going to discuss the approach in acute leukemia. We are going to discuss what are the various different gating strategies, what is the panel designing for acute leukemia, sample preparation, and finally the case-based discussion by Dr. Richa Chauhan. So she is Dr. Richa Chauhan, who is working as a assistant professor in Department of Hematology, All India Institute of Medical Sciences, New Delhi. She is a DM in hematopathology, and her key areas of interest are MDS and acute leukemia. So over to you, Dr.
Good morning, everyone, and thank you for the opportunity. Sir, do I have 15 minutes?
Yeah, 35.
Okay, thank you, sir. So after such a simplified version of flow cytometry from Tathagat sir, I will skip some of the part of what I had wanted to present, because sir has already simplified most of the things, but I would like to make some comment on basic concepts before delving into approach to acute leukemias. So one of the most important thing for any approach to any hematopoietic neoplasm is that your instrument validation, calibration, your assay which you have designed, panel designing, choice of antibodies, which antigen markers you want to study, for what type of leukemia, all these steps, including how your technician runs the processes the sample and how it is acquired, to how the hematopathologist or pathologist analyzes the sample. All these steps are, each of them are very essential to reach a proper diagnosis. Any falteration can lead to a misdiagnosis. So panel designing is very important, as sir has covered about fluorochromes. So fluorochromes are molecules that are excited in a particular electromagnetic radiation, basically a visible spectrum, where they are excited by a particular wavelength, and they are excited to a particular level, and then when they fall back to their baseline, they emit energy which is lower in strength but power, but it is higher in wavelength. So this is Stokes shift that sir had described. Okay, so whatever the difference is in the energy, the excitation is by a laser, and the emitted energy is later on gathered by the photo multiplier tubes, and they are converted into electronic signals that are studied by the instrument, software in the instrument. Okay. So what are fluorochromes? Fluorochromes can be some naturally occurring molecules. They can be small in size, they can be large in size, or they can be artificial polymers, and they can be tandem conjugates. Okay, so going to the concept of tandem conjugates, I'm skipping all this because most of this has been covered by sir. So tandem conjugates are very important because they are the ones which create nuances while doing flow cytometry, because they tend to break. So tandems are like friends. So one of them is the donor and the other one is the acceptor, and they are together, they are making the spectrum wider, so that a laser which has lower power can produce a signal which is emitting in a higher wavelength. So otherwise single molecule has a specific spectrum, okay, of excitation and emission. But if we use a combination of donor acceptor, then together they function together to give a wider spectrum of emission, excitation and emission. Okay. So various small molecules can be combined with large molecules, small molecules can be combined together, but the problem is that they tend to break if exposed to light. Okay. So there are precautions that have to be taken while working with the tandems. Also, compensation and spillover is something that we need to see while working with tandems. So sir also already covered about relative brightness of fluorochromes, and there are various websites. You can go to Fluorofinder, proprietary vendors' websites to know how your fluorochromes work. So while choosing the panel, so while designing the panel, which antibodies you want to see in one particular tube, you should have a target in mind, that with this tube and with having these many particular markers, I want to identify any blast with any lineage. So your first tube in acute leukemia will have lineage specific markers, including common markers, common leukocyte antigen marker. Okay, sorry, here. Okay, then lineage specific markers like cytoplasmic MPO, CD19, CD3, cytoplasmic and surface, sorry, this is surface, CD7 and CD79A. Then additional tubes in the panel would be to categorize those cells, whether they are mature, immature, or which type, subtype, to finally categorize your leukemia or lymphoma. So these are the websites that you can use to assess the properties of the fluorochrome conjugate for that particular antigen of interest. Okay. So while choosing, buying antibodies, you have to be very careful whether, if you are choosing between two fluorochrome conjugates, whether you want CD7 in APC or you want in Pacific Blue or V450, then which spectrum is suits your panel, and what are the overlaps with other antibodies. Okay, so these software tools help in that. And you have to know that while choosing your specificity or the target species should be human. If you by mistake buy a mouse antibody, then it will not work. So very carefully you have to see what all you are buying and choosing. Then compensation also sir covered. So we do not want signals from one fluorochrome to be spilling over to the various channels. So if it's a 10 color instrument, it will have 10 channels. If one channel has PE, we don't want PE signals to be spilling over onto other channels like FITC, per-CP, Sci-Fi, APC, etc. So the negative MFI of the negative population and the MFI of the positive population for PE should be same. So in any other channel, FITC, APC, APC-H7, whatever is positive in PE, it should be negative in those channels. Okay, so that matching and all, sir talked about manual compensation. This is available as a automatic method in softwares. But ultimately, some manual compensation is still needed, because still some tube settings or specific experimental design can lead to background fluorescence, and you may need to do further compensation spillover settings. So what is stain index? Stain index is basically, it's a method to determine relative. I am talking of relative fluorescence here, not absolute, because absolute standard units of fluorescence is MESF. And it's very expensive to validate, standardize it. So relative fluorescence we see of the negative population in the sample and the positive population in the sample. And we compare. So we can take a ratio of the positive MFI, positive population MFI, and a ratio of the negative population MFI. And if the ratio, you want to choose APC for CD7 or V450 for CD7, if your MFI, if your stain index is higher for APC, then you choose that fluorochrome combination. If it is higher for V450, better for V450, you choose that. So how much separation you obtain from positive and negative population. So it's a formula, difference between the positive negative MFI divided by robust ST of the negative population into 2. SNR, signal to noise ratio, is just this ratio of the positive to negative. Both the parameters should be used while doing antibody titration. So while preparing the panel, so proprietors, companies, they provide recommended volume of antibody to be used. Usually it is 2 to 5 times the concentration, and you can go to further dilutions to be cost effective also while panel designing. So I'm not going into the details, but this is the way we choose the peak value of stain index and SNR while doing the experiment. This is the recommended Euroflow consortium guidelines, which we can see they recommend, but it's not always possible to imitate what the international guidelines quote. You have to design the panel, what works in your lab, which reagents you are using, how your instrument is working, everything plays role. So a lot. So acute leukemia orientation tube is basically a BD term, but we use it loosely. So acute leukemia orientation tube will have some surface marker and some cytoplasmic marker. What is meant by surface? What is meant by cytoplasmic? So antigens are proteins or modified proteins that are present on surface. You want to target it with antibody that has been conjugated to a fluorochrome. Certain antibodies are inside the cell. To target those, we have to make pores on the surface of the cells of interest. So while making those pores, first we have to fix the cell, and once it is fixed, then we use permeabilization reagents, which are mostly detergents, not specified by the companies mostly, but they are saponins, tritonics, etc., which make pores in the cell so that antibodies can go and bind to the proteins of interest. So cytoplasmic lineage specific antibodies are for myeloid series, MPO, for B lineage CD79A, for CD3 for T cell lineage, and megakaryocytic lineage CD41, CD61, 42A, B. Now, within the cell cytoplasm, there is another cell membrane that has to be made way through, that is nucleus, because TDT lies there. So there are some issues that laboratory face with TDT staining, but that is not, I'm not covering that. So you have to have very proper steps while doing staining. And so what is a blast? Coming to the approach to acute leukemias, blasts are immature cells that are derived from hematopoietic stem cells, but because of certain mutations, they tend to lose the capability to differentiate, and they have potential to proliferate unchecked. Okay. So blasts are either lymphoid lineage or myeloid lineage. So this is a table chart depicting what all antibodies, markers are useful to pick up the lineage. Okay. So CD19, 22, 20, 79A, PAX5. PAX5 is usually used in IHC. So you can use a combination of markers by immunophenotyping, means by flow cytometry or IHC, So you can use different methods to pick up these antigens. Then T lineage, CD7. CD7 is retained in immature hematopoietic neoplasms, but it tends to be lost in mature neoplasms. Very few TCLPDs express CD7, but it is commonly expressed in TLLs. That is why your first marker for gating in TLLs is CD7, because CD3 is commonly lost. Then myeloid lineage, MPO, if it is there, it is the lineage specific marker for myeloid. But what about monocytic? Monocytic NSC positivity, CD14, CD36, CD33 bright, and CD64, along with presence of CD11B, 11C. Erythroid lineage by CD36, CD71, 105, 117, and 235A, though it comes in very late stages of maturation of erythroid CD cells. This we have covered. Then PDCs. So there's a neoplasm, blastic, plasmacytoid, and dendritic cell neoplasm, which will have a combination of markers which are expressed on immature plasmacytoid dendritic cells, 123, 303, HLA-DR. So initially it was said 456 combination is for BPDCN. But now we know that there are a lot of markers that characterize this neoplasm, and it can be 4 and 56 negative. So commonly 56 and DR are very bright in BPDCNs. So gating, sir has covered. This slide is important. I have taken it from ICCS. So basically your typical blast region is CD45 dim, low site scatter. Okay. Now blast, where all can they be? Should they be here only, or you should analyze every region of the plot to find blast? Actually, yes. So you will find a normal normal plot will have neutrophils, lymphocytes, monocytes, but look at this plot. Most of the events are lying here only. That means there is some problem with the sample. There are cells which are monotonous, which are lying only in one region, that is abnormal. So this is a blast region, and you should definitely go ahead with your A-lot, further markers for A-lot, after characterizing which lineage markers come in the first tube. So this site scatter is low in the first plot. This site scatter is slightly higher in the second plot, and you can see some of the neutrophils here. So that means there is some maturation. Okay, so they are blast, they have high site scatter, that means they can have granularity or rods, and they are showing some maturation, and you are seeing some lymphocytes. Then there is one plot here, third one, where CD45 shows split in the cell. So there is one CD45 dim population and one CD45 bright population. Okay, so similarly here. So these blasts, so they need not always be CD45 dim. So monoblast or T lymphoblast can have higher CD45 expression. Then similarly here, can you guess which leukemia is this just by looking at the plot? APL. So this is typical teardrop pattern. Okay, so all are normal promyelocytes. So blast and blast equivalence, you have to catch hold of blast equivalents also while analyzing your flow cytometry. Blast equivalents tend to be placed at abnormal positions in CD45 site scatter plot. So promonocytes will be with higher CD45 and erythroblast will have negative CD45. Okay. This we have covered. So this is just which markers are, so we have BALL, TALL, ETP-ALL, AMLs. We have now WHO category has two broad categories, like classification has two broad categories. One is this with defining genetic abnormality, and the second is where there is no defining genetic abnormality, that is morphologically defined. So for morphologically defined, we have to have the knowledge of combination of markers so that we can characterize them for the lineage. So for myeloid markers and monocytic markers, see the difference. So CD34 is usually commonly expressed in myeloid blast. But if you look at the monoblast, CD34 is commonly down-regulated. CD14, 64 are not expressed in myeloid blast, but in monoblasts, CD64 is expressed along with those other monocytic markers. 14 may or mostly it is diminished. Okay, that means they are immature, they are promonocytes. Okay, they can have aberrant cross lineage. If so, there is cross lineage expression of markers, that is blasts at a stage where it should show only immature markers. It can be normal also, no? Bone marrow has normal myeloid blast also. But if it starts expressing cross lineage antigens, or it expresses some marker which is usually expressed in down the line in maturation, then that is abnormal. Simultaneous expression of a mature marker like 11B or 15 should not be there in a blast which is showing bright CD34. Okay, so you have to catch hold of these abnormalities. So what is acute leukemia of ambiguous lineage? Ambiguous lineage, we have defining genetic anomaly, or then again we have, it is very important while doing flow that we identify the lineage. If we are not able to identify the lineage and exclude all the rare markers, then it is acute leukemia, undifferentiated acute leukemia, but acute undifferentiated leukemia. But we should try our best to pick up the lineage, because it's basket diagnosis. Mostly what is labeled as AUL will turn out to be something else when we have the marker for that lineage. Okay, so for example, commonly BPDCN may be mistaken as AUL, but because see, CD56 bright, but nothing else is coming. CD123 is also not coming. Then what will you do? You will label it as AUL. But if you have special markers like 303, 304, or TCL1, TCF4, then you may be able to pick that up. Then it is recharacterized into BPDCN. So it all depends how elaborate your panel can be. So there are various clues to the genotype that you can gain from just by having a look at immunophenotypic patterns of your sample. So we know commonly that myeloid markers are expressed in BCR-ABL positive ALLs, and they are commonly expressed in ETV6 transferrin positive BLLs. Then there can be loss of CD10 and gain of CD15 in KMT2A rearranged ALLs. Then they can be CD40, typically BLL, which is totally CD45 negative, and CD123 very bright is turns out to be hyperdiploid. That is good prognosis. Then you have a category where you find CD34 and DR negative, but it is not APL. Morphology may or may not mimic APL. What can those, and you can find cup-shaped indentations in the nucleus in the blast. So what can be the genetic anomaly in those kind of? NPM, also FLT3 mutation. Okay. So for the residents, can anybody identify what case this is? This is y-axis is CD19, x-axis is cytoplasmic 79A. This is CD10, and this is CD34. Anyone? The blasts are lying in CD45 dim region. They are expressing CD19, CD10, cytoplasmic CD79A. They are negative for MPO, positive for 34, negative for T markers. What is this case? B. Okay, Kala positive, BL. Okay, good. Then this case, CD45 is spreading like this, heterogeneous. Then it is showing cytoplasmic CD3, and CD3 is down regulated. It is showing CD38, CD1656 is not bright, and it is negative for CD4 and 8. What is this? Loudly please. Loudly please. TLL. Okay. Then this. Again. So I'm not sure. Cytoplasmic CD3 is positive, but it is showing CD34 also. CD7 is also positive. Surface 3 is negative, and 4-8 is negative. DR and 34 are positive. Little bit of CD33 also. CD5, if you look at it, it is. So out of the T cell lineage, there is CD3, cytoplasmic, loss of surface CD3, CD7 is positive, but CD5 is negative. What could it be? It is showing stem cell markers and myeloid marker expression. This is early T cell precursor ALL. Okay? What is this? I have written. So MPO is negative, and typically it is showing slow site scatter, CD45 dim, very less neutrophils, and 33 DR is positive. MPO is negative by both flow or cytochemistry. So what is the cutoff of MPO on cytochemistry? Cutoff for MPO positivity on cytochemistry? Yes. भूल गए. Okay, chalo. Then monocytic leukemia. You have to have your morphology and cytochemistry with you while diagnosing a case in flow cytometry. It all works in tandem, nothing in isolation. Okay. So can you guess the genetics here? Spotter. NPM when mutated AML. Okay. Can you guess the lesion here? Yes, what's your name? Dr. Shan. Okay. So what do you think is the morphology showing? Vacuolation. What are these vacuoles positive for? And what is the lesion showing on, actually this gating I am not shown, but I have shown the arrow that I picked up these from here, these cells from here, CD45 negative. And they are showing 117. aberrant CD7, no 34DR, but they are all positive for CD71 and E-cadherin. It is strongly positive for P53. All these cells are more than 80% in bone marrow. What diagnosis is this? Yes, acute erythroid leukemia. And this was positive for TP53 with high VAF. Okay. Again, you want to continue, sir, or we can bus now, sir? Okay. Yeah. Last case, can you guess the lesion? This is, these are the blasts with blebbing. This is CD61 IHC. You can see CD45 dim, CD34DR is positive. What is it positive for? This is cytoplasmic CD42A. Yes. So acute megakaryoblast leukemia. Okay. Okay. Anybody, if that person gets this lesion, then should get a chocolate ideally. What if this M7 shows this marker combination? Then what will you think of? 56 bright, 45 negative, DR negative, 38 negative, 56 very bright. I'll leave it to you to ponder upon. Okay? It's showing megakaryocytic differentiation. 41 is positive. I'll leave it for you to ponder upon. Thank you so much.
Thank you, Dr. Richa, for such a wonderful talk. And I hope the delegates must have gained a lot of insight in the approach for acute leukemia cases. Thank you.
Thank you, ma'am, for that very wonderful talk. You really drew from the earlier message of keeping it simple and simplified a very complex topic for the rest of us. May I now request our chairpersons, Dr. Renu Saxena and Dr. Monica Gupta, to felicitate our guest speaker, Dr. Richa Chauhan.
Felicitation. Felicitation.
Oh, I'll also come.
May I now invite Brigadier Dr. Tathagata Chatterjee to felicitate our guest of honor and first chairperson, Dr. Renu Saxena.
May I now request our most revered professor, Dr. Professor Manjula Jain, to felicitate our other speaker, Dr. Monica Gupta, other chairperson, Dr. Monica Gupta. We are waiting for our respected dean, sir, for the next 5 minutes to inaugurate the ceremony.
So as we wait for the inauguration, which will be, hopefully in the next 5 minutes we'll have that, I'll tell you about a case from ESIC Basai Thara. Dr. Jayati is here, Dr. Sujata is here, and Dr. Rana is not here. So they had sent a case, a fascinating case of a myelodysplastic syndrome, and they had correctly pointed out that this is a prodrome of myelodysplasia, which is converting to acute erythroid leukemia. If you remember our teaching, Dr. Renu and Dr. Pati used to say, unlike other AMLs, the acute erythroid leukemias have a myelodysplastic feature, maybe 6 months, 7 months, and then they convert. So we put this as a letter in 2012 to Ellen Jaffe, and she agreed. Yes, this is actually so the biology of the case is so important. You should not just sit behind a slide and just give a bombastic diagnosis. The clinical behavior of the patient, the biology of the patient is so important, and all of you must go to the clinics and examine patients. You are first MBBS and then an MD in pathology. You all are clinicians first. Okay? So there is no harm in going to the ward, meet your senior residents, examine the patient. This fascinating case, they did the IHC, it was E-cadherin positive and TP53. You see the ICC has now classified acute erythroid leukemia as leukemia with a TP53 molecular mutation, right? Whereas WHO still calls it acute erythroid leukemia. But ICC has completely removed this term. So the TP53 IHC is an excellent surrogate marker for the molecular TP53. And I'm so glad that ESIC Basai has standardized this IHC, and it was fascinating because we went and did the molecular of this patient, and it came exactly the same way that Dr. Jayati, Dr. Sujata's team, and Dr. Rana's team gave it. So a big hand for ESIC Basai.
We are truly grateful to have with us today the Dr. Mandeep Sachdeva, Registrar and Honorable Observer, Haryana Medical Council.
This is my job to wait for, to cover the gaps, So let's have a quiz. So here is a patient who has gone to the gynae department. A lady, 30 years old, with bleeding PV. And Dr. Sharma, who is professor of gynae, he is intrigued that this bleeding PV has got nothing to do with gynecology. Right? And he contacts us. Why is this patient having severe anemia? So one of us answers, oh, itna bleeding ho raha hai, anemia hai. You know what this patient turned out to be? This patient turned out to be acute promyelocytic leukemia. So you will have this unusual presentation. So that's why I'm telling you again and again, see the clinical and the biology of the patient, like Dr. Pati used to tell. Yesterday he was telling us the same case, So clinical features of this patient is so important. So you, the pathologists, are integrating all the other departments. I welcome the Dean and the MS to our August gathering. Thank you so much.
Vidya dadati vinayam, vinayadhyati patratam, patratva dhanamapnoti, dhana dharmam tata sukham.
Knowledge makes one humble. Humility begets worthiness. Worthiness creates wealth and enrichment. Enrichment leads to right conduct, and right conduct brings contentment.
Our conference is graced by the pioneers in the field of medicine and the best of the best. As we prepare to begin the inauguration ceremony of this remarkable event, it is our honor to invite our esteemed dignitaries to the dais.
May I now please invite Dr. Anil Kumar Pandey, Dean Sir, ESIC Medical College and Hospital, Faridabad, to kindly grace the stage.
Next, I would like to invite the Medical Superintendent, EIC Medical College and Hospital, Faridabad, Dr. Sandeep Kumar Sir, to please come up on the stage.
Now I would like to invite the chief guest of the ceremony, Dr. H.P. Pati, to please come up on the stage.
Next, I would like to call upon our guest of honor, Dr. Renu Saxena, to please grace the dais.
I now request respected Dr. Mandeep Sachdeva, Registrar and Honorable Observer, Haryana Medical Council, to please come up on the stage.
It's my pleasure to invite our very own mentor and senior professor and the chief organizing chairperson, Brigadier Dr. Tathagata Chatterjee, on stage.
I now warmly welcome Professor and Head of the Department, Department of Pathology, and the co-organizing chairperson of this remarkable event, our very own Dr. Mukta Pujani, on the stage.
I now take honor in inviting the organizing secretary of the conference, Dr. Deepthi Sidam, Associate Professor, Department of Pathology, ESIC Medical College, Faridabad, on the dais.
May I now request our organizing chairperson, Brigadier Dr. Tathagat Chatterjee, to felicitate our chief guest, Dr. H.P. Pati.
I now request our organizing chairperson, Dr. Mukta Pujani ma'am, to felicitate the guest of honor, Dr. Renu Saxena ma'am.
May I now request our dynamic organizing secretary, Dr. Deepthi Sidam, to felicitate the Dean, ESIC Medical College and Hospital, Dr. Anil Kumar Pandey.
I now request our multi-talented Dr. Aniruna De, Professor, Pathology, ESIC Medical College in Faridabad, to felicitate the HMC observer, Dr. Mandeep Sachdeva.
I now request Dr. Charu Agarwal, Professor, Department of Pathology, ESIC Medical College, to felicitate our Medical Superintendent, Dr. Sandeep Kumar.
I now invite our very dynamic Dr. Shilpi More, Assistant Professor, Department of Pathology, to felicitate Brigadier Dr. Tathagata Chatterjee, Senior Professor, Department of Pathology and Hematology.
Next, I would like to invite Dr. Varsha Chauhan, Associate Professor, Department of Pathology, to felicitate our Professor and Head of the Department, Dr. Mukta Pujani.
I would now request Brigadier Dr. Tathagata Chatterjee to please felicitate Dr. Deepti Siddham, Associate Professor, Department of Pathology.
She has been working tirelessly for the last 2 months. She hates me for all the languages I use. So I have to do it. You're especially good.
We have also got special felicitations for our most revered professors, Dr. Rajkumar Chandok and Dr. Manjula Jain. Dr. Chandok has had one of the longest associations with the ESI family, and Dr. Manjula ma'am's expertise in the subject has been a cornerstone for the development of our department. May I now request Dr. Mukta to please felicitate Dr. Rajkumar Chandok.
I now request Brigadier Dr. Tathagata Chatterjee to felicitate Dr. Manjula Jain.
May I now request Dr. Mukta, Secretary, Haryana Chapter of Indian Association of Pathologists and Microbiologists, to felicitate Dr. Sunita Singh, President, Haryana Chapter of Indian Association of Pathologists and Microbiologists. May I now invite everyone to join us for the traditional lamp lighting ceremony and the formal inauguration of this prestigious workshop. Let us take a moment to honor our collective commitment to knowledge, healing, and discovery. I now request Dr. Neha, Senior Resident Pathology, and her team to seek the blessings of Goddess Saraswati through a melodious Saraswati Vandana.
जय जय हे भगवती सुर भारती तव चरणों प्रणमामः। जय जय हे भगवती सुर भारती तव चरणों प्रणमामः। नाद ब्रह्ममयी जय वागीश्वरी नाद ब्रह्ममयी जय वागीश्वरी शरणं ते गच्छामः। जय जय हे भगवती सुर भारती तव चरणों प्रणमामः। त्वमसि शरण्या त्रिभुवन धन्या सुरमुनिवन्दित चरणा। नवरस मधुरा कविता मुखरा नवरस मधुरा कविता मुखरा स्मृति रुचि रुचिरा भरणा। जय जय हे भगवती सुर भारती तव चरणों प्रणमामः। प्रणमामः प्रणमामः प्रणमामः।
I now invite Dr. Anil Kumar Pandey, Dean ESIC Medical College and Hospital, Faridabad, without whose guidance this conference wouldn't have been possible. Dr. Pandey is one of the founding faculties of our college. Sir is a prolific author with numerous research papers as well as book chapters under his name. He is also the editor of 4 books and the reviewer of numerous indexed journals. He has headed the organizing committees of numerous conferences and workshops. Sir is the Chief Coordinator of Central Research Lab as well as the Molecular Biology Lab of our hospital. He was also elected as General Secretary of Association of Physiologists and Pharmacologists of India. May I please invite Sir now to speak a few words and address the gathering.
Good morning, distinguished guests, speakers, delegates, invited guest of honor, chief guest, medical superintendent, professor and head pathology, hemato-oncology, especially the postgraduate students and all the delegates from various parts of the country. It's my great pleasure and privilege to welcome you all at this national conference and workshop dedicated to flow cytometry in hematopathology at ESIC Medical College and Hospital, Faridabad. Especially, I am grateful to honor and welcome Dr. Renu Saxena and Dr. H.P. Pati. Both are stalwarts in their own field to grace this occasion as a chief guest and guest of honor. So, welcome you both at ESIC Medical College and Hospital, Faridabad. So we are honored to host such a esteemed gathering of the researchers, clinicians, technologists, thought-provoking leaders, and all united by a common goal to advance the science and application of flow cytometry in understanding, diagnosing, and managing hematological condition. I am also grateful to Dr. Mandeep Sachdeva sir for gracing the occasion on behalf of Haryana Medical Council. We know that the quality of any of the workshop, conference or panel discussion is based on the how you gain the credit hours. So definitely credit hours is one of the parameter to assess the quality of any of the conference or workshop. So definitely Department of Pathology and Hematology deserve a big round of applause for getting 8 credit hours. And we know that there is a structured parameters to accord the credit hours by Haryana Medical Council, and if they gain 8 hours, definitely it said that the this conference is going to be a quality workshop and conference. So especially I am really grateful Dr. Sachdeva sir that he also visited on previous occasion at this institute on behalf of Haryana Medical Council and also as a clinician. So let me show that if you join the conference for 2 days, you see that the 8 hours credit accorded by HMC is very, very much justified by, because the quality of training and workshop and hands-on will be given itself will say that the credit hours award is very, very justified. Now let me say about few other stalwart. Professor Brigadier Tathagat Chatterjee, you see how he project the junior people, and without taking the credit of anything for him, whether it comes to projecting in front of the national fora or in the institution, when it comes to publications, research work, he always project the junior staff, and that all other junior faculty sitting here, they should learn that when someday they will be the HOD or professor, they should project their junior staff, because I know, because he does not require any acknowledgement or recognition. So when a new faculty join, he came to introduce her, see Dr. Silpi or Dr. Sidam, see they are working a lot, so and we should appreciate their work and acknowledge their work. So this is way how to, run a department, so that everyone should be motivated and feel that they are working under a very good mentor faculty whose guidance will upgrade the department. So definitely, and sir, role is not only to head a department, you see whether it comes to bone marrow transplantation, the hematology department, the pathology department, even he himself taken initiative to start the OPD also for some of the hematological cases, and he is doing that throughout. Dr. Mukta Pujani, definitely she is here since the first batch of MBBS admission, so she is helping not only the department of pathology, but lot of administrative responsibility has been given to her, and she is doing justice. So definitely means with the lab services, when it comes to integration of the lab, the quality services, because institutions credit means whether what is your parameters, the quality of services, especially ESIC is going to improve the quality, because it's now mandatory to go for NABH, NABL. So we have the full-fledged NABH for our research lab, ethics committee, and then the blood bank, hematology, and entry-level NABH for hospital and NABL for the lab. Now she has taken initiative to, we should go for full-fledged NABL as well as NABH. So definitely, whenever there is assessment of the quality of services, it is very difficult or impossible if your diagnostic services is not upgraded. So this is where we differ from many of the hospital. Our diagnostic services, be it the radiological or pathology, hematology, microbiology, entirely central lab. So where we make a difference with the other hospitals. You know that I have just few of the data related to pathology. In the last 1 year, we did more than 21 lakh hematological tests, and more than 1.5 lakh clinical pathology, more than 10,000 histopathology, and more than 5,000 cytology. I am not getting out biochemistry micro because it's a pathology conference, but you see we are getting patient not from Delhi NCR, but for this especially flow cytometry from across North India, Delhi NCR and beyond also. So this is the first probably among the all ESIC where flow cytometer has been installed, and very soon we will see that we are working to progress to molecular genetics in hematology, and very soon NGS platform also will be operational. And I am sure that once Professor Chatterjee and Dr. Mukta Pujani are, definitely hematology and pathology will be one of the best across India, not only in ESIC. So definitely the purpose of such type of conference is training, so that more people should be trained, the postgraduate student, maybe up to resident and assistant professor. So ESIC currently means across India, you see that we have covered around 16 crore population under ESIC and more than 4 crore card holders, and planning is to upgrade it to 50 crore coverage. Means unorganized sector already it is labor code has been in place, gadget notification awaited, and you see that ESIC has a huge health infrastructure expansion because they plan to start 50 more medical college. Already 10 applied to NMC, and for 2026 another 10 has been identified. So in next 5 years 50 medical college and 100 hospitals has been planned, because you need a huge health infrastructure to cover 50 crore population. 50 crore means almost 40% of the entire population. So definitely ESIC set example that how medical college, if we are going to start, this is the way to start, and these are the benchmark where all the diagnostic services, the clinical services, and teaching and research. Obviously, when I see the quality of the research, the number of projects, the number of publication, Department of Pathology lead from the front, and other departments also doing the same. So definitely without Department of Pathology, many of the clinical department cannot complete the research. Obviously, so your diagnostic services are better, so you will excel in clinical as well as research also. So definitely I am sure that today's scientific event will be very helpful and informative. All the delegates coming from across the various parts of the country, and we will continue to start some time such type of conferences and CME with the Department of Pathology, and support from ESIC will always be there. Again, I congratulate team pathology and organizing committee members, office bearers of this conference, and wish a grand success of the event. Thank you very much.
Thank you very much, sir, for your guidance, support, and leadership. Every great gathering is marked by a voice that inspires, a mind that ignites, and wisdom that transforms life. It's my singular pleasure to introduce the chief guest of the ceremony, Dr. H.P. Pati, presently the Dean of prestigious Indian College of Hematology. Sir has been one of the most popular and brilliant professors of pathology in the prestigious AIIMS, Delhi. During his tenure as the president of Indian Society of Hematology and Blood Transfusion, the society has made remarkable progress. As an editor-in-chief of the esteemed Indian Journal of Hematology and Blood Transfusion, the impact factor and the international status of the journal has grown leaps and bounds. Sir has also been past president of Delhi Society of Hematology. Sir is known for his scholarly excellence and morphological expertise by all his students, including our very own organizing chairperson, Brigadier Dr. Tathagata Chatterjee. It's an honor to request Dr. H.P. Pati to deliver the keynote address on flow cytometry in hematopathology. We are truly privileged to have you with us, sir, today.
Dignitaries on dais, the senior invited teachers, students, and the delegates. It's a great pleasure to be here at the National Conference and Workshop, Flow Cytometry in Hematopathology, being organized at this hospital with the guidance of Dr. Tathagat, our student, and his team here. It's really good to know that with his guidance and expertise and commitment, we are seeing a large number of the audience have shown their interest to understand, to take forward the flow cytometry in hematology, diagnosis of the hematology. The Indian Society of Hematology, Blood Transfusion, and the Indian College of Hematology, only recently, last 2-3 years, have looked more to expand in the field of the academics. It is not just we are happy with just having conference and workshop during the annual conference, but throughout the year and smaller places. Of course, I will not say FARIDABAD is a smaller place, but definitely the more focus is always at Delhi. The conferences are always held at Delhi. But why not the NCR? The NCR has to get its own recognition. And this, particularly this type of the college, medical colleges here, which are doing excellent work, as the Dean has already given the details. So these people should know by holding the conference and what much type of acumen is there, the faculty of the department already have. So that is the motto. Coming to the flow cytometry, look at how flow cytometry has become an integral part of hematology. Just before the flow cytometry was used for diagnosis of acute leukemia, imagine what are the diagnosis of acute leukemia depending on? It is depending upon simple the cytochemistry. And I do know how much the error in diagnosis of cytochemistry if based only on all the cytochemistry, just the MPO or NSC. You would get 20% diagnosis error because most of the acute myeloid leukemias, a large percentage of acute myeloid leukemia are negative for MPO. Are they not myeloid leukemia? Because the treatment, unless you give the lineage is so important. If you give a wrong diagnosis, the treatment will, if you say non-myeloid, the treatment will be lymphoid. Obviously, you don't expect the myeloid patients will respond to the lymphoid. So flow cytometry application has tremendously changed the improvement in actual management of acute myeloid leukemia. Particularly if it is myeloid, is getting a myeloid treatment, then only we can know the prognosis, the responsiveness, true responsive or non-responsiveness to a leukemia. So these are again in the flow cytometry has become a gold standard of a large number of the hematological tests. So much must be in other fields as well. So the application of a flow cytometry is so important in both not only the diagnosis but also in several spectrum of disorders in both benign as well as the malignant hematology. I'm sure in 2 days of the conference we will not only stimulate you but will give you that direction to take it forward in your workplace, and many of you will be able to start it. Thank you. I wish a great success.
Thank you, sir, for that insightful keynote. It's said that better than 1,000 days of diligent study is one day with a great teacher. I now call upon one such teacher whose name needs no introduction in the field of hematology, Dr. Renu Saxena. Dr. Saxena was the former head of the department Hematology at the prestigious All India Institute, New Delhi. Ma'am was also the past president, Indian Society of Hematology and Transfusion Medicine. May I now request her to speak a few words on the overview and scope of hematopathology in India.
So thank you very much. Respected dignitaries on the dais, colleagues, friends, and delegates. A very good morning to all of you. At the outset, I would like to compliment Dr. Tathagat and his team for organizing this very relevant and very well-attended workshop that we have here today. So I will just take you through what has happened to hematology till now and what is about to happen, and then what are the kind of training programs or anyone who wants to pursue in hematopathology a future, then what one can do. So briefly, when we started off, that's about 3-4 decades back, hematology was hemogram and maybe a PT, prothrombin. That's about it. Maybe BT-CT, which is today not really even advocated. As time went on, we went into more morphology, peripheral smear morphology, bone marrow aspirations, automation came, went into automated coagulation analyzers, automated hematology analyzers. A large number of hospitals and colleges today have this kind of a hematology setup. As part of MD training, the MD students are exposed to this kind of hematology. But then as time went on, it progressed further. So from morphology, we incorporated immunohistochemistry and immunophenotyping by flow cytometry, as Dr. Pati said. So we have today flow cytometry for not just diagnostics, but also a prognosis of various hematological disorders. Moreover, now we have also progressed on to use of flow cytometry for monitoring of oncology, hemato-oncology patients in the terms of MRD. So minimal residual disease for ALL is available, AML is available, multiple myeloma, CLL, everything it's available today. And that makes it easier to tailor the treatment of these patients. In line with this in keeping with this kind of a tailored precision medicine, cytogenetics came into forebeing. So we're familiar with karyotyping for ages together, but karyotyping takes a long time. Many times the result comes later, your patient has to be treated immediately, so you needed something more quicker, and so FISH came. So then karyotyping and FISH made it much quicker. So today, when we suspect APML, we do FISH. Morning we suspect, 4 hours we have our FISH results saying PML RARA is positive. So that's how you expedite the diagnosis, and then you tailor the treatment to that particular disease. So with FISH, simultaneously molecular genetics came, and today a lot of hematological malignancies during their classification require not just cytogenetics and FISH, but also require molecular genetics. So today we have targeted molecular tests which are available, which can be actually set up in a lot of colleges. And then of course we move on to something which is the next generation sequencing. So next generation sequencing a lot of people are doing today, and that also comes into hematology, and one can actually again precisely find out what is the target which needs to be addressed during treatment. Apart from this, NGS has moved further from just small targeted panels. It's moved on to exome sequencing. So previously genome sequencing was the hallmark, but genome sequencing is very expensive and it gives you so much of data which is really irrelevant. So we move on to more targeted exome sequencing. Exomes, as you know, are the total coding genome of the body. So that has something like 23,000 genes, but then that again gives a lot of data which may be irrelevant. So we've come to more targeted. You have a clinical exome sequencing which covers about 6,000 genes, which is for all clinical hematological conditions, not just hematological, most clinical conditions it is there. We move on. We keep going, move on. We're moving on to predictive medicine, and so actually every day one is progressing, not just in molecular genetics, different kind of probes for FISH, different technologies, moving on to microarray technologies. You have gene-based chips which are available today. So a hematologist can actually keep progressing with these advancements in science. So if we see what are the options that are available for an MD pathology person, if they're interested in hematology. So they will eventually have access to all these technologies which are there, which will be very actively involved in patient care. So today we have post-MD, we have DM hematopathology, which is available in AIMS and PGI Chandigarh as of now. We have also fellowships in hematopathology. For example, CMC Vellore is running a fellowship in hematopathology. SGPGI is running a fellowship in hematopathology, and at Medanta we are also running a fellowship in hematopathology. Apart from fellowships, you have small trainings. So you have access to trainings in a particular specialty. You can have training in, for example, cytogenetics or FISH or molecular hematology. So one can do your MD, can then go on to specific trainings. If you want to go particularly into, let's say, cytogenetic hematology, have your trainings and then move ahead in that. So there is a lot of scope, and also it is not that you finish these trainings and you are everything. You're never everything, because science is advancing so fast. all of us have this opportunity to keep getting ourselves trained in the newer technologies that are coming and keep moving forward in the field of hematology. So with this, I hope I've motivated some of you who are at the crossroads of what to do after MD. I mean, one option that you can think is also hematopathology. Of course, finally, it depends on what your interest is, because I feel you should have a passion for what you want to do if you want to do well in any subjects. So with these few words, I hope you enjoy this workshop, and it's a very nicely drawn out program. You have very good faculty. Please make full use of this and ask all your clarifications so that when you go back from here, you can actually set it up in your institutes, or you will be able to do an independent flow cytometric analysis of all cases. So wish you all the best and have a great workshop. Thank you.
Thank you, ma'am, for the pearls of wisdom and insights on the scope of hematology. I'm sure all the residents will benefit from this motivation and information. And now, it's an honor and privilege to welcome Brigadier Dr. Tathagat Chatterjee, who needs no introduction. He is dynamic, knowledgeable, highly established, and ever-inspiring senior professor of pathology.
Thanks a lot. So the high tea is waiting, right? We all want a cup of tea. But this is an occasion for me to thank Dr. Pati, Dr. Renu Saxena, my Dean, my MS, and Dr. Sasdeva, who has come all the way from Chandigarh. At the same time, I want to thank the stalwarts from Banaras Hindu University, Dr. Vijay Tilak, Dr. Deepa Rani, and so many others, the AIMS faculty, the delegates, all of whom you are here. You are going to have a feast. We are going to teach you. We are passionate about what we are doing, and we have, divided your workshop into the benign and the malignant. Day one is benign, day one is malignant, and day two is benign. The benign is so important. Particularly, Dr. Jagdish Chandra is here. The immunodeficiency disorders, A gamma globulinemia. There's so many things you can pick up by flow. So we're going to discuss that tomorrow. We are also discussing PNH tomorrow. And we've got a plethora of speakers from all over NCR who are all experts in the field. So make full use of it. I am also aware that by the time we give a vote of thanks at the end of the conference on day two, there's no one to hear you. So I'm going to give my vote of thanks also now, okay? So that we can concentrate in a conference. So I thank my co-chairperson, Dr. Mukta, the most dynamic pathologist and a brilliant head of the department. And she has been with me throughout these 2 months, and really, we have what we had to do, right? And we have done a great job. Dr. Deepti, really give her a standing ovation. She has worked day and night to put this conference where it is. Not only that, she is officer in charge of a flow cytometry, along with Dr. Shilpi, who is here. Would you like to stand up, please, Shilpi? We want people to see you. Yes. So Dr. Shilpi, Dr. Deepti, and Dr. Mukta, Dr. Charu, who is here, who set up the molecular genetics. We are going to discuss that later. The NGS, next month we are starting our 126 gene panel NGS in AML. I'm very proud to say that the Dean has always supported us, and whatever, he just wants a direction, and if he is convinced, he gives it to us. So we are one of the first ESIC hospitals and one of the very first government hospitals to do so many things under one umbrella. Last but not the least, I have to thank Dr. Manjula Jain, Dr. Chandok sir, Dr. Sunita Singh, Dr. Monica, so many people, and a person who has always motivated me, Lieutenant General Gupta. Would you like to stand up, sir? The handsome man. Yeah. He was doing his PhD under Professor Seth, virology in AIMS. And I came running because I had got a fellowship program in CMC Vellore. He told Tathagat, DM karna hai. Okay? I still remember. Ya kuch mat kar. So that's how I landed under Dr. Saxena and Dr. Pati and did my DM. I became their first student. So thanks once again, and let's go and enjoy ourselves with a hot cup of tea. Thank you.
Thank you very much, sir, for those inspiring words. We now request all the delegates and dignitaries to please proceed for high tea. being served right outside the hall. We'll reconvene shortly for the next academic session.
ऐश्वर्या मैम ओके ओके एक टेबल पे रखना है वहाँ पे।
बहुत अच्छा कर रहा
मिल गया।
ठीक है, इसी में आप एक बार देख लो, जो आपने अभी भेजा था वही है ना लेटेस्ट वन।
उसके लिए मुझे सिर्फ एक लास्ट स्लाइड चेक करना है। आई थिंक दैट इज इनफ।
आपने जो पहले भेजा था,
ये वो वाला है।
आपने जो पहले भेजा था वो ही वाला आपने दोबारा भेजा है या आपने चेंज किया है? हाँ,
मैं
तो
दे रही हूँ आपको
वो
change।
पेन ड्राइव में है हमारे पास। अच्छा, आप भी लाएंगे तो आप किसी डाल देते हैं। आप जब तक टी ले लो। हाँ,
आई एम गुड।
ले लो।
अरे आई एम गुड।
ओके, एक सेकंड। अच्छा,
दिस वन।
आप इसको चाहे तो मान डेस्कटॉप पे करो।
डेस्कटॉप पे रखे थे ये वन स्पीकर्स में। यही वाला है ना मेरा एग्जिस्टिंग प्रेजेंटेशन।
प्रेजेंट तो ये है। आप जो पेन ड्राइव में लाए हैं उसमें चेंजेस हैं, आप ये कह रहे हैं ना? हाँ, तो
थैंक यू।
आप ये रख दो।
मैं कर लेती हूँ। आप चिंता मत करो मेरी। आप चाय पी के आओ। जब आप ऑर्गेनाइज कर रहे होते हो सो मच दैट यू आर डूइंग। खुद का खाना पीना सब।
वो तो मैं छोड़ रही हूँ, लेक्चर के बाद ही करूँगी जो करूँगी। आई एम हियर ओनली।
ऑनेस्टली
नो, आई एम हियर।
थिंक वी आर।
आप तो सब कुछ कवर कर रहे हो तो मुझे कुछ बोलना ही नहीं है, तो आई एम तो जस्ट
आई
डूइंग।
कैन
मैंने सबको यही बोला।
मैम आपका हो गया कॉपी?
मैंने तो कॉपी कर दिया। अब ढूंढो कहाँ है।
नहीं, ये रहा, इट्स हियर।
हाँ चलो मैं चेक कर रही हूँ। आप लोग चलो। आई विल कम।
दिस इज योर प्रेजेंटेशन ना?
दिस इज़ माई प्रेजेंटेशन बट दिस इज़ नहीं, दिस इज़ नॉट माई फाइनल प्रेजेंटेशन। मैंने अभी जब कॉपी किया था वो कहाँ है।
डाउनलोड में क्या होगा? आप मैम जो आप इसी को ओपन कर दो, आप इसी में से ही प्रेजेंट कर देना। नेक्स्ट आप ही का है ना?
ये वाला है मेरा
हाँ, मैंने-
प्रेजेंटेशन। इसको डिलीट कर देते हैं।
हाँ, मैंने यही
ताकि-
वाला खोला था, सेकंड वाला। इसको नहीं खोला मैंने, पुराने वाले का। हाँ, यही ओपन किया था मैंने। आप इसी को ओपन रहने दो।
ठीक है। हमने देखा नहीं है। अरे मैडम आपने देखा नहीं तो भी आप तो अच्छा आ जाओ देख लो।
मैम एक मिनट, आपको आपका पेन ड्राइव देना है।
बंद कर देती हूँ मैं। मैम का खुला है ना
मेरे पास छोटा
मेरा
उसमें एक ही
क्या मैम
नहीं मैम प्लीज
बताओ।
प्लीज पेन ड्राइव। थैंक यू।
मेरा एक्चुअली मेल किया था डॉक्टर ज्योत्सना। हाँ प्लीज़।
डॉक्टर ज्योत्सना का गर्मा, डॉक्टर ज्योत्सना का अभी इसमें आ गया है या फिर पेन ड्राइव में है? पेन ड्राइव में अभी मैम पेन ड्राइव में इस पे लगाएंगे। आप टाइम के बाद है ना, इसलिए भी हमने डाला नहीं है। जस्ट आपको इसमें देखना है मैम?
हाँ नंबर 5 होगा ना।
फटाफट बुलाओ उसको या तो। हाँ 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 हो गया फिर। शालिंदर जितनी तुम्हें नई दी है ना वो एक बार डाल दो। ये वाली तो डल गई है। ये है अब डॉक्टर जसमेता की, इसको रीनेम कर दूँ? रीनेम का ऑप्शन क्यों नहीं आ रहा है इसमें?
क्या है?
अरे सेकंड वाली, माइक बंद करो एक तो तुम पहले। रुक जाओ, छोड़ो छोड़ो, कोई नहीं है अब।
पर एक माइक सर को चाहिए, सर कह रहे हैं। मैम नहीं,
भी ऑन नहीं है, स्क्रीन भी ऑन नहीं है। हो गया, हो गया। एक और भी करो, दूसरा ये भी करो। स्क्रीन
आ गई थी, आई थी मैम जब आपने से किया था ना।
हाँ, आ गई।
A very good afternoon, everybody. I hope everyone is recharged after the high tea. As we look forward to our session 2 on principles of immunophenotyping in CLPDs, including comprehensive flow panel designing, may I now please request Dr. Sunita Singh, Senior Professor and Head, Department of Pathology, PGIMS Rohtak, to chair this session. Ma'am has contributed immensely to this field and has been a distinguished guest speaker in numerous national and international conferences. Ma'am is a prolific author with more than 200 publications in renowned journals. She is the most popular amongst her students and has guided and co-guided more than 50 postgraduate students till date. Ma'am, we are honored to have you here with us.
Thank you. Thank you so much for the introduction.
I now take the privilege of calling upon Dr. Jayati Chakraborty, Director, Professor and Head, Department of Pathology, ESI PGIMSR, Basai Darapur, to chair this session. Ma'am has had a distinguished career spanning various states from West Bengal to New Delhi. She was formerly in the West Bengal Medical Education and a former professor at ESIC PGIMSR, Maniktala, Kolkata. She has authored numerous prestigious national and international journals and is a remarkable teacher. Let's please welcome Dr. Jayati Chakraborty with a huge round of applause.
I now request Dr. Sunita Singh to introduce our first speaker, Dr. Jasmita Das, who will be guiding us through the session on principles of immunophenotyping by flow cytometry in CLPDs.
Hello, good morning, still good morning or good afternoon. Dr. Jasmita, she is an associate professor of hematopathology at AIIMS New Delhi. Her academic qualifications are DM Hematopathology, and earlier she was at Gangaram as consultant and Medanta as associate consultant, Gurgaon. Her area of interests are flow cytometry, hemoglobinopathies and coagulation disorders. And today she'll be speaking on principles of immunophenotyping in CLPD. As you all know, CLPD is a heterogeneous group of disorders, and many a times it becomes very difficult to differentiate them on just simple morphology. And then how the flow cytometry can help that. Please, Dr. Jasmita, welcome to the dais.
Thank you for the kind introduction, ma'am. So, in the while that they're sorting out this technical glitch, I would like to thank Tathagat sir, Dr. Deepti, Dr. Shilpi More, and Dr. Mukta for calling me here. I'm sure the team here has worked very hard to ensure that all of you enjoy today's meeting. So, before I start, let me ask the delegates a question. How many of you are postgraduates? Please raise your hands. So, most of you are postgraduates. How many of your institutes have a flow cytometer? So I see a lot of hands. How many of you actually get to work on the machine? Great. So at least I'm not seeing 0 hands. I'm happy about that, because that is one thing where, thank you. Thank you. जी सर, धन्यवाद. So it is heartening to see that at least a few of you are using the machine, and are the rest of you not using it because there's no access or because you're scared? How many of you are scared? Let's raise hands. Nobody is scared? An occasional one. Not of yours. I don't mean scared of your faculty. I mean scared of the software, the machine, losing the gates. There are a lot of reasons why we can be scared, right? And it doesn't harm us to admit that we are scared. The best way to learn technology is to use it. So trust me, I hope that after today's meeting, you'll realize that everything is generally retrievable on a flow cytometer, that you can play around on your equipment. So I'm going to talk to you about CLPDs, and I'm going to show you cases. If the time runs short, I'll stop on the cases so that you don't sleep here waiting for your lunch. So the outline will be the following. We'll talk about BCLPDs in the beginning. Following that, we'll talk about T-cell lymphomas. So when we talk about chronic lymphoproliferative disorders, we start with a case history. We always have to examine the patient. The key findings are lymph nodes present or not, liver present or not, spleen present or not, any other lumps present or not. The first minimal evaluation is at least do a CBC and a peripheral smear. There are a lot of clues of CLPDs that you can pick up on the peripheral smear. After that, the next step usually comes as immunophenotyping. That is, if you have gross involvement, if you're seeing abnormal lymphoid cells on the peripheral blood, you will straight away move to immunophenotyping. If not, in that case, then a further evaluation is with imaging. You may require biopsy of the lymph node masses, and in certain cases, you may have to send out special investigations, including FISH and NGS, for specific translocations. Okay. Okay. Glitch. Not moving. Okay. So why do we want to subject CLPDs to immunophenotyping? The first is there are situations where all of you would have seen certain reactive lymphoid cells. You were not sure whether they were just reactive or whether they were abnormal. So this is one tool that can help you distinguish reactive from CLPD. It is a tool that can help you assess clonality of the B cell population by seeing the kappa lambda split on the B cell population. In addition to that, you can also look at the clonality of majority of the T cell disorders if you use TRBC1. Lineage typing, whether it is a BCLPD, a TCLPD, or a NKCLPD. Then the further classification of the individual CLPD. It also helps you determine therapeutic targets. So how many of you have seen rituximab being used, or you know of rituximab being used? It is a molecule that's against which CD antigen? 20. So it is a molecule against 20. So this tells you whether the cells are expressing 20 or not. And rituximab is just a small example. For myeloma, there is daratumumab, there is isatuximab, and the list is ever growing. So we also have realized that a lot of our PRCA and cold type AIHA cases may harbor B cell clones. So this is another, and T cell clones as well in PRCA. So this is another one of the indications for which we do flow. And there are times when the morphology looks very blastic, but you're not sure whether it is truly a blast or whether it is a large abnormal lymphoid cell. So that is another situation where flow comes to our rescue. It has prognostic role as well. So in CLL, there are specific markers that are associated with poorer prognosis, and you will have the next session on MRD in BALL. In CLPDs as well, for hairy cell leukemia and chronic lymphocytic leukemia, flow can help you pick up MRD. I'll show you one example of that as well. So what are the samples that you will get for CLPD? You will get peripheral blood, you will get bone marrow. Some of you may get FNA from the lymph nodes. Very few of us do get solid tissues. So if you have a solid biopsy of a lymph node coming to you, it has to be freshly done. You cannot have a formalin fixed tissue. You have to have living cells. Then you have to mince the tissue, because flow is only for single cell populations. To convert that tissue into single cell, you have to mince it, pass it through the mesh and get your tissues. You have to understand the germinal center and non-germinal center patterns if you're going to specifically look at lymph node FNAs or lymph node flows. Those. देखो बेबी. So the gating approach on FNA of a node or lymph node biopsy, if you're passing it through a mesh, is slightly different. I'll show you one case of that as well. In addition, any effusion fluid, if there is a suspicion of a lymphoma involving the abdominal ascitic fluid or the pleural fluid or even CSF or pericardial fluid, those are the samples that can also come to you. Now, flow is a tool in which forward scatter is a measure of the relative cell size. So if you see this example, I've just tried to show you that the normal lymphoid cells are over here. These are light blue in color, and your abnormal cell population here is colored as green. So it tells you that the abnormal cells are larger than the small lymphoid cells. This is just a small clue that your flow also gives you at times. So for BCLPDs, this is the long list of B-cell lymphomas that is published in the WHO classification. Among the ones in the boxes are the ones which we commonly end up seeing in the BCLPD category. This includes CLL and the monoclonal B-cell lymphocytosis, hairy cell leukemia and other splenic lymphomas, lymphoplasmacytic lymphoma, follicular lymphoma, then mantle cell lymphoma. These are frequent spillers into peripheral blood. Among the large B-cell lymphomas, you can see marrow involvement, and you can actually pick up peripheral blood involvement and marrow involvement by flow cytometry. Burkitt's is another category. Increasingly, we are seeing that flow is being now done for myelomas and for plasma cell dyscrasias of all the various types. So this is another graph which multiple people will show you. This is about the normal B-cell maturation. So if you actually see this graph, the question that I want to ask you is, what should be the gating marker for BCLPD, the starting marker, by which you at least identify or you characterize that, okay, this is likely to be a B-cell. So I've given you a list. Can you pick out one? Perfect. So CD19. Why not CD20? If you look at the CD20 molecule, in any mature cell, it is still expressed. It is only the earliest hematogones which don't express CD20 at all. But why don't we use CD20? So post-rituximab, it is lost, yes. And there is notably one particular CLPD which very frequently spills into peripheral blood, that is CLL. So CLL can have very, very heterogeneous, very, very low expression of CD20. So therefore, CD19 is an accessible marker that you can look at. So any change in the sample being evaluated, I mentioned that for lymph nodes, the GC and non-GC have to be separated. So for that, 19 and 20 combination can be used as a start point. So now this is the standard way that you process your samples. There are three ways to process samples. You stain everything, stain the sample, lyse it, wash it, and then acquire. Or you do a lyse wash, stain wash, that is RBC lysis happens first, followed by washing, staining, and then acquisition. Or it can be large volume washing of the entire peripheral blood sample that you're going to submit. Then you stain, then lyse, and then wash. Now, for BCLPDs, I told you clonality is by kappa and lambda. So, which of these three modalities would you do if you have to process kappa and lambda? So, if you have to process kappa and lambda, stain, lyse, wash becomes an inadequate modality because your sample can have free light chains. It can have small M bands circulating. So, you don't want your anti-kappa and anti-lambda antibodies to be sequestered in the plasma. Therefore, you have to wash your sample first in a high volume wash. So Euroflow protocols typically recommend that 300 microliter sample to be submitted for flow is washed with 10 ml of PBS. So that's a very large amount of washing. Or you can also do lysis of the RBCs first, followed by a couple of washes, and then you can start your staining protocol. So what is the impact? I'm showing you a case and example here. So if you actually have a look, the dark blue ones are the B cells. So in this particular sample, if you actually have a look at this, 1920, so these are B cells essentially. B cells don't seem to express any kappa or lambda. So this was without that excess wash. If you process it without wash, in the same sample, you can actually clearly see clonality for lambda here. So this is one thing which we keep in mind. There is one more key point about kappa and lambda, that is on the antibody selection. So for kappa and lambda, monoclonal or polyclonal is a question. For flow, which antibodies do we prefer, monoclonal or polyclonal? Generally, for any flow cytometry situation, any marker, any antibody that you order, it is usually a monoclonal antibody. However, for kappa and lambda, it is actually a polyclonal reagent that is preferred rather than monoclonal reagent. And so usually kappa cells, they sort of exceed the lambda positive cells. In addition, the normal value, if your kappa to lambda ratio is less than 0.5 or it is more than 3:1, that suggests clonality. Now this dictum will work if you have majority of lymphoid cells, but if you have a low number of lymphoid cells, then this dictum sort of fails and you cannot pick up small populations of abnormal B cells in a background of polyclonal B cells. So the standard algorithm that we follow when we approach a case of BCLPDs, you start with 19, pick up the 19 positive cells, see if 5 is positive or not. If 5 is positive, look at these markers. So 200 bright, 23 positive, 20 can be dim or heterogeneous, surface light chains will be dimly expressed, 79B can be either dim or negative, ROR1 can be positive, and in this case the diagnosis will be CLL. The other situation can be when the case is CD5 positive but 200 is completely negative, and the other markers of CLL, you have a brighter expression of 20 surface light chains and CD79B, in which case you are possibly looking at mantle cell lymphoma. So which is the classical translocation for mantle cell lymphoma? 1114. So be loud. The only thing that we can do when we answer is we can either be right or wrong, and we are in a workshop, nobody's going to judge us for being wrong. So 1114 is the classical translocation for mantle cell lymphoma. Now moving on to the 5 negative cases, you assess for CD10. CD10 is a marker of germinal center. So all the GC lymphomas, be it follicular, be it DLBL or Burkitt, everything will be usually CD10 positive. The situation that you need to remember is Burkitt's presents many time in childhood. So childhood, the other CD10 positive lesion is an acute leukemia, the BALL cases. So you have to be conscientious of the fact that you will have to rule out a BALL if you're thinking of a CD10 positive lymphoma which has a blastoid morphology. Now 5 negative cases, again look at CD10. Among the CD10 negative cases, you have the following differentials, which is splenic marginal zone lymphoma, hairy cell leukemia, the splenic B cell lymphoma with prominent nucleoli, and lymphoplasmacytic lymphoma. There is a list of markers, among which the first 5, they are very strongly positive in a case of hairy cell leukemia. Now, typically, a patient of hairy cell leukemia is supposed to have a large spleen, cytopenias, monocytopenia. But trust me, there are situations that you will find that there is no splenomegaly and you may have hairy cells. So, again, don't just rely on the clinical history. You have to keep the entire picture in mind. Now, CD180 is a marker of marginal cells, and usually it is more brightly expressed in marginal zone lymphomas. So with this basic concept, let me show you a few cases so that you can see how the concepts are applied. So on this PS, what is your diagnosis, or what would you suspect if you have to sign out the PS? So what are the predominant cells? Lymphocytes, right? Lymphocytes. What about, say, this here? What is happening to these? These are smart cells. So all of you have picked up the finding. I think I'm going to show you a CLL. You also know I'm going to show you a CLL. So what is the name of this classical morphology of the nuclei? It's the soccer ball chromatin or the football chromatin. Okay, so these are the standard housekeeping gates that you will start with. So you essentially pick up the singlets, you exclude the debris, and you have the 45 side scatter. Don't worry about the gray color, you're going to see more colors. So beyond that, you start your gating. You've gated your lymphoid cells, you've gated your granulocytes. So 19 and 3 are the separators. If you look at my panel, it has 19 and TCR gamma delta. Now this was a Euroflow tube, and they have a 12 color and 8 fluorochrome approach. So they bundle a few markers together on the same fluorochrome on the presumption that the markers are expressed by different cells. For example, TCR gamma delta will not be expressed by B cells, but it will be expressed by T cells. So gamma delta cells will be here, and this will be your B cell population, and this is the T cell population. So we choose to just color them like that. Subsequent plots, the B cells here, they seem to express kappa, correct? Kappa is dim, and how can I say it is dim? Because I know where my normals lie. So I'll show you the subsequent cases where you realize that my normal kappa and lambda are at, so kappa normally is at this location. So this appears to be expressing all kappa. Here we have looked at the 56 on the rest of the lymphoid cells. So we have NK T cells that are expressing both CD3 and CD56. Now after excluding our B cells, we are looking at the pure NK cell population. So now the B cells, do they express CD5 on this plot? Yes or no? This is a workshop, guys. Yes. So it does express CD5. So you're looking at a 5 positive BCLPD. What are the two common differentials? CLL and mantle cell lymphoma. So these are the two common differentials. So 20 seems to be very spread out, right? So 20 is like very wide. It's not forming a very uniform, very homogeneous cluster. It's like sort of spread out. That is the kind of 20 that you have. I already showed you that they were kappa clonal. Now I'm showing you lambda as well so that you can confirm that, okay, all the cells are expressing kappa. If you actually have a look at this plot, there are certain small subtle features. So this is where my normal B cells lie. So therefore I know that this kappa is dimly expressed. For lambda, it is on FITC, that is a relatively dimmer fluorochrome, so this is where my normals lie. So I know that the cells, they are expressing a dim kappa and they are not expressing lambda at all. So to pick out the normal B cells, you can increase the size of the dots, or you can lower the resolution of the plot depending on the software that you're using, so that you can pick out the normal cell populations as well. Now CD38 was one of the prognostic markers for CLL. So here the CLL cells that don't express CD38 at all. So essentially I've shown you a CD5 proliferation with a dim kappa expression. With that, what would you like to see? Further markers for CLL, right? So you look at the further markers. How about 200? Very brightly expressed, isn't it? So 200 is very homogeneously expressed on your malignant cells. CD23 also is positive. In addition to that, what about 79B? It is very dim and practically negative. And CD43 is another marker which is very frequently dimly expressed in CLL. So the answer here is CLL. Did you need flow to diagnose it? You picked it up on the PS, right? Correct? But that will not be there in all the cases. There can be atypical findings. Sometimes things that look like CLL may not turn out to be CLL. In CLL, you can have transformations for which you will need flow as well. Now this is the next case. This is a patient with a supraclavicular node and intra-abdominal lymphadenopathy and lymphoid cells of this morphology. Any clue about the lymphoid morphology? There is a notch or a possible cleave in the nucleus, right? What about these cells? So the one on the right, does it look like a blast? This one? It looks very blastoid, right? If this was the lecture with Dr. Tathagat or Dr. Richa, you would have called this a blast and you would have thought I'm going to show you possibly an acute leukemia, right? So this is a blastoid cell, but you also have to remember that there are cells like this as well. So this is a case in which a few of the cells had blastoid morphology, the other lymphoid cells were showing a bit of cleaving. We again start the same approach, and here you have the B and the T cells gated. So no expansion of the T cell compartment, NK T cells or NK cells. What about CD5 on the B cells? B cells are pink in color. Again CD5 is positive, right? 20, now you can see in the CLL 20 was spread out. Here it is forming a single tight cluster, so that is the 20 expression. 38 again is negative on the B cells, and the B cells now look at the kappa. Earlier kappa was also spread out, it was dim. Here kappa is expressed at a very bright intensity. So on the initial screening tube itself, I know that I am not dealing with a CLL very likely, because 20 is very bright, kappa is very bright. So we look at the further markers. 200 turned out to be completely negative, 23 is negative, 43 is negative, and 79B is exceptionally brightly expressed. So this is a case of mantle cell lymphoma. So mantle cell lymphoma versus CLL, the plots on the top you have mantle cell lymphoma, and the plots on the bottom are plots from CLL. So this is the kind of difference in the marker expressions and the patterns that you can see. So this was the Matute score, the Murrow modification of the Matute score, which was extensively used, except for the fact that in my lab now we no longer use FMC7. And this score has stood the test of time because it has worked for mantle cell lymphoma and CLL differentiation for a very long time. But now there are more markers that we have at our disposal which possibly would work better. So CD200 is at least one of those markers which is exceptionally important to differentiate mantle cell lymphoma from CLL, because it is brightly expressed in CLL while mantle cell lymphomas, they tend to be negative. There are different ways of scoring systems that have been developed, equations that have been developed. We've also assessed it, so we find that in addition to CD200, CD43 is one more useful marker. So ERIC and ESCA initiative was to standardize CLL phenotyping. So in that, they've also dropped FMC7. Why? Because FMC7 was nothing but an epitope of CD20. So therefore, CD20, what the information is giving you is nearly the similar information you would derive from FMC7. Therefore, it has been dropped from the list of essential markers as recommended by these two organizations. In addition, you see that they've added the further list of markers, which are the recommended markers. If you are going to get samples for MRD in CLL, the recommended markers are what you would want to include in your panel so that you can pick up MRD better. In addition, as I mentioned in the beginning, CD148 is a marker which is a marker for mantle cells, and it is strongly expressed in mantle cell lymphoma that can also aid in differentiating between these two CD5 positive lesions. The rest, the third one is the 180, which is strongly expressed in splenic marginal zone lymphoma. So we go back to our case of mantle cell lymphoma. This is the bone marrow biopsy. So if, say, you didn't end up flowing it, these are the two IHC markers that you could do, cyclin D1 and SOX11. Both of them were found to be positive in the lymphoid aggregates, and we confirmed this on the FISH to be 11-14 positive. Now the last CD5, so-called list of CD5 positive lesions, but in this case 5 was negative, was a CLL in which you did not have CD5 expression. So there can be CLLs which sometimes lack CD5, but they have typical marker pattern for all the other markers. This is one such example. This is a 5 negative CLL. One more thing that this case shows you is that if you look at kappa and lambda, it seems like there is no kappa lambda restriction in CLL. So since in CLL there's a very dim expression of light chain, sometimes despite all the washings, all the polyclonal reagents, everything that you do, you may not get a nice light chain restriction. Now this is another case. So we are finished with the 5 positive lesions. Now we're going to see the 10 positive ones. So on the morphology, I showed you the notch in the mantle cell lymphoma. Would you consider this a notch or a cleave? Cleave. So the classic buttock cell that you read about, that is the typical cell of a follicular lymphoma. So the lesion that I'm going to show you, you would expect 5 to be negative, which it is. 20 again, very uniform, very bright. Kappa, exceptionally brightly expressed. So till now, not 5 positive. So among the 5 negative lesions, you look at the further markers, you realize that 10 is nicely positive. So 10 is positive, 79B is nicely expressed. In addition to that, you have CD200, but here 200 is sort of variable, ranging from dim to intermediate. So the best utility of 200 is when you have to distinguish CLL from mantle and hairy cell leukemia from the other splenic lymphomas. So 43 was negative. Hairy cell markers, they were also negative in this case. So CD10 positive lesions, what differentials did I talk to you about? Follicular lymphoma, DLBL, and Burkitt's. Morphology, absolutely not like Burkitt's. Presentation, not like Burkitt's. Again, not an aggressive presentation. So very likely, we are looking at a follicular lymphoma. Classical translocation for follicular lymphoma? 14-18, perfect. So Now, this was one case which we got and we were really stuck on it because this was a patient who had undergone multiple lymph node biopsies. I would not want to diagnose a follicular lymphoma purely on flow. I would like to see a lymph node biopsy as well. So now this patient had only abdominal nodes. Multiple core biopsies had been all reported as follicular hyperplasia. So we received a FNA. Now FNA for this patient was interesting. Why? Because if you look at the 19 and 20 plot, what do you see? Do you see two splits here? Do you see a very bright 19, 20 dim population? And do you see a 20 bright but 19 sort of heterogeneous population? Okay, let me color these two areas. So if I color these two areas, now you see, you're able to see two different things over here. So let us go further. So the kappa lambda plot, if you see, the green cells which are very bright 20, they don't seem to show any light chain restriction, do they? No. The pink cells on the other hand, do you see some kappa lambda? You see something, right? So it seems like the 20 very bright cells are negative for kappa and lambda. Should that be the case? We only saw dim kappa lambda when we talked about CLL, which is a 20 very dim lesion or a 20 heterogeneous lesion. So we ran it with the polyclonal markers. At this time, we were using a commercial LST tube. So commercial LST tube had monoclonal kappa lambda reagent. So we used the lab panel, which had polyclonal kappa lambda. Now what do you see? The green cells, do they express lambda or are they clonal for lambda? So the green cells, 20 bright cells, are now showing lambda clonality. The pink cells, they are still polyclonal, as you could identify even in the earlier panel. So this is how it looked, and now you have the green cells which have moved from negative into the lambda positive space. So basically you do have clonal B cells on your hand in one of the populations that you studied. So the message that I want to get across to you is that for kappa lambda you have to use polyclonal reagents. The lack of surface light chain expression on a 20 very bright population is also abnormal. It is not normal for a 20 bright cell to not express kappa or lambda. So with that, I'll quickly show you one case of DLBL. So this was leukoerythroblastic blood picture. And here in the 19-5 plot, you see that 19 positive cells are present. They don't seem to express any 20, correct? Only 38. So can they be plasma cells? Normal plasma cells? This is a normal peripheral blood, okay? So unlikely that the entire population is that of plasma cells. It can be, but unlikely. So what are the atypical features here? The kappa lambda again seems to not have worked. So the kappa lambda on this entire population again seems to be negative. So these are the atypical features. Lack of 20, no kappa lambda, and a bright expression of 38. So we went back to the patient. The patient had prior history of rituximab therapy. So therefore, CD20 may be entirely lost upon rituximab therapy, and this is something that we have to keep in mind if the pattern of 20 is lost. This you will see increasingly in ALL cases as well, because in acute lymphoblastic leukemia also CD20 is being employed for therapy. So these were the further markers. The lesion was 10 positive. 79B was expressed. So 79B and 20, they sort of go hand in hand. So if you have complete loss of 20 but you still have 79B, think of rituximab in a case of CLPD as well. So again, we did polyclonal kappa lambda and we realized that lambda was clonal. So GC lymphomas can lack clonality using monoclonal reagents because there are ongoing somatic hypermutations in the germinal center. So your monoclonal reagent may not be able to pick up that particular rearranged clone. So therefore, polyclonal reagents have to be used. Now this case is an interesting one and I hope that you can pick up the morphology. Can you? So I'm trying to show you cells with projections, with hairy projections, right? So this looks like a hairy cell leukemia on the gross. You have circumferential projections in the cell with some bilobed or reniform nuclei like you have. So what about this graph? So how many of you are familiar with this particular graph from a cell counter? How many of you walk towards your cell counters or you just look at the printouts? How many of you have walked towards the cell counter? Raise your hands. Sarika ma'am, don't scare them. So So a lot of you. That's good because there are plots and these colors will not be apparent to you if you're looking at printouts alone. They will be apparent to you only when you walk over to your machines. So what is this green population for all the people who raise their hands? The green population on the plot. Grands are here. I've cut down one. Monocytes, right? Monocytes. So I show you hairy cells and I show you monocytes. Is it supposed to happen? No, monocytopenia in hairy cell leukemia, correct? So how am I seeing so many monocytes? So the scatter properties of hairy cell leukemia, they are very similar to monocytes. So if you have hairy cell leukemia spilling into your peripheral blood in decent numbers, it will actually sit in the monocyte region. You will not see monocytes on the morphology, but your counter will be tricked into thinking there are monocytes. So this is what this case was. Now on the top here, I am showing you gating for lymphoid cells. We have picked up the B. And then I've extended the gate of the lymphoid cells up. So this is the part I've done. Lymphoid cells are here. Now I've moved the lymphoid gate a little up to include part of the monocytic region. What do you see? Do you see an increase in the number of dots in the B cells? From the top to the bottom? The number has increased, right? Yes? Which means that B cells are sitting in this particular region, which is otherwise the monocytic region, or it is just like a cap above the lymphoid cells. Now this is an area where classically hairy cell leukemia goes and sits because the scatter properties, the cells, they have ribosomal lamellar complexes. Those complexes lead them to have that kind of internal complexity that they land up in the monocytic region rather than the pure mature lymphoid region. So this is like a cap over this. So you see 19 nicely expressed. 20, I think among all the cases that I've shown you, this 20 is like the brightest 20 that you can see, correct? Very, very bright. And they are all showing lambda restriction. 79B is positive. 200 again is positive. Hairy cell leukemia is one of the brightest 200 positive lesions. In addition, there is CD10 expression in part of the cell population, and this was peripheral blood that was run. So hairy cell leukemia in about 5 to 10% cases can express CD10. Now this is another one of the cases, and what I want to make you realize here is that in addition to the hairy markers that are positive 103, 11C and 25, 200 very, very brightly expressed, and you see all of them are nearly in the monocytic region. So if you concentrate only on the standard area of lymphoid cells, you may miss this. So always use a MNC gate in the beginning, then you gate further. MNC gate will include your lymphocytes and it will include your monocytic region as well. So remember about hairy cell leukemia, they sort of lie as a cap above the T lymphoid cells. They will express the hairy cell markers 103, 123, 11C, etc. They have the brightest CD200 expression and may express CD10 in 5 to 10% of the cases. Now I think I'm nearly at the end of time. So hairy cell leukemia is an example where you can do MRD assessments as well. So this was a case that was submitted at the time of MRD, and we could pick up both a 103 positive and a 103 negative suspect population. At this time also it retained the same marker profile. It was still in the monocytic region. Okay. So Tathagat sir, I think I need to end here and leave T out. Okay. So in the interest of time, I'm not talking to you about TCLPDs. If you can remember the B and you can continue with that, it's great. If in the workshop we get time, I can teach you TCLPDs as well. Don't look so sad, darling, please. There is one audience member, she's looking very sad. Trust me, in the workshop, I will teach you T. Don't worry about it. Okay, with that, I end and I thank all of you for a patient hearing.
Thank you, Dr. Jasmita. Thank you, Dr. Jasmita. I was going to tell that time is over, but I thought the students will hate me then. So they will hate me if I stop you.
Thank you, ma'am, for that all-encompassing yet well-rounded talk. Up next, I request Dr. Jayati Chakraborty to introduce our second speaker, Dr. Deepti Sadam, who will be further delving into the topic of comprehensive panel designing in CLPDs.
Good afternoon. So it's a pleasure to welcome our own Dr. Deepti now from the ESI fraternity. So she has completed her education from prestigious institutes in various states, and she has published a book chapter also. She has many publications to her credit, and after the beautiful presentation of cases by Dr. Jasmita, she will walk us through the comprehensive panel designing in chronic lymphoproliferative disorders. So over to you, Dr. Deepti.
Thank you, ma'am. अब मैं कहाँ खड़ी होंगी ऐसे? चलो कोई नहीं, मुझे नहीं चाहिए. कैसे होगा ये? कैसे करेंगे? इधर देखो. क्या हो रहा है मुझे तो? अब इसमें करें? नहीं, ये तो नहीं चाहिए था. चलो, yes sir. So, good morning, good afternoon, respective seniors and my dear colleague. Okay. So today I am going to discuss about the comprehensive panel designing in chronic lymphoproliferative disorder. So what do you understand by panel? So panel is the combination of antibody tagged to different fluorochrome that enable us to characterize our cell of population. In simple word, you can say that it is a combination of marker and which will answer your question that you want to answer. So there are 5 important steps in the panel designing. The first is define biological marker, which will include your lineage marker. What are the specification of your instrument? How will you select antibody and fluorochrome? What are the resources for panel design? And finally, test refine your panel. So I will discuss all this one by one. The first is define biological marker. So first of all, this is the biological aspect. You should know what you want to do and how you want to do. So for this, you should know the key cell population. For this, you need lineage marker. These are the pivotal for identify your population of interest. For example, if you want to study the T cell, you will need CD3. If you want to study the B cell, you will need CD19. The next is, you should know your instrument. What is the specification of instrument? It will include your fluorochrome, antibodies. So you should know what is the optical filter configuration, how many lasers are there, what are the laser wavelength available for excitation, how many number of detector per laser were present, and filter available for detecting specific fluorochrome. And if possible, always go for the cytometer with more laser because it will decrease spectral overlap, and especially when we are dealing with the large panel, it will decrease spillover and enhance the resolution. So make sure the performance and calibration of the cytometer are optimal. Now, how will you select your antibody and fluorochrome? Choose well-documented high-performance clones. Choose fluorochrome to minimize spectral overlap. Chart the expression level of your marker and choose fluorochrome with an appropriate brightness. You also remember the cross-lesion excitation and fluorochrome spillover. So here in this graph, here this is the excitation spectra of APC and BV650. In y-axis there is relative intensity and in x-axis this is the wavelength. You will see from this plot that APC will not be excited by the 405 nanometer laser, and the exact excitation spectra of the APC will start from the 450. And this is the emission spectra.
in which you can see both the fluorochroma overlap with each other, and the band pass filter for both the fluorochrome is same. So here, this is the uncompensated form which shows the cross laser excitation, and this is after the compensation. You will see the difference. So compensation required both pre-acquisition and post-acquisition. Now, pairing the antigen with respect to intensity expected, as already said by Dr. Tathagat Chatterjee sir in his lecture, that dim antigen, bright dye, bright antigen, dim dye, and in co-express marker, one with dim intensity, spread of other markers matter. Then how will you select the marker? That will depend on how many needed to answer your question. Suppose if you want to go for the lymphoma panel, you will need CD19, 45 for gating. For T cell, you will need CD3 and T, and for clonality, you will need kappa lambda. So depends on your population of interest, you will choose your marker. So there are 3 types of marker expression. Primary antigen, which is expressed in high density. For example, CD4, which is brightly expressed in CD4. CD4 is brightly expressed in T cells. It is moderately expressed in monos, and it is negative in neutrophils. So basically, this is a lineage markers. Secondary antigen, which is expressed over a continuum, means sometime it is present low, moderate, and bright. Tertiary antigen, these are the critical marker, which is low express antigen. For example, when we assess the tree regulatory cell, we will use the CD127 and BV421 fluorochrome. So basically, these are critical marker, which is low expressed. You should know the gating strategy needed to be identify the population, and also the choice of clone is important, and you will use the published literature for that. The final is from where you get the panel resources for panel design. The first is, of course, the published literature, and according to your experience also. And there are multiple websites where you can see the antibodies across the multiple vendor. That is BioCompare and FluoroFinder. Then final, the review, test, and refine your panel. Titrate all antibodies and fluorescent dye to minimize noise. Proper control, compensation control, and unstained tube control will be good. The prepare antibody cocktail, try to avoid the single pipetting, because cocktail will avoid or reduce the pipetting error. Change antibody clone or fluorochrome if needed. Make adjustment if something is optimal. So the panel for assessing CLPD was selected based upon the proposed Euroflow panel of antibodies with some modification and customization as per availability of antibodies. A primary lymphoma screening tube was designed using 10 antibodies with an aim to identify the major subset of B, T, and NK lymphoid cells. So the BCLPD Euroflow antibody panel consists of 4 tubes with lymphoma screening tube, and each tube in the panel contains the backbone markers, which is CD19, 20, and 45, which is efficient to identify the all clonal B cells. And with these backbone markers, you combine the characterization marker to provide an efficient differential diagnosis among the majority of CLPD entities. So what is the utility of flow cytometry in BCLPD? This already discussed by Dr. Jasmita, that we need to diagnose the CLPD, whether it is reactive or lymphoma. Of course, the typing, whether it is B or T, and further subclassification for prognosis, and nowadays for MRD also. So flow cytometry has a well-established role in the diagnosis evolution of mature lymphoma. Its role is in effective lineage assessment, aberrant antigen expression, and immunoglobulin light chain restriction. So limitation must be appreciated with regard to morphology, ISC, cytogenetic, and molecular testing. So the two basic role is assignment of lineage and distinction between the neoplastic and non-neoplastic B and T cells. So for the identification of B cell lineage, we use CD19, 20, 22, and 79. CD19 is a gating marker, which is expressed in both mature and immature B lineage cells. And all mature B cell neoplasm express CD19 except the plasma cell myeloma, plasmablastic lymphoma, and primary effusion lymphomas. Now CD20, it's evaluate the maturity pattern in bone marrow, but it decreased or down-regulated in CLL, SLL, and it is negative in anti-20 therapy, that is rituximab. CD22 is variable with bright expression in hairy cell leukemia, and you use at least 2 markers to define lineage. Now, identification of abnormal mature B lymphoid cells by immunoglobulin light chain restriction and by aberrant antigen expression. So here in dot plot you can see kappa and lambda. Some of the cells shows both lambda and kappa positivity. It denotes that it is polyclonal or reactive. But in this dot plot you will see most of the cell shows kappa positivity. That means kappa restricted. So the normal kappa to lambda ratio in a non-disease state is 2:1. But a ratio if outside the range of 3:1 or 0.3:1 is generally considered to be monoclonal. Now some light chain caveats are there. Some B cell can be light chain dim or negative, like in case of CLL and SLL. Germinal center B cells, blast cells, hematogones. Not every monotypic population is lymphoma. It could be monoclonal B cell lymphocytosis or in situ follicular neoplasia. Expression of aberrant antigen. So aberrant antigen means the expression of those antigens that are not normally present, like expression of CD13 and 33 in B lymphoid cells, expression of CD5 on B cells, and expression of BCL2 in CD10 positive cells. These are aberrant antigen expression. So the monotypic light chain expression is not only the indicator of B cell neoplasm. Aberrant pattern antigen expression and other lineage expression is also a clue. Flow cytometry data should always be interpreted in context of morphology and clinical features. So approach to a case of CLPD, it is very well explained by Dr. Jasmita ma'am. So I will not go in detail. So this is the primary panel which includes these markers. If CD19 mature B cells which show CD5 positivity or CD5 negativity, with further evaluate for differentiation between the CLL and mantle cell lymphoma in addition to antibodies like CD23, FMC7 or CD10, or the mature B cell neoplasm which show CD5 and CD10 negativity. We further differentiate into hairy cell leukemia and splenic margin zone lymphoma with the addition of characteristic immunophenotype that includes CD25, CD103, 11C and 123. If CD19 is negative, you will suspect the case of T, NK and plasma cells. So if T cell, you will put CD2, 3, 4, 8, 7, TCR alpha, beta or TCR delta gamma. For NK cell, you will put CD16, 56 and 57. For plasma cell, you will put CD38, 138 and 56. Again. So now I am closing this. So these are the panel for CLPD. This is the 4-color panel and this is the panel which we are using here. So here we have the gating marker CD19 and CD45 and CD20, which is the backbone marker for CLPD. And for clonality, we are using lambda kappa. And for further characterization into the CLPD entities, we are using other marker also in this. You can see 11C, 138 for plasma cell, 11C, 103 and 25 for hairy cell leukemia and on. Thank you very much.
Thank you, Dr. Deepti, for the very lucid presentation. It can never be possible to cover all aspects of CLPD within this short period of time, but I'll advise all our residents to make maximum utilization of the workshop so that you can clarify all your doubts during that period from the faculty. Thank you.
Wonderful presentation by both the speakers. So that blast cell Jaspita showed. in the CLL case. And she gave a hint that Richa's or my seminar. What was that blast cell in the CLL case which Jasmita showed? And she said that it is technically not the leukemic blast. What was that blast cell? Hematocone. So what was that blast cell? Suppose you get a blast-like cell which is 5 negative, 23 negative, strong 20 positive, kappa restricted. And it looks like an acute lymphoblastic leukemia. And then you see patient got a big spleen crossing the umbilicus. Nahi. And now it is called splenic lymphoma with a prominent nucleoli. Great.
May I now request our chairpersons, Dr. Sunita Singh and Dr. Jayati Chakrabarti, to felicitate our external speaker, Dr. Jasmita Das.
May I now request Dr. Tathagat Chatterjee to please felicitate Dr. Sunita Singh.
I now request Dr. Mukta Pujani to felicitate our chairperson, Dr. Jayati Chakrabarti.
Moving on to the session 3 on concepts of measurable residual disease in B acute lymphoblastic leukemia and comprehensive panel designing in MRD-B ALL and its clinical utility. We request our very own Dr. Brigadier Tathagat Chatterjee to chair this session. Sir is an alumnus of the prestigious...
To chair this session alongside Sir, it's my pleasure to also introduce Dr. Sarika Singh, Senior Professor, Department of Pathology, Maulana Azad Medical College, New Delhi. Ma'am has shown her remarkable academic skills by being National Faculty of DNB and paper setter for various entrances and exit exams and super specialty exams. Ma'am has over 90 publications in various esteemed international and national journals and has won the Best Paper Award in many conferences in India and abroad. She is the Vice President of Faculty Association and Co-Chairperson of Protocol Committee of MAMC. She has done a research fellowship in hemato-oncology from the prestigious AIIMS New Delhi and is an executive member of Delhi Society of Hematology. Let us warmly welcome Dr. Sarika Singh.
May I now request Dr. Brigadier Tathagat Chatterjee to introduce our first speaker, Dr. P. Lalita Jyotsna.
Thanks. So before I introduce Dr. Lalita, I was the actual chairperson with Dr. Sarika was Dr. Sunita Sharma. She is presently the Director General Health Service. So mother's got sick, so she couldn't come, and really we really miss her presence, and we hope that her mother recovers quickly, right? So this is Lalita Jyotsna. She is one of my very dear friend, and she's a professor and consultant in the hematology lab at Lady Hardinge Medical College. She's got 12 years of experience in hematology, including immunohematology and transfusion medicine. And not the least, she has got so many publications. I've read a few myself. And her interests range from hemoglobinopathy to immunodeficiency and, of course, flow. And I still remember her getting the best paper award in one of the annual chapters where she standardized one of the very, what is that, CD81 to what? Which antibody?
86.
CD86. So she had standardized CD86 and she got the best paper award. That was a fascinating paper. Over to you, Lalita.
Screen touch next.
Touch me.
Touch is next. Screen touch.
Thank you so much, sir, for the introduction. So I would like to thank Tathagat sir and Mukta ma'am and the organizers for having me here. And so I'll be talking about the concepts of measurable residual disease in BALL. So let us start with some trivia. So for the trivia buffs, I think that this would be easy. So who is this gentleman? So this is the same gentleman in both the pics. In the first, that is on the left-hand side, you can see him with a microscope, and in the other, he's with a child, young child. So any idea who he is? So he's Dr. Sidney Farber. He was a pathologist, and he's known as the father of modern chemotherapy. And he went on from looking at the leukemic blast cells under the microscope to trying to eradicate them using antifolates and bring out remission. So he was the one who paved the way for research into multi-pronged and multi-phase chemotherapy, especially in pediatric BALL. So basically, I have shown this slide to highlight the role of a pathologist in hematopathology and in the management of the hematological malignancies. So once we have diagnosed BALL based on morphology, cytogenetics, immunophenotyping, the next step is risk stratification, followed by risk-adapted therapy. Now, we know this WHO Hem5 classification. So it involves basically the B lymphoblastic leukemia lymphoma, not otherwise specified, and of course, those with defined genetic abnormalities. Now, what is the therapy? Okay. Most well characterized in BALL in children, of course in adolescents and young adults, and these include the various protocols, including the BFM-based ESIC, UCAL, GMOL, etc. Now, this is a multi-phase therapy involving multiple drugs, which include the induction, consolidation, interim maintenance, delayed intensification, etc. Of course, now we have targeted therapy like TKIs for the Philadelphia positive ALL, immunotherapy, and of course the transplant. Now, before we start therapy, risk stratification is important. So, taking the example of the ESIC protocol, risk stratified into standard risk, intermediate risk, and high risk. Now, what are the factors based on which this stratification is done? These include various levels of stratification using various risk factors which have been proven to affect the prognosis. So at baseline, we have age, TLC, testicular involvement, CNS disease, and bulky disease. Then at day 8, we have the peripheral smear blast count, the CSF for CNS involvement. Meanwhile, we have the cytogenetics report, and based on that, whether we have favorable or unfavorable prognostic cytogenetics, we re-stratify. So at the end of the induction phase, we define complete remission. So here begins the role of course, the pathologist's role is continuous in the management of ALL. So we look at the recovery of the counts, and also a bone marrow is done, and less than 5% blast in the bone marrow on morphology, recovery of counts, no extramedullary disease, all these constitute complete remission. Okay, so now we have risk stratified. What next? So at the end of post-induction therapy, we look at the MRD report. And if it turns out positive, so clinical significance is at 0.01%, if it is more than 0.01%, we re-stratify it. So standard risk and intermediate risk with MRD positive at the end of post-induction therapy will be upgraded to high risk. And then we will intensify the therapy. So now that brings us to the question, why are we doing measurable residual disease? What exactly is it, the definition? So why are we doing it? What is the clinical significance? How? What are the various methods? And what is the role of multiparametric flow cytometry in the evaluation of MRD? The sample required and methods, when, that is, what are the time points of performing MRD, and approach and calculations, and challenges and perspectives. So what is MRD? It was earlier called as minimal residual disease till the end of the late 2010s. Now it is mostly called as measurable residual disease. So measurable residual disease or minimal residual disease, abbreviated as MRD, refers to residual leukemia cells that remain following the achievement of complete remission. So at the end of complete remission, we have said that on morphology there are less than 5% blasts, but MRD goes beyond that and detects blasts below the limits of detection of conventional morphology or microscopic assessment. So why are we doing it? So this is the ideal situation. So this is the ideal situation that we have when we provide a cytoreductive therapy, that the leukemic cell number, it is eradicated at the end of therapy. Okay, so majority of the patients do achieve complete remission, but 1/3 will eventually relapse. Now, why is that happening? This is attributed to the submicroscopic levels of leukemic cells that are lurking in the bone marrow, which lead to relapse. Okay, so this would represent the diagnostic morphology full of blasts, very abnormal. And at the end of the induction therapy, this is our aim: nice looking marrows, regenerated, all cell lines are normally present. But what actually happens is, what we are looking under the microscope is not the complete truth. In fact, there's the issue of the hidden blasts lurking within those normal cells. And those are the ones we are trying to pick out, because those are the ones which are responsible for the relapse. So another question is, more than 5% blasts, even if we say, are they really blasts? So you must have been taught about the difference between hematogons and the blasts. So can anyone tell me which of this is represents hematogons and which are blasts? What is the difference? Anybody? Yes, you want to answer? So yes, theoretically we are told, and of course it is easy to identify at times, that the hematogons, they are larger, they have a homogeneous chromatin, no nucleoli, all these things. But many a time it is very difficult to differentiate between the two. Okay? And of course there are other kinds of blasts, myeloid blasts and everything, which become very difficult. So this is the need for MRD assays. Now what is the clinical significance of measurable residual disease? Now there have been various studies done in pediatric patients, more than the adults, of course. So this is one meta-analysis by Berry et al. In 2017, and this studied this included various studies in pediatric population as well as in adults. So here you can see they had 16 studies with 2,065 patients studying the EFS, and for OS there were 5 studies with 806 patients. And all of these studies unanimously said that MRD positive, so these are the survival curves that you see, so MRD positive had poorer outcomes as compared to MRD negatives. So this is the event-free survival. You can see it is much lower for MRD positive cases than the MRD negative, and similarly for the overall survival, even that is low. Okay. So similarly in the pediatric patients as well, so MRD is associated with worse survival and poorer outcomes. Okay. Also, conventional therapy, there have been studies which say what is the level of MRD and how does it correlate with the risk of relapse. Even in the transplant setting, patients with MRD both prior to and after transplant are more likely to relapse. So now, I hope I have been able to tell you that MRD is both a strong prognostic marker. So you know what a prognostic marker is? That which tells you about how the patient will fare, what will be the course of the disease. And it is also a strong predictive marker. So it will be able to tell us about what is expected, what is the expected outcome of specific treatment, etc. So remember that the currently accepted clinical level of significance for MRD is 0.01%. Okay. So what are the time points at which you do the MRD assessment? What was my purpose of telling you about the treatment and all? Because, yes, so this is done at the end of induction and end of consolidation. So within the various protocols, this is when it is usually done. So end of induction, that is at day 29 or 33 or 35. Some take recovery or complete remission as the time point where the MRD assessment is done, followed by end of consolidation. Okay, so this is just to show the ESIC protocol. So these are the various, what all we have to do, what all, yeah. So here you can see that flow cytometry based MRD should be done basically at the end of induction and end of consolidation. So what are the methods of MRD estimation? Now, we have seen that light microscopy is not a very good way of establishing whether there is remission or not. Okay, so it is just the tip of the iceberg. So we need more sensitive methods. So over the past 20 to 30 years, more and more sensitive methods have developed, which have been able to, establish the MRD. So these include the multicolor flow cytometry, the real-time PCR, and the next generation sequencing. Now, some more detail about the methods of estimation of MRD. So, most commonly used is, of course, the multi-parametric flow cytometry. And while in Europe, the molecular diagnostics, that is the PCR-based MRD, has also been very commonly used. So, about multi-parametric flow cytometry, what we are doing is, basically, we are performing the immunophenotypic analysis to detect the abnormal expression of specific antigens. Here the sensitivity goes down to 10 to the power of -4. That is the ability to detect one leukemic blast in 10,000 cells. Okay, so this is a rapid method, and there are various approaches, of course, which I'll be talking about. That is the LEIP and the DFN methods. So it's a widely applicable method. Now the second, of course, is the molecular diagnostics, in which basically it can be the ASO PCR or the RTQ PCR. So what is the target? You are either doing the immunoglobulin or T cell receptor gene rearrangements or the fusion transcripts, which you have already identified. Okay, so it's a specific method, quite sensitive. So while flow has a sensitivity of 10 to the power of -4, here it goes down to 10 to the power of -5. Okay, but the problem with that is that patient-specific primers may be required, or the presence of fusion transcripts is required. So the applicability is not as high as the flow cytometry, and it is also time-consuming. Now, of course, we have even more highly sensitive methods like the next-gen sequencing, where from 10 to the power of -4, 10 to the power of -5, now we have come down to 10 to the power of -6. That levels of sensitivity we are talking about. Okay. And one word about the next-gen flow. It's not that the flow cytometry, the work is ongoing to make it as sensitive as the next-gen sequencing, or at least as the molecular diagnostics. Okay, so now we'll talk about the multi-parametric flow cytometry in the analysis of BMRD. So it's the most commonly used method, and it detects the abnormal cell surface markers on leukemic cells using fluorochrome tagged antibodies. Now, it has evolved over the past 30 years from 3 colors to 6 colors to 8 colors. Now, sky is the limit, so we have flow cytometers with 12 colors and more. So more markers, more colors, more cells analyzed. This is only increasing the sensitivity of MRD analysis. So what is the sample that we have to take? Whether peripheral blood or bone marrow. So for BALL, remember that leukemic blasts in MRD sample are 1 to 3 logs higher in the bone marrow than the peripheral blood. So yes, bone marrow should be preferred, and bone marrow is mostly taken. So the sample is, of course, the EDTA or sodium heparin, and you need to process it as soon as possible. Now, one word about hemodilution, MRD quantification. Look at the numbers that we are talking about. 1 in 10,000, 1 in 1 lakh. That is the kind of sensitivity that we are talking about. So hemodilution is not at all tolerable in this. So MRD quantification is highly sensitive to hemodilution because of the number of leukemic cells is divided by the total number of cells. Therefore, any hemodiluted sample may report an artificially low proportion of leukemic cells or may give a false negative report. Therefore, what is to be done, whether you are the ones doing the marrow or your clinician is doing the marrow, it is important that always the first pull bone marrow aspirate should be taken. And it has to be not more than 5 ml. So 2 to 5 ml is the optimal sample and the first bone marrow sample. The later samples will be hemodiluted. Processing and acquisition, of course, there are various protocols, but the preferred one is bulk lye stain wash protocol, not going into the technical details of this. Now, how many cells do you acquire? So a minimum of 5 lakh and a maximum of 50 lakh events must be acquired. In leukemias, you may see that this blast count is so much, even 1 lakh cells, if you acquire, that's enough. But here, it's not so. That is why you need to acquire a large number of cells. Now, antibody panels, of course, will be spoken about by Dr. Astha, but let us briefly talk about them. So this is the panel that we have. So here on the top, we have the diagnostic panel for BALL. Here below is the panel that we have for BMRD. So this is the 12-color panel. You can see these various fluorochromes and the various markers that we have. So this was a previous panel. And 5 colors, that is lower number of colors. And so we have had to use more number of tubes, 25 antibodies. You can see this is being repeated in every tube. So it was more expensive and less sensitive. So we are happy with the 12-color panel. Now coming to analysis. Okay. So in order to be able to do BALL flow MRD, this is very, very important because, there is a lot of overlap. So you need to first understand the phenotype of the normal B cell precursors, that is hematogons. So hematogons are a problem in morphology, hematogons are a problem in flow cytometry as well. But once you understand them, it becomes easy to identify. So this is the maturation that you can see from early to late. And remember this picture. Has anyone seen Vidya Balan in Hum Panch? Yes? So this is her evolving over time. So this is the picture that has been often shown, and Dr. Jasmita ma'am has also shown you this. And why this is important, why is this being shown every time is, this is something you need to keep it in your mind to know what is the difference between the hematogons, the expression of markers of hematogons, and the blasts. So okay, so let us talk about this. So hematogons, as you know, the most immature ones are the stage 1. Then you have, as they mature, they're called as the stage 2. And of course, the stage 3 are basically more of the mature lymphocytes. So this is basically the basic markers that you have, which will help you differentiate. So as they mature, the CD34 is lost. CD38, again, the expression becomes less. 10 expression is lost, so it is maximum in stage 1. CD20, as we have seen earlier also, so it's not expressed in stage 1 and it becomes bright in stage 3. CD45 expression varies, and CD19 was the one, as ma'am also showed. It's the one that is constant, that is why it is the gating marker for us. Now, analysis of MRD has 2 approaches. So first we have identified the normal hematogon pattern. So one is the leukemia-associated immunophenotype, okay, and the second is the different from normal, okay. So life is not so easy as I have shown in this picture, so we have to be very careful. Now, what are these 2 approaches? The LAIP is the leukemia-associated immunophenotype. So it requires the identification of leukemia phenotype. You should know what was the phenotype at diagnosis. So it aims at identification of cells with predefined immunophenotype, and then you're looking for the same markers, cells with abnormal cells with those same markers in the MRD specimen as well. But the problem with this is there is immunomodulation after therapy, especially with all the immunotherapy that is being given. Even without that also, even with the conventional therapy, there is a lot of immunophenotypic shift that takes place. So so some markers which were overexpressed initially, they will be underexpressed, there will be loss of expression, and vice versa. So this approach may not be helpful all the times. So we look at a deviation from normal pattern. This does not require the pre-treatment IPT results. So it can be applied to almost any case, but the shortfall is that it requires a lot of experience, and still some cases may be missed, and there may be mimics, various mimics, which we won't be talking about right now. So best thing is, the best approach is a combination of both. Okay, so remember this, what I call the 3 ACEs of LAIP or DFN approach. First is abnormal expression, second is aberrancy, and third is asynchronous expression. So abnormal expression is either underexpression or overexpression of a few markers. So these are the markers, so CD73, 58, okay? Some of these are underexpressed, some of these are overexpressed. Aberrancy, cross-lineage markers. That means, say, myeloid markers being expressed in the lymphoid cells. So CD304 was one that we did last year. We had added to the panel 2 years back. It's a plasmacytoidendritic cell marker, which is not seen positive in hematogons, but it is seen to be positive in the blasts. So these are helpful markers. Then asynchronous expression, asynchronicity vis-a-vis the hematogons. So in the hematogons, remember, we had seen the type 1 or the most immature hematogons are the ones which trouble us in differentiation. So there the CD38 is bright, CD10 is also bright. But here in blast, you will see a CD38 dim and a CD10 bright. So this will help us in differentiating the hematogons from these. So then you have other patterns like CD10 bright, CD20 bright, CD10 dim, 34 bright, CD45 dim, and CD20 bright. Okay, so I hope you remember the 3 ACEs of LAIP when you perform your analysis. Now next, coming to the gating strategy. So once all the processing is done and once we have understood what are the various patterns of the hematogons, of course, we have the basic singlet selection. We have excluded the doublets, then we have excluded the debris. This will be dealt with more in the subsequent lecture as well as in the workshop. Then, of course, the sequential gating is done by the initial gating with the CD19 positive cells. After that, we characterize our hematogons. So before you start reporting MRDs, you need to run as many normal controls as possible. What do we mean by normal controls? Regenerating marrows, which will have a lot of hematogons, so that we are able to identify the hematogon pattern and save these normal maturation patterns as templates. Anything be outside those templates, that will be characterized as abnormal. Okay. So this is an example of a marrow which was used to create these templates. So you can see we have neatly categorized them. So these are the templates that we have already made, and further analysis will be in these templates, and then we are able to identify the abnormal blast through this. Okay, so let us go to the plots, individual plots. So we have seen type 1 hematogons. Just a few plots I'll try to show so that to give you the concept of hematogons versus the different from normal. So type 1 hematogons, this is the 34 versus 10 plot. So type 1 hematogons will have 34 bright and 10 bright. Type 2 and 3 will have loss of CD34, and CD10 will be present in type 2 hematogons. So there's this concept of empty spots. That is, so once you have made your templates and characterize where these hematogons will be falling, so anything outside this normal patterns will be said to be abnormal. Doesn't mean that the abnormal blasts will not fall in these patterns. Of course, that's a further thing, but at least the basic you should know, anything beyond this will be abnormal. So here you can see this red population, just to make it very obvious. So this is CD34 bright, and CD10 is brighter even than our hematogon population. So this is an example of the abnormal population. Okay, so anything here will also be abnormal, and you can see here in this case we are talking of a loss of CD10. Okay, so of course you know the famous waterfall pattern. Okay, so type 3 will be CD20 bright, CD10 negative, type 2 will be heterogeneous, and type 1 will of course be CD10 positive and 20 negative. So anything beyond this of course is abnormal. Similarly, for the CD38 and 10 plot, okay? So very clearly, a dim expression of CD38, bright expression of CD10. So this could be an abnormal population. So I told you that you have to look at empty spots doesn't mean that there won't be normal cells over there. So this is an example of an empty spot, CD38 bright and CD10. So this is the space where normal plasma cells will be there, okay? So this is what could be called as the Wi-Fi pattern. And for the 73, so a lot of new markers you can use to characterize the abnormal population. So once you start, assigning some of the other patterns, the duck pattern, the rooster pattern, on all these, it becomes fun to analyze, right? So now coming to the MRD calculations, after that I'll show you cases. So how do you calculate? So X is the number of abnormal events. So in that abnormal population, if you say that X is the number of abnormal events and the denominator will be Y, that is the nucleated cells. So ideally you have to use a cyto13 dye paste to identify which are the nucleated cells, or you can take the non-debris cellular events in the side scatter versus forward scatter, and that will be your denominator. So MRD will be calculated as the number of abnormal events divided by the nucleated cells multiplied by 100. So that will be your percentage of the MRD. Okay, so this is just a quick two cases I'll show you. So a 12-year-old girl diagnosed as BALL standard risk, and post-induction chemotherapy there was recovery of counts, and bone marrow showed morphological remission. There were numerous hematogons, and the sample was received for flow cytometry MRD assessment. So this is the entire plot. You can see the doublet exclusion and the debris exclusion, then the CD19 gating. So what you can see here, maybe it's not visible to those at the back, but what you can see here is the cells are neatly falling into our templates, okay? So nothing in the empty spaces, right? So after analysis, we gave it as negative for measurable residual disease. So this is a very basic way of telling you, just to explain how you go about it. Now coming to the case 2, 8-year-old male diagnosed as BALL high risk, post-induction chemotherapy. There was pancytopenia, but bone marrow was given to be in morphological remission, and the sample received for flow cytometry. So what do we see here? Again, after doublet exclusion, debris exclusion, CD19 gating, and we had a lot of cells which were falling into the templates, but here in the 3410 plot, you could see a population which was, you can see this population red, I hope, all of you. So 34 bright and 10 bright. So we got hold of this population and looked at it in the other plots as well. And lo and behold, you could see that they showed abnormal patterns in most of the plots. So in the 3810, you can see it as dim 38. 10, of course, was bright, we had already seen. In the 2010, you can see this red population is 20 negative. In the 81, so 81 is characterized by, underexpression of the abnormal blast. So you can see this loss of expression, underexpression. 73, you can see overexpression. 58, again, overexpression. 123, 304, some amount of heterogeneous expression, and 44, loss of expression. So definitely abnormal cells are present, and now these are MRD positive, so now we have to calculate the MRD positivity. So the abnormal population came out to be 286 events, and the non-debris cellular events from this plot came out to be 14,68,892 events. Look at the numbers that we are talking about. Look at the sensitivity that we have to aim and achieve. So on calculation, the MRD came out to be 0.019%. So that was the basics about the MRD. And I made a mention about the next-gen flow. So what are the future perspectives? So more colors, more antibodies, more markers, more events is making it all the more sensitive. So this is an ASH annual abstract meeting poster from last year. You can see that they talk of 23 color, single tube 23 color panel for detecting measurable residual disease in pediatric BALL. So this is, of course, our Indian study, and they have talked about by Dr. Prashant Embre, and you can see the sensitivities that they are talking about using these more markers and colors, and so achieving sensitivity near to the molecular methods. So to conclude, flow cytometry for BALL MRD gives very good results. So knowledge of normal maturation patterns, use of LAIP and DFN methods helps in MRD assessment by flow cytometry. And being the strongest prognostic marker in risk group assignment, monitoring of MRD has become routine clinical practice and an excellent tool in frontline management of virtually all ALL cases. Now, so this is what I want to highlight. So this is the role of a pathologist or a hematopathologist. So we have to be constantly involved with the clinician, with the patient, and our role is therefore a continuous one in the management of BALL.
Hello.
So this is the take-home message. Yes, we can do it. And of course, with newer tools, I wanted to highlight, this is the only part of the presentation that involves AI. And I asked ChatGPT to write a poem for me on BALL MRD flow cytometry, and it gave these beautiful verses. So just look at the words, a pattern traced through every gate to read the fate we contemplate. Is it remission, clean and pure, or do a few rogue cells endure? At 0.01% they hide, yet flow will drag them from their tide. So quite interesting, just to highlight, of course, AI tools are also being used to increase your sensitivity and to make life easier for you in BALL MRD as well. So I would like to acknowledge my teachers. Thank you so much.
Thank you, Lalita. It was a wonderful presentation. I mean, the art of making a complex subject so simple with such fantastic, projections of the rooster effect and the Vidya Balan effect. Oh, it was wonderful, and those funny faces, which we saw. I mean, it was a wonderful lecture, and, you take me, this last part of your lecture, the poem takes me back to my Tata Memorial Hospital days, where there was no ChatGPT. It was just Borges and Roshni Chinoy, and they used to, actually, they were poets. They used to start their classes with the poetry, and that poetry was depicting the entire class. So those were not the ChatGPT days, they were real poets, okay? And they knew the subject. So thanks a lot, Lalita, it was a wonderful presentation.
Thank you, ma'am, for such a creative presentation. May I now request Dr. Sarika Singh to introduce our second speaker, Dr. Astha Gupta.
Welcome, Dr. Astha. It's my pleasure to introduce you here. And she's a well-known hematopathologist who is very, very passionate about flow cytometry. And we keep hearing her in various conferences and the effort she put up in her presentations. So currently she's the additional director and head of the hematopathology and molecular oncology at Fortis Hospital and the Institute in Gurgaon. She is an alumnus of AMU, and then she pursued her passion in Gangaram Hospital and Hinduja, followed by her DM in hematopathology in prestigious Institute of All India Institute of Medical Sciences, and here she is with her presentation. I will not come in between her and the lunch and the process, so please proceed with your topic.
Thank you so much, Sarika ma'am, for a kind introduction. And thank you, Tathagat sir and the team, for giving me this opportunity to talk on my favorite subject, MRDs. Okay, so I hope you are not asleep or hypoglycemic. It is a short and sweet presentation. I'll try to be very, cover the basics. Most of the things have been covered by Sarika ma'am, Jyotsna ma'am, so she has made my job very, very easy. This is the place where we work at Fortis Hospital, Gurgaon, in association with Ageless Diagnostics. So what are our learning objectives in the last 1 hour of this workshop? What we have to what is MRD, what is measurable residual disease, why do we do MRD, and how to do MRDs. And my job today is to tell you how to do MRDs, or at least brush you through the basic concepts of MRD. Okay. So what is MRD? How do we, at least just one, what is the full form of MRD? Why it is measurable and not minimal as of now? So it is measurable because, any answer, just one line? Because it's measurable. But why the name has been removed from minimal to measurable? So minimal term means that the very minimal number with which we can detect, and there will be no number or no residual leukemic cells beyond that or lesser than that. But that is not the case. Every test, every methodology has its own limitations, has its own sensitivity and specificity. And as you, like ma'am said, next generation flow, next generation sequencing, as we are coming up with the more and more advanced technologies, the sensitivity is increasing, and therefore that minimal number is again going lower and lower. That is why we call it as a measurable residual disease. Why has been explained very beautifully. The only thing I will like to highlight is risk stratification with the help of MRDs. Second thing is to understand whether your patient is properly responding to the treatment or not. And third thing is for an early relapse detection. So the time points have been designed according to them. Post-induction is the first phase of induction therapy given to BALL patient. After that, we will get first MRD, which is time point 1. Then there is a next phase of chemotherapy, which is consolidation. After that, we are going to get time point 2, which is post-consolidation. In private setup, we also get the MRDs at the time of maintenance and after completion of the therapy. And then any time, if the patient is having cytopenia, if the patient is having any CNS symptoms, or if the patient is even having continuous fever which cannot be explained, then we get MRDs in these patients so that we can do an early relapse detection, and hence the clinician can take, they can decide the treatment accordingly. And this is simply a multicentric 5-year study showing that as your MRD is positive at each time point, then the overall survival and the event-free survival, it goes down at each positivity. That is why the high risk or the highest risk of BALL is when it is time point 1 MRD positivity as well as time point 2 MRD positivity. That is when we say that the patient has the highest risk of BALL, and such patients will be taken up for transplant. So that is how important we hematopathologists are, because we are guiding the treatment for the patients. And flow cytometry is currently the method of choice because of all these reasons. So let me just focus on how. First is the panel designing. Okay. Now panel designing, and Dr. Deepti has really very beautifully explained what are all the factors involved in a panel designing. Without going in details, simply a collection of all antibodies which we need to put it in our panel is called a panel, along with the fluorochromes. Then the second part, the standardization and validation of each panel is very important. That in itself is a lecture of 2 to 3 hours. Third is the gating strategy. Now, gating strategy can vary or can be different for different people. You just need to understand the basic concepts, and then you can have your own gating strategies. And final part, how do we report it? So what is the aim of our MRD panel designing? We want to accurately identify the leukemic cells in the presence of normal hematopoietic cells, like those one blue minion in the background of yellow minions. So why the normal hematopoietic cells are coming back at the time point 2, if I say that I want to identify my leukemic cells, but there are also normal hematopoietic cells. So what is the reason that there are normal cells or normal hematogones coming up in such cases? Because of the chemotherapy that has been given, so the normal marrows also starts regenerating. The second aim is to detect even the smallest residual population, which is measurable, which is precise, and you can produce a reproducible result. What is difference between accuracy and precision? I'm sure everyone is a MD pathology or SR. You must be knowing accuracy is to hit that dartboard in the center. That is the accuracy. Precision is that every time you repeat the same test with the same sensitivity and the same panel, you will get the same result. So our aim is to identify those residual population which if we do it again tomorrow without giving the treatment, we will get a positive result. Okay. Then we have to quantify the residual disease. The clinically actionable cutoff for BMRD is 0.01%, more than equal to it. It is different for AML MRD, so it is different, but for BMRD it is 0.01%. And then we also have to identify the targeted therapy. BALL as of now is not only the BFM protocol or the ECOG protocol, they even give the targeted therapy. For example, inotuzumab for CD, inotuzumab targets CD22, rituximab targets CD20. Now CAR T therapy, it targets CD19. So we have to give the markers for the targeted therapy. Okay. So okay, this didn't work. Like my GIF, I wanted it to work. So first thing is basically all are following the same panel. Normal hematopoiesis is a very strictly regulated concept, and all the normal cells will follow a strict regulation and a strict pattern, and any deviation from the normal will be an abnormality. So with what is the odd one out in this list? All whole smileys. Second row, third one. Okay, so basically what has happened is eyes, the tongue, the eyebrows. What are these? And if I try to understand in terms of flow, then what are these eyes, tongue, and eyebrows and all? They will be basically the markers and the deviation which we are looking for in our normal background concept. So the selection of markers will require all the following. First, for BMRD, you are trying to identify the B cells. Now everyone has said that CD19 is the most sensitive and specific marker. CD22 can be used, cyto CD79A. But why CD20 cannot be used in BALL as a identifying marker for B cells? Because it is lost in BALL, or it is not present even in the earliest hematogones. Okay. So one marker that we can use is CD19. Then we have to have the markers which will help us to identify the hematogones. 10, 34, 20, 38. Then we have to have the markers for the deviation and aberrancies, and one universal marker which will be present in all your flow cytometry panels for that matter, which is CD45. Now this is a list of almost 15, 16, 17 markers, but how complex your panel will be, that will depend upon what flow cytometer is available with you. Okay. And you have to balance complexity with the sensitivity, again reproducibility and affordability. Tata can afford to have a 23 and a 25 color marker panel, but we may not, because then our patients will be charged accordingly and there is a whole hoopla around it. But we have to make a very, very concise but at the same time an effective panel. Currently for BMRDs, an 8 to 12 color panel is optimal. It is considered to be an optimal panel for discrimination of hematogones from the blast. Just a word about the concept of backbone markers. Why do we need backbone markers? Because if you are making a 8 color panel, which will be 2 tube panel, or if earlier days when seniors used to report on 6 color flow and all, they used to have 4 tube panels. So you need to have the markers in each tube in a way that you are able to identify your B cells in each tube, you are able to identify your hematogones on each tube. So that is why it is important. That is why ma'am said that we are happy with our 12 color panel, because then you need not to have 2 tubes, 3 tubes and then repetition of your backbone markers, and it will be more cost effective. Now these are the panels at our fMRI. Initially we used to do 8 color flow cytometry, where the 20, 45, 81 can be replaced with 58, 34, 19, 10, 38 were the backbone markers, and currently we are using a 10 color flow cytometry BMRD single tube panel. Stages of hematogones have been theoretically told to you, and all those beautiful patterns have been told. I'll just share it with the live case analysis. This is CD34 on x-axis and CD10 on y-axis. This is a stage 1 hematogone which will be positive for both 34 and 10. This will be a stage 2 hematogone which will be negative for 34 but positive for 10. And this will be most likely a stage 3 which is the mature B cells, negative for both. Now on 38 and 20 if we see, both stage 1 and stage 2 hematogones will be positive for 38. But as the stage 2 it starts gaining CD20. So this x-axis is a CD20 which is started to gain CD20, and this green population is our mature B cells which is losing CD38 but strongly positive for CD20. And CD45 also we know that as the hematogones mature they start gaining CD45. This is another article from where I have taken these patterns for hematogones. But let's start going to our cases. Gating strategy, the x and the y axis, that whole thing is called as a plot, and the gate which you make around your population of interest is called as a gate. So case 1 is a 30-year-old male, known case of BALL. It was a post-consolidation marrow in morphological remission, and bone marrow sample was sent for MRD. First pull bone marrow sample is very important. It is the duty of us pathologists to tell or guide us, our clinicians, that you have to send the first pull, because we are trying to identify that one cell in 10,000 cells. So the first pull of the bone marrow has to be a MRD sample. Now, this first plot is basically a time versus forward scatter plot. Now, why am I showing this time plot? Because no one else showed this time plot in leukemia lymphomas. It is not required. But why in MRD analysis you need this time plot? So time plot will tell that as you were acquiring your sample, as the time was passing by, all your events, all your cells were acquired properly. If there is some interruption in between, that will be recorded, and you will have to understand that you don't gate non-specifically your bubbles, and your analysis is not affected by those interruptions. Why it is important in MRD? Because we are trying to acquire at least 2 to 3 million cells. That will take a longer time for acquisition, okay? And since it will take a longer time, there will always be a chance that some bubble will enter, or some or the other air will enter, some debris will enter. That should not happen, and this is the first indicator that your acquisition has happened very smoothly and beautifully. Then you make your singlet gate on forward scatter area versus height, and then you make the viable gates on forward scatter and side scatter. These cells are debris. Anything, like Tathagat sir told, that anything which is smaller than lymphocyte will be your debris, will be your platelets or unlysed erythroid cells, will be anything non-viable, and so they have to be excluded from our MRD, because we are trying to look at only the viable cells. As we proceed further, so this is my CD45 versus side scatter gate, where always make it a habit to look at this gate even in your MRDs, so that you will have a fair idea of what you are looking. Like you can see that your lymphocytes are there, your monocytes are there, grans are there, and this could be a debris or this could be a BALL clump which is present in the sample. Then the next, this is about the gating strategy. This is my gating strategy, but you can always design your own. I will just convey you the concept which I use. So, and CD38 and versus side scatter, my idea is to remove these granulocytes, because the granulocytes have a tendency to non-specifically bind to the antibodies, and it can interfere with your analysis. So we will remove these granulocytes, and we will take only the viable mononuclear cells, MNCs. Because my blast, my hematogones, my progenitor cells, all will be mononuclear cells. So why do I need to see? I am not looking at AML MRD. I am looking at BALL MRD, right? So I don't need these high scatter cells. So I will remove these high scatter cells. Then the next, when we follow a Boolean gating, a subsequential gating strategy. So once I have made my viable cells, from viable cells we will isolate our viable mononuclear cells, and then in viable mononuclear cells, we will identify our CD19 positive cells, because we are looking at BMRD. Again, any MRD panel cannot be directed to multiple diseases. That is why BMRD panel will be directed to BMRD. It cannot tell you about the AML MRD, okay? Because many times clinicians ask me that we have sent a BMRD, can you tell us whether there are any abnormal myeloblasts there or not? So it is not possible because they are targeted panels. So CD19 positive cells, and the first thing which we remove, like ma'am said, like in an empty area, which is 38 bright and 10 negative, are the plasma cells. So in my strategy, we first remove the plasma cells. We know that the brightest 38 positive cells, which are 10 negative, will be plasma cells. So we identify them and then we remove them. And then the remaining cells, we will again look at CD19 versus site scatter, trying to remove more of the debris. And these are the clean B cells. So now we will go ahead with our clean B cells. First is the 10 versus 34 graph. And we can see there are some 10 positive cells, some 10 positive, 34 negative cells. We have started getting them. I am assuming that they are hematogones, but we need to prove that they are hematogones. So the blue population, which is stage 1 hematogone, positive for 10, negative for 20, positive for 34, it shows that beak pattern or the cuckoo pattern, these are strongly positive for 81 and 34. And side by side, I am going to look for the absence of any aberrancies. Like hematogones will not express CD123, hematogones will not express CD73 or 304. So we know that there is no any abnormal expression of these markers. And this is the stage 2 hematogone showing that waterfall pattern, acquiring 20, negative for 34, negative for aberrancies, and positive for 81. 10 positivity is slightly more than in stage 1 hematogone as compared to stage 2 hematogone. 81 positivity is slightly more in stage 2 hematogones as compared to stage 1 hematogones. But all these patterns, I am saying this confidently because I have seen like thousands of normal marrows as MRD negative cases also, where I am seeing this consistent pattern. So whenever you are going to design an MRD panel, you have to look at those normal or the regenerating marrows where there are hematogones, and you will see that they will beautifully try to follow the same pattern. So that is the beauty in BMRD, that hematogones try to stick to their own path, and any deviation we know that it will be abnormal. 38 positivity is a very, very important identifier of hematogones, stage 1 and stage 2 hematogones. And this is another where I like to see at 38 with 34, just to see the capping by the plasma cells. These are the stage 2 and these are the stage 1 hematogones. So these are hematogones and no abnormal cells seen. Always the CD45 is like the peripheral smear of a flow cytometry, because it will tell you about all the cells, it will follow a certain pattern, and you can always backgate your hematogones and see that this is stage 1 hematogone trying to gain CD45, stage 2 hematogone, and these pink dots are stage 3 hematogones with the brightest 45, of course less than T cells, and with the low site scatter. Now in MRD, it is very important to have your quality checks. How do you know whether the peripheral blood has not been sent? Or how do you know whether it is not a hemodiluted marrow? Or how do you know that it is truly representative of the proper marrow or the first full sample that has been sent to you? So there are certain quality checks. First is there will always be normal myeloid progenitor cells which are regenerating in the marrow. That will be there. Literature defines it as if it is more than 1%, then it is normal. But anything more than 0.5% will tell you that an adequate bone marrow sample was collected for this case. Okay. Then plasma cells. Circulating plasma cells are very rare. So if you are having a 0.7% plasma cells, I am very happy, because in my normal marrow, any healthy person, plasma cells normally will be 1 to 2% only. So 0.7% is a good number to tell me that this is a representative marrow. And definitely how many total number of cells we could acquire, what was our viability, and all those things. Case 2, and then we'll go to the reporting. How do we do the reporting? Known case of BALL, morphological remission, MRD sample sent to us. Then again the housekeeping plots. Time is very important, that as and when you will start working on MRDs, you will realize that a smooth acquisition of your sample is very important. What do you mean by events? We have been talking about this term event. What is an event? one cell. Each cell is an event. So all these each dot is each cell, and these are called as events. Then we do the singlet, and then we remove the debris. This is the debris, and we see the viable gates. CD45, it's my habit to see the CD45, and here I am worried that what is this CD45 dim region, which is forming a sort of a cluster. Then again, exclusion of the granulocytes, viable mononuclear cells, gating on CD19 versus site scatter, and then removing my plasma cells. But this cluster I am not happy, because as you saw in the previous case, it was sort of two clusters sticking together with the dimmer 10. Okay, so we start suspecting something is wrong from here itself, and then we do the clean B cell analysis, and then we see our first gate as CD34, 10 and 34 and 10 and 20. So whatever it is strongly positive for 10 and 34, but there is, I will expect that some cells, more cells should have been here if it is following the normal pattern, and CD20 is completely negative. So are these cells abnormal? Most likely yes, but we have to see the further plots, and when we get them as query abnormal for this workshop purpose, there is absence of CD20. They are bright positive for 10. They are also expressing the LAIPs, CD73 and 304. They are also expressing CD123. They are dimmer. They are not forming that cuckoo or beak pattern. They are one single cluster which is dimmer for CD81 and strongly positive for 34. And CD22, I was telling you about the targeted therapy. So we have CD22 in our panels because our clinicians, and when the patient is not responding properly to your induction therapy, they will add inotuzumab. It's a common practice at our hospital that they will add a targeted therapy. So they will want to know whether my residual cells, whatever residual blasts are there, whether they are expressing CD22 or not. So this is a common practice which we have in our MRD panel depending on the requirement. And this list is ever growing. Maybe we'll have to modify the panel when more and more targeted therapies will come, and we need to give it to our clinicians that yes, my residual cells are positive for so and so marker. So this is just for comparison that my normal hematogones will be strongly positive for 81, and as compared to this, my CD10, these abnormal cells are brighter. So when you start seeing more and more normals and comparing with the abnormals, you will get a hang of it where your normals will lie and where your abnormal is lying. Like CD38, this is the place where my hematogones are usually coming, and this is slightly dimmer for CD38 and brightly positive for 10 and showing dim CD45 expression. So this is MRD positive. And when we look at the statistics with the calculation, normal divided by non-debris cellular events, or you call them viable events, multiply by 100 is the simple calculation for positivity to derive whether it is more than 0.01% or not. Just 2 or 3 slides for reporting. The reason I had put my reporting this thing is you need to know what all you have to incorporate in your reports. Okay, first is the patient details. We have to know the clinical history. We try to get the diagnostic phenotype, but because BMRDs, the normal hematogones, they follow a very stringent the maturation pattern. So even if the diagnostic phenotype is not available, you can still very well do a BLL MRD even without that. But if we have it, we put it in our reports for every time, because each time point the cases will be coming. So for all time references. Now this is the time point. In our reports, we always give what is the time point of MRD. This was an end of consolidation time point 2 assessment. And what is a specimen EDTA bone marrow? You should always tell about the methodology. What flow cytometer you are using, what software you are using, then what is the protocol, bulk lysis stain wash protocol that you are using, what are the markers you are using. So that is important for a good comprehensive report, which I feel. Now what are the total events acquired? When it is 3 million, 3.5 million, the clinicians are also at peace that we have acquired enough number of cells to reach the sensitivity of 0.01%. So if I consider 100 events as a cluster for my positive MRD, how many number of cells should I acquire to reach a sensitivity of 0.01%? That we can discuss in the workshop also. But how many? If we can do the calculation quickly. 10 lakh, yes. So 1 million cells. So 10 lakh is 1 million. 1 million cells should always be minimally acquired to reach the sensitivity of 0.01%, but definitely if you reduce your confidence like at confidence level to 50, if I am confident that at 50 events also I can say that my MRD is positive, then we can reduce the number to 5 lakh. But we as a practice always try to acquire more number of events, somewhere between 2 to 3 million, so that our viable viable cells are always more than 2 million. Then we give the abnormal events which are nil, impression is negative, and you always say what are you finding in your MRD, like hematogones 2%, plasma cells, mature B cells, and this CD34 positive non-B cells are basically your regenerating myeloid progenitor cell. 1.1% gives me also the confidence that this was a representative marrow. And lastly, LOD and LOQ, that are the part of validation studies, but LOD means lower, LLOD basically, lower limit of detection. What minimum number of events you will be confident to call as positivity, and lower limit of quantification, what lower limit number of cells or events you will be confident to give it each time. And the sensitivity of any MRD will depend upon how many viable events you have been acquiring. So as in when you will start practicing MRD, you will understand that these are important concepts, and in our reports we give the LOD and LOQ which has been attained for that particular patient, depending on the viable cells which we have acquired for that patient. Okay. This is the same concept, but this is a positive MRD, and always in a positive MRD, please try to explain or write it in your reports that what is the phenotype of your positive or the residual cells. So these are the reporting points which are important and must be included in your MRD reports. The challenges are basically the validation. Most importantly, standardized protocols have to be established. Many labs should be trying to follow, the similar panels so that we fall on the same page. And technological advances are there like NGS, next generation flow. The MRD should be comprehensive and should be clinically applicable. Finally, it is the clinician who has to take the decision on your MRD. So you should be very clear what you are giving and what you are reporting. And a collaborative work is the future to go. Thank you for your attention. And this is our team at Fortis.
It was a wonderful talk, Dr. Astha, as usual, and we really enjoyed, and I really appreciate your efforts which you have put in, and all the delegates and the students who are present here will echo with my sentiments and my expression that it was a wonderful talk indeed. Thank you. We'll have a discussion during the lunch time. Thank you.
Thank you, ma'am, for this wonderful talk. May I now request Brigadier Dr. Tathagata Chatterjee to felicitate our speaker, Dr. P. Lalita Jyotsna.
After 10 minutes, we'll have lunch, right? Because the breakfast was quite heavy. And so 10 minutes. I know you're empty, but it was heavy. It was heavy. इतना भी खाना नहीं चाहिए. पढ़ने आयो, पढ़ाई करने आयो, ठीक है? You can't. Army marches on bellies, but इतना भी मत खाओ, ठीक है? पढ़ने आयो, और हम पढ़ाएंगे, नहीं छोड़ेंगे तुम्हें, ठीक है? So 10 minutes, we'll have a presentation of MRT, various MRTs by BD. But before that, okay, I have to felicitate you. Sorry.
May I now request our chairperson, Dr. Sarika Singh, to please felicitate our speaker, Dr. Astha Gupta.
I now request Dr. Charu Agarwal to felicitate our chairperson, Dr. Sarika Singh.
we request the audience's attention for another 10 minutes because now we have a small Friends of the Meat session where clinical application specialist Dr. Shashank Mishra is speaking on the use of BD in clinical cytometry. It's a very important take-home message for everyone, so I request everyone to please pay attention to Dr. Mishra's valuable words.
आप लैपटॉप यूज करोगे? हाँ। आपका बैंक हाँ दे नहीं सकते ना। HDMI डी पोर्ट है ये। आया? आपको प्रॉब्लम है तो फिर मैं इसको सामने ले लूँ। ट्रांसफर नहीं कर सकते ना प्रॉब्लम तभी तो मैं आया कि नहीं आया डिस्प्ले नहीं आया डुप्लीकेट स्कैन किया आप अपने उससे आगे इस वाले में आ रहा है ये नहीं ये तो उन्हीं का चल रहा है हमारे इस पे आना चाहिए डुप्लीकेट कर तो आया डुप्लीकेट कर दिया नहीं आया मैं दूसरी वाली केबल लगा के देखूँ कनेक्टर नहीं है कोई और होती क्यों नहीं है हर जगह इसी से लगाते हैं ये ले ही नहीं रहा अडेप्टर है नहीं दिखाना
एक्सटेम्पोर बोल आया टेक अ माइक एंड स्पीक एक्सटेम्पोर ज़्यादा दिखाने का नहीं चलो चांसेस है क्या So Dr. Shashank is our go-to person. Please give him a big hand because he is the person along with his team, Dr. Vinay. So they are the people behind the scene. They are the doctors who has actually taught us flow cytometry. ठीक है? And in the workshop you'll see that they'll be standing in all strategic places. अगर हमने मिस्टेक किया ना हम इनके पास जाएंगे. ठीक है? But right now it is beyond him. है ना? Technical glitch. चांसेस है क्या
So hi, I'm Dr. Shashank, working as an application scientist with BD. So we are a people of 4 who are assisting you during the workshop, as said by sir, headed by Dr. Vinay. So we are here to just to give you a glimpse of what BD has been doing for past years in the field of flow cytometry, whether it could be research or it could be clinical. So starting with, BD has a past history of around now more than 50 years in flow cytometry. We started with the very basic 4-color instrument. Now we have gone up to 50 colors, even 78 colors in terms of sorting. So that is the kind of a small history. Then we have developed different kinds of reagents, software supports to give you a better picture in terms of flow cytometry, not only in terms of clinical as well as in research. So we have come from a 4-laser system which was Equiri, or you can say Fax Via, which people were using, or Caliber, which people were using in the initial days. Then we came up with FaxCanto in a clinical setting for the 8-color, and now we have moved to a 12-color instrument, which is your BD Fax Lyric. So now something which we wanted to highlight more, because BD has been innovative from years. So we have come up with an imaging flow sorter also, which is in the name of Discover Acid. So now you can see the image also along with your flow data. So that is also possible. We are also coming up with an analyzer in the same line. Okay. Now on the clinical front, we have come up with a sample automation system for you. So that could be totally avoiding the manual errors which are there. Sometimes we have some manual errors that the sample was not properly prepared, or it could not have been properly executed or acquired. So all those errors have been taken up by this sample preparation system which we have now. So that could be linked with your lyric system. So you have to just put in your sample there. That is the vial which you have in the coagulative system. You have to just put that vial and you will get your result on the screen on lyric by just giving some commands. Okay. So now the other advantage which we have got is, we have also got now, by acquisition of Cytogenos, we have taken over the Infinicide software, which depends on the Euroflow database. So if you are working with an 8-color panel, although most of us have moved to 10-color or 12-color, but still if you are working with a diagnostic panel of 8-color, you can do automated analysis with Infinicide software, which is available there. So that gives you also a proper analysis for your data, as well as give you a reporting format. As Dr. Asta said, that reporting is very important. So it gives you an impression that you can do those MMRDs, or you can do those LST, CLPD reporting in a very correct manner with those automated software. As well as it also gives you the privilege of using the manual. You can create your own database. It gives you the opportunity of creating that also in that concept. So that was a little bit of what we were going to show you in the presentation. Now coming to the clinical applications which are applicable with flow. So you have seen some of them which have been discussed in the morning. We started with acute leukemia, that is hematologic malignancies. So we started with acute leukemia, so you can do acute leukemia detection. Now how it is different from microscopic? In microscopy you come to know what kind of, what is the blast, is there a blast or not, but with flow, So that was all what we were trying to show you. So we started with the history. So these are all the flow cytometer which we started with. We started with the very basic fax via, then we moved to fax canto, and now you have the latest fax lyric with you. This was the flow sorter, which is an imaging flow sorter, which I was talking to you about. So we have used a patented BD cell view technology, which doesn't use a camera to take the image. It's basically different wavefronts which we combine. That is you can say a cellular based technique which we have used to create the image in this. Then we have spectral. This is completely spectral instruments, because now flow is moving towards spectral. So maybe in near future clinical also you might see what we are seeing, 23 color, 25 colors are coming up. But we have to see the costing also, as ma'am said. That is also an important factor in India. This is the duet sampler system which I was talking to you about. So it's a complete automated system for you. It gives you the flexibility. You can use any manufacturer, any size vial to get to use to it. You can program your own recipe. You can program you can put your own programs into it. Then it gives you the flexibility. It gives you the importance of standardization also. That is, it gives you error reduction. So there is no manual intervention, and then there is ease of use. You have to just put, as I told you, you can just put the rack with your sample and you can get the result at the end of the day. So no manual intervention, no problems whether the sample was properly processed or acquired or not. Okay. This is the Infinicide software about which we were talking about. So it's a complete automated solution for you, as well as it gives you the privilege of doing manual analysis also. So it is based on the Euroflow database. Okay. So it could give you the privilege of doing both manual analysis as well as automated. So automated you can use the Euroflow database, but the restriction is that the panel of Euroflow is still up to 8 colors only. So you can use only a 8-color panel. But if you want to create your own database, you can do it with manual analysis, and then you can create your own database and submit it there. Okay. Now this is what the major thing was. What are the applications which is possible with flow in clinical scenario? So we have been discussing them from the morning. So some of them, the major highlight was these today. Some will be discussed with you tomorrow. Okay. So leukemia, lymphoma, we are very much clear now what is a leukemia and what is a lymphoma. We can clearly differentiate. Leukemia is immature, lymphoma is Mature? Immature opposite is mature? I think they are crazy for lunch now. I am taking their time. So now, the other thing which we can do is we can also process with solid tumors. There was discussion in the morning that we can process with DNA solid tumors. So we can do, as Dr. Tathagat said, that we are now moving, everybody is moving toward DNA ploidy studies where we are trying to see what is the ploidy levels. We could do express fractionation also there possible. Then one of the more approaches which you will understand tomorrow is PID. So PID is one of the aspects which is now coming more and more in clinical kind of a scenario, and in flow we can do them. Initially we were only able to do with TBNK, but now we have specialized kits available for them in the terms of PI-DOT, which is on a Euroflow basis. So that gives you more advantage. So BD has those kits available for you, and we gives you the privilege of doing the initial analysis for you also in that, so that you get used to it. Then we would be moving towards the hemolytic anemia conditions also tomorrow. Major highlight would be the PNH, that is the disease which is associated with your loss of glycosylated proteins. So there we would be looking into the levels that how they are. So we will be looking into the WBC levels, which is now the major thing to be looked into. But if you still have doubt, you can go into the old way also of looking into the RBCs. Then we can also look into the different platelet disorders, where we can look into the levels of CD41 and 61, all those more kinds of molecules, whether there is activation or loss of those. Then the important is transfusion and your transplant. So this will be at tomorrow an important topic where we will be discussing the stem cell. So this is another important aspect. So flow cytometry is a kind of a major technique which is nowadays used for it. So where we take into the guidelines of ESIC guidelines, follow the ESIC guidelines for it. And then we can also use it in the terms of cross match, where we do kidney and liver transplants. So it gives you the idea whether your HLA system is getting activated or not. Initial indications. So this is all we have, and just as promotional event, VD has all the things available for you, starting from the instrument to the reagents to the software. So BD gives you all. Another promotional event I have been told to be, because I am a science person, but I have to go commercial also. So we have a stall there outside. So if you want, if you have any queries with BD products, you can go in there and have a look at it. Thank you.
May I now request Dr. Shilpi to please honor Dr. Shashank Mishra for his wonderful talk.
Kindly wait for a minute before proceeding to lunch.
I request the audiences to keep their patience for another minute, please.
So we'll be proceeding with the lunch now.
Felicitation.
We'll be proceeding for lunch now. Lunch will be served in the examination hall, ground floor. Following lunch, we'll begin with the workshops and on live gating and hands-on session 1, 2 and 3.
We will have 3 sessions going on simultaneously in Dhanvantri, Charak and Sushruta Hall with each session of 1 hour. The delegates are welcome to take turns going to these halls. Following the workshop, there are some quiz sessions for the delegates in a couple of the workshops. We wish you all the best for the same. We also invite you to conclude day 1 with high tea at the end of the workshop, and everyone is requested to return to their designated halls after lunch. Enjoy your meal. Thank you.
Please refer to your schedule that is given in the brochure. Please refer to your schedules.
For the workshops, we meet sharp at 2:45 in your designated halls.
हाँ हमें मैसेज मिल गया है प्रोजेक्शन पे से लिखा होगा यहाँ पे अदरवाइज है दूसरे इस पे है और अच्छा इसलिए वो आपको टेंशन नहीं कोई ना मैं अपना लड़की यार प्लीज से अपना था उन लोगों ने जाने की कोशिश कोई लड़का होगा नहीं भी हो तो लेकर आ जाएगा तो यार अपना लंच कब करेंगे सेट कर लेते हैं बस सेट करके लंच करने चलते हैं पहले यहाँ एक वहाँ बोला
पीढ़ी वालों के पास मैम 3 नंबर का हाँ वही पूछ चार्ज नहीं है कल ओके
हेलो नहीं वही अपने पीजी रूम में पड़ा हुआ प्रेजेंट कर रहा वैसे हम कर लेते हैं हाँ वो एक और है वो थोड़ा सामान हाँ वो ले जाएंगे मैडम लगाइए आप अपना ना डोंगल भी एक बार देख लो बस
चार्जिंग पे लगाना पड़ेगा इसको
नहीं यहाँ पे पता है एक फायदा है अगर ये
आगे का पासवर्ड बीडी का क्या है आगे का पासवर्ड तो पता है
जो है वो ले आओ एक बार
आप डिसाइड करो क्या करना है ये
हमारे लिए वही होगा
पहली सी डेट आप लोग जाओ, हम लोग भी सेट करेंगे। चार्जिंग पे लगाए हैं क्या? हमारे चार्जिंग पे चार्ज नहीं है।
इधर से ना कोई बात नहीं, इतना कोई बड़ा इशू नहीं वही सबसे आसान है, अभी भी कह रहा हूँ। अच्छा नहीं नहीं मैं भाई मैं वो नहीं चल रहा है इस पे ना, सो सॉरी। एक बार तुम लोग फाइनल यहीं पे रख रहे हो, मैम यहीं पे लगना पड़ेगा, सो सॉरी। वो उनका प्रोजेक्ट चार्जर तो लग जाएगा, चार्जिंग पे लगाना है।
नहीं उनको पता है खाना लेकर। आप पहले इसको लेके आइए। अरे एक्सटेंशन का एक और काम हो गया। एक्सटेंशन का हो गया। कोई नहीं, मैं बात से ये ठीक है, सब जो यहाँ बैठ के 10 जगह यहाँ पे और वहाँ पे देख तो सब मैम कुछ आ रहा है हाथ में नहीं ये लो आगे सर, ये वाला आ गया, कोई बात नहीं, अब रहना अब इधर उधर बाकी दो लैपटॉप किधर है? बस अब एक सर प्रोजेक्टर का वो दे ठीक है, स्क्रीन के लिए एंड इसको 10 के लिए शेयर अच्छा कंफर्टेबल सेम पर्सन में एक माइक्रोफोन कनेक्टेड आ गया हो नहीं बैठे हो और यहां पे आएगा ऐसा घुमा करेक्ट समझ गया। अच्छा हाँ समझ गया, मतलब वो जो पर्सनल रहेगा उसमें एरिया फॉलो करेगा, बोलेगा वही पर्सन। हाँ भाई मुझे भी ज़्यादा दिक्कत नहीं है, ये परफेक्ट है। मुझे एक्सटेंशन के बिना नहीं होगा, वो तो ये करेंगे ना, नहीं वो तो भी करेंगे, पूरा डोंगल पड़ा हुआ है। सॉरी डोंगल को पावर कैसे देनी है? ये लो अब आपका
तो बाद में भी आती तो भी मिलती बेटा मिलती नहीं मैंने उसको बहुत पहले बोला था, वो लेट हो गया ऑलरेडी। कौन सी क्लास में? सर्जरी की पोस्टिंग मैं वहाँ पे टीचर ऐसा उसको बोल देती हूँ, ठीक है, तब तक के लिए, जब तक मैम के साइन नहीं है, लेकिन मैम बिजी है। अरे कौन से हैं? जब मैंने उनसे पहले बोला था कि पेपर ले आओ, लेकिन नहीं कॉल कर देती हूँ, यूनिट 4 में पीजी है ना वहाँ पे, इतने में उनको इन्फॉर्म कर देती हूँ, ठीक है।
ये प्रोजेक्टर ऊपर है, दूर नहीं हो जाएगा सर? अच्छा नहीं
हाँ, और इस वाले को आप लगा दो इसमें, बस
हो गया, ऑन हो गया। इस पे भी आ गया इनपुट।
ठीक है, इजी है, हो गया। बट इस पे बहुत स्लो आ रहा है अभी।
अभी नहीं आएगा, अभी इसमें स्लो क्या, वहाँ से करूँगा ना।
अच्छा ठीक है करो।
ये आ गया चार। ये हो गया उस पे, ठीक है। लैपटॉप कहाँ लगेगा? तो ये केबल हो जाएगा।
कौन सा पॉइंट है ये?
पासवर्ड इज
स्लाइड पर ठीक है।
यहाँ पर प्रोजेक्ट होगा लैपटॉप में जो है।
हाँ ठीक
है, फिर लैपटॉप को कनेक्ट करना होगा, लैपटॉप भी कनेक्ट होगा।
यहाँ पे हम यहाँ कनेक्ट कर सकते हैं कि वहाँ पे कनेक्ट।
अभी लाइट जला दो।
हाँ, अभी लाइट ऑन कर देना, ये भी मैं बता दूँगी।
आप ही बैठोगे? मैम आप सो मैम यहाँ पे आप शायद
साथ ही चलते हैं, यहाँ पे लैपटॉप नहीं रख पाएगा, साथ ही चलते हैं। चलो मैम, फिर आप चलो, मैं लैपटॉप लगवा आशना नीचे मुँह कर लेके मानसी को ले मानसी किधर है? मानसी चरक में है, उसको ले आते हैं, देखने जा रहे हैं भाई। चलो देखते हैं, मानसी का कनेक्ट हो गया, फिर उसको भी ले आते हैं। तो सुनो लैपटॉप, हाँ ये मानसी का लैपटॉप दे दो, ठीक है। अशोक बता रही है मानसी के पास चल रहे हैं,
लगाने दो मैडम मुझे बस।
अशोक कब आई थिंक अभी यहाँ से ना
चीजें सारी होंगी, आप जो मांगोगे।
लोग नहीं आने वाले, लोग आने वाले हैं।
लैपटॉप है तो मुझे चालू करके।
अब क्योंकि लैपटॉप कंटिन्यू 3 घंटा यूज होगा, तो उनको बस चार्ज के पासवर्ड लगा के पासवर्ड डाल के और पीडीएफ एक्सेस में खोल के 117 पीडीएफ एक्सेस अकेडमी पासवर्ड, हां एवरीथिंग इज एज सेम पासवर्ड 136
हमारी कंपनी वालों ने आधा अधूरा काम किया है हमारा।
किसने पासवर्ड
दोनों का डिफरेंट है, एक 36
सबके पासवर्ड।
मैं करता हूँ अभी, रुक जाओ, मैं थोड़ा सा भी बिजी हूँ, मैं थोड़ी देर में फ्री होके मैं करता हूँ, ठीक है। हाँ लैपटॉप दीजिए इधर, मैं करता हूँ जी, लैपटॉप दीजिए। बिल्कुल चार्ज है क्या है मैडम, इसकी क्या स्थिति है?
जो ऊपर मीडिया एडमिनिस्ट्रेटर लिखा है वो छोटू में है, स्मॉल में सब मीडिया लैपटॉप हो गया, क्या हो गया कनेक्ट? मैंने एक्स्ट्रा में उसको दे
कर रहे हैं 5 मिनट दे दो, सारा कुछ हो जाएगा। बस ये है कि मुझे थोड़ा सा
कुछ नहीं दिख रहा वैसे, कुछ नहीं दिख रहा वैसे, मतलब जस्ट टेलिंग बस एक हफ्ता तुम वापस वहां से क्या हो रहा है?
पानी बोतल हाँ पीने।
ठीक है, लैपटॉप पर प्रेजेंटेशन उस पे, ठीक है। और मैडम को ये भी है, अगर आप बोलो तो इसको मैं डाउन कर दूं, नहीं तो अगर आपकी जो स्लाइड इस पे चलेगी ना, वो स्लाइड यहाँ भी दिखेगी, पीछे भी दिखेगी, ठीक है। और यहाँ से बंदा जो भी डॉक्टर खड़ा होगा वो यहाँ से इसको भी देख के प्रेजेंट कर सकता है, हैं? ये तो हो गया, अब मैं उसमें जाऊँ, हैं? प्रेजेंटेशन इन पे चलेगा,
एक पे माइक्रोस्कोप चलेगा, एक पे प्रेजेंटेशन चलेगा।
ये माइक्रोस्कोप इस पे चलेगा, इस पे आप जैसा बोलो मैं वैसा भी कर दूंगा।
ये तो धर्मंत्री और ना हाँ इसलिए तो मैं बोल रहा हूँ हमें ना जाएंगे नहीं पता कि मैं देख रहा हूँ आपको कौन सा पहले सोशल जाने से चले फिर
एक बार ग्रुप पकड़ ले उसके बाद जहाँ ग्रुप जाएगा वहाँ चले जाइए मुझे कहता है
ग्रुप तो है
मेरे से ही है अरे रेल बनी है हमारी
क्या देख रहे हैं
कहाँ तू ले गया तेरे
तो ये अपने पे भी तो लग सकता है। iPad मतलब ये स्क्रीन बन जाएगा कैमरा इसमें स्लाइड नहीं है, चेक करके कैमरा
बिल मेरे को 10 मिनट हो जाएगा।
Hello, mic check.
आपको ऊंची चेयर चाहिए? हाँ, ये लीजिए। भाई उसका रिमोट रख दिया हमने, रिमोट से उसको हेलो। अरे एलईडी वाला रिमोट है उसमें क्लैरिटी के लिए। हाँ हाँ वो हमारा है, ठीक है। इसका रिमोट सेट करा है। हाँ ये भी कुछ सेट हो गया। तो मैं बस साइड से आपने इस वाले को इसको मैं जा रहा हूँ कि वाली टीम के वहाँ पर लगा सेंटरिंग की लाइन है अच्छी। अब आपने इतना आउटपुट करके चेयर पर्सन से ये तो हमारा सारे लोग देखेंगे फिर इधर से। अच्छा वहाँ अब ये इस वाले पे कह रहे हो उस वाले में? इस वाले खाना खा लिया?
माउस वहाँ के आगे के माउस।
आगे के लैपटॉप में माउस लगा है। हाँ
सर को माइक दो। ये रहा।
So we are 1 hour late, aren't we? But we had good breakfast and we had good lunch, right? So now the workshop will start. This is the acute leukemia workshop. Originally what we had designed was 2 cases live gating and maybe if time permits 1 quiz. Okay? So the quiz will do extempo, but the live gating is the must, right? So Richa, where are you? Come. So let me introduce Richa. You all know Richa. And where is Shilpi? Ajay, idhar aao. Come, Shilpi, come. Where is Sujata? Come. And where is Himani? Come. बस हम लोग तो और कोई नहीं ना?
डॉक्टर सुजाता मैम।
हाँ?
सुजाता मैम।
आ गए सुजाता आ गए. So let me introduce that. Where is Deepa Rani? So we are the faculties in the acute leukemia. Professor Sujata, Professor Pathology, earlier in ESIC Faridabad, now in ESIC Basai. ठीक है? You all know me. You all know her. Shilpi is the master of ceremony. She'll be doing the live gating. She's the one who has established the flow cytometry in ESIC Faridabad, besides so many other things she's done. A great girl. Give her a big hand. And Himani, one of the best persons I know. She's going to show all the cases of leukemia. We are going to show you one BALL case, which she's going to present. And Shilpi is going to simultaneously do the live gating. And then she'll present a microgranular APML, which again Shilpi is going to do live gating. So the BALL case will be presented by Professor Sujata, and the microgranular APML will be presented by Himani. Both the live gatings by Dr. Shilpi. And Richa and I are going to give expert comments, and I'll ask Dr. Pati to give the final remarks. We are reducing the time of 1 hour to 40 minutes because we are fighting against time, right? We don't want you all to be awake till 6 o'clock. So let's start. Our chairperson is Dr. Vijay. Vijay, please come here. Vijay is pronounced as Vigai and not Jay. So be careful. He is Vijay with an I, right? He is professor and head of pathology of Banaras Hindu University, Department of Pathology, Institute of Medical Sciences. He is also the president of the UP Hematocon. He has introduced the Kashi Hematology Group. In Banaras, they love to call it Kashi. So he's introduced the Kashi Hematology Group. He is a joint, secretary of the Indian Society of Hematology and Blood Transfusion. He is a very, very senior executive member. And I wish him all the best and hope in the next 2 or 3 years we see him as director of the BHU. I'm very sure he'll become the director. Okay? And we are waiting for Professor Deepa Rani. She has not yet come, but we can't wait any longer. So, sir, with your permission, we are starting. Right. Come, Vijay. Yeah, you can take this or whatever you want.
Okay, it is there, sir.
Okay, fine.
So, very good afternoon, everyone. So, we start with the session of acute leukemia workshop. So, I will be presenting the first case. So, a 58-year-old male presented with complaints of generalized weakness and fever for 10 days. Hemoglobin was 4.5 gram per deciliter, TLC 27,000 and platelet count was 5,000. On peripheral smear, as you can see, the RBCs are microcytic hypochromic with few of them showing polychromatophilic picture. Occasional nucleated RBCs are noted. However, there is no evidence of schistocytes or any fragmentation of the RBCs, so there is no evidence of hemolysis as such. So the total leukocyte count is 27,000 as we saw, so it is increased, one can make out. So we are seeing large cells which are like blast, about 3 to 4 times the size of the lymphocytes mature. They have amphiphilic cytoplasm with cytoplasmic vacuolations in some, if you can appreciate. However, there are no OR rods. The nuclei are irregular with clumped chromatin, and there are prominent nucleoli in few with perichromatin condensation. The platelet count is markedly decreased, it was 5,000. So our impression on morphology is acute leukemia, likely lymphoid on the basis of the PS findings. So now I would invite Dr. Shilpi to come and show us the live gating of this case on flow cytometry.
Good afternoon, everyone. Before I start, I would like to invite 5 delegates on each of the individual laptops placed here. They can do the live gating as I do here. Any of you can volunteer.
How many laptops are there?
Sir, 2 laptops, one here, one there.
So आगे आ जाओ, इधर आ जाओ, we'll go behind. Come. 3 of you sit here. 3 of you sit here. 3 of you sit here. Come. Or 2. अरे भाई आ जाओ यार, ये हो गया है. आ जाओ. कहाँ उधर? ये आ गया. Visible cells, बैठा. So 2 है, ठीक है? Now to the line. इसको खोल दो. जल्दी जल्दी. हाँ लाइव मीटिंग शिल्पी कर रही है. शिल्पी you start.
Okay, so now you have already seen the history and the morphology of the case. So, and you know about the basics. So how we design the panel, how we select the tubes. So any flow cytometry is based on the morphology which you have. Once you have the morphology, you will decide your flow cytometry panel according to the morphology. So here we suspected acute leukemia, and first tube, as you all know, is the A-lot tube. So A-lot tube, in that you have these markers. So it has the cytoplasmic markers as well as the surface markers, which has been clearly spoken about by Dr. Tathagat Chatterjee and Dr. Richa. So now you want to analyze your singlets. What are singlets? Singlets are the single cells which are entering into the flow cytometer, and you do not want to analyze the doublets because your doublets will interfere with your gating and the acquisition and finally your interpretation. So here is my first gate. So how do you gate? So when you acquire any population in the flow cytometer, this is how your population will look like. So here is my forward scatter height versus forward scatter area. Here you can see you are getting two populations. So the one which is in line, those are your singlets, okay? And the one which has a higher area, higher forward scatter, those are your doublets. So you do not want to analyze your doublets. What do you want to analyze? You want to analyze your singlets. So how do you make a gate? Can you see this icon here? These three icons? So the first is a rectangular gate, second is a elliptical gate, and third is a polygonal gate. So polygonal gate are the best gates which you can employ when you are drawing a gate to identify a population of interest. So you click on the polygonal gate, you go to your selected population, and you make a gate around the cells which you want to analyze. So here I have gated my singlets. Then you click on the gate, right click, go to properties. You can rename this gate. So I am naming this as singlets. From the color bar, when you click on the color bar, you have a multitude of options, and you preferably choose a dimmer color for your singlets, because later on when you want to choose your blast, your lymphocytes or any other population of interest, you want to give a bright color to that population. So here I am selecting this. Are you able to follow? Vinay ji, can you please help them? Are you able to follow?
I'm not able to change the color for the singlets now. Singlets I labeled it as singlets.
You were able to label? Okay.
Take a right click. आप लोग का कोई प्रॉब्लम तो नहीं है? Done it?
Neha, help them. So, once you have your singlets, what do? Now you want to analyze the further population on singlets as well, only. So you choose your singlets, you go to drill down. In the drill down, you can choose what you want to analyze next. So here now, what do you want to rule out next? Any debris. Okay. Acha. See, these are the plots. As it was discussed in the morning, this whole thing is one plot. Okay, this whole thing. This individual gate is an individual gate. When you want to change the property of a gate, you have to click on that specific gate. And if you want to change certain thing about the plot, you have to click on the plot. So right now I'm clicking on the plot. When you click on the plot, your properties will have a property of the plot. So on the plot, what you can change is what type of plot it is. Like this is the contour plot. See, this is the contour plot. These are the properties of a plot. In the plot, you can go to the dot plot, you can choose contour, you can choose density plot. And you can see what type of plot it is. And again, I am going back to my plot. When you have selected a certain population, when you want to change the property of that gate, you have to click on the gate. So now I am going to my gate. Now my properties of the gate will appear. Are you able to follow?
So what will you name this now?
हाँ जी sir.
Go quickly to the next plot and magnify the plot and show it.
Okay sir.
Camera थोड़ा दूर है. Thanks. अब वो तो NDC.
Yes sir.
बड़ा कर दो.
So the next plot which I want to see is, I want to remove my debris. So what are debris? Like ma'am has already said, any of your platelet clumps, any of the RBCs which have not undergone lysis, they will appear as debris. So they will have low side scatter, low forward scatter, and you do not want to analyze your debris. You want to analyze non-debris cellular events, that is NDCE. Okay? So this is my NDCE plot on my side scatter versus forward scatter. Again, these are my debris. How do you make a plot? you select your polygonal gate and you draw the area. Is that clear?
So why hasn't she drawn a gate like this and like this? So what all falls here, here, here? So what all falls here, here and here? Grams here, normally here, and here. So what are mirror plots? Forward scatter, side scatter is seen like this. And if you see side scatter 45, it's ulta. 45 comes here. See, 45 and side scatter plot. 45 comes here, the mononuclear cells here, and the
in a norm. So after, like ma'am has said, in a normal plot, you will be able to identify your lymphocytes, monocytes, as well as granulocytes. Now comes your most important gate, which is the CD45 versus side scatter, which you are using for gating. Okay? So for the gating, here in my plot, I have changed the parameter to the dot plot. So you can see two populations here very clearly. For basic, I'll just remove these gates. So whenever you want to move the gate, abhi I am not removing all the gates because we have more cases to show and you have 3 workshops in total. So you can see clearly there are two populations. If you have a doubt, what you can do is, you can again go to the properties of the plot. In the plot type, you can select contour. Here you can clearly see there are two populations. One which has a brighter CD45 with dim side scatter, other has a dimmer CD45 with low side scatter. Okay? And if you want to cross check again, you can see the density plot of these two, and again you can see there are two different populations. Can anybody tell me which is the one which is having a brighter CD45 and dim side scatter? They are mature lymphocytes, correct. So they are your internal control for any kind of analysis which you are making, because without internal control it is very difficult to interpret any of the gated population or the population of interest. So once you have done that, the other thing which you can do is, you can go to the dot plot and reduce the number of events. So now you can clearly see there are two populations and not one. When you decrease the events of the dot plot, you can clearly see there are two different populations. No sir, it's the percentage of event which you go in the dot plot.
Dot plot events, sir.
So basically you go to the dot plot, you go to the properties. In the properties, plot type will be dot plot only, and in the drag down dot plot, you reduce the percentage events to 10.
And you can also increase the size of the selected population. So if you want to see small dots, you can select small. If you want to see a small population as large dots, you can select large.
So if you have a smaller number of population, you can go and make the dot size larger. So like in some cases when you have the blast percentage of 10% or maybe lower, you can increase the size of the population of the interest to large, so that you are able to recognize those cells easily. So once that is done, now I am again shifting to the small, and it is 100%. Now I am putting my populations, I am gating the population of interest. For gating, again you go to the same thing, you choose the polygonal gate and you gate your cell of interest. Alright? So these are the two populations. Those are the mature lymphocytes in blue, and the red you always give a brighter color to your population of interest. So blast we usually gate as red population. Alright? And these are grants which you have already seen in the morphology. They were very few in number. So also what you can do is, there are various patterns which you can follow. So if you have your tubes in the run pointer, whenever you change the setting of the gate, when you go to the other tubes, the setting will change. So what you can do is, you can make these plots unique. So for that, again, when you do a right click, you make that gate unique. So now even when you go to the next tube, this gate will not move. All right?
Okay, so Dr. Shilpi, one question. So the gate you have drawn around the 45 dim population. So how do you decide there only it is to be drawn and the blaster not falling in the glands or the lymphocytic or monocytic region? So the first gating should be slightly loose, and when you identify your blast later on 34, 117 or any other useful marker combination, then you can go back to this plot and replace your gate.
So ma'am is very correct. So basically, what ma'am is trying to say is you can always check the population where the expression is coming, and you can choose whether your blast is ending at a certain point or it is going above or below that point which you have drawn as a gate. So now coming to individual markers. So A0 we have like already spoken about. So it will have the cytoplasmic markers such as cytoplasmic MPO, cytoplasmic CD3, CD19. CD19 is not a cytoplasmic marker, but it is a lineage marker. So A0 you will have cytoplasmic markers as well as the lineage markers. So for B cell it will be CD19, for myeloid it will be MPO, and for T cell it would be cytoplasmic CD3. Plus we have CD34, which is the immaturity marker, nuclear TDT in the first tube.
What about cytoplasmic CD79A?
So that is also there, which is the B marker and the cytoplasmic. So CD19 as well as cytoplasmic CD79A. That is present.
Cyto CD3, Cyto CD7, Cyto MPO.
Cyto CD3, Cyto 79A and Cyto MPO.
These are the three cytos.
Yes, sir. And nuclear TDT.
Nuclear TDT she will be.
Yeah. So like here, so when you are choosing your plot again, so these are the plots which are already there, but if you want to draw a plot, these are the plot types which you can see in the top. So this is for the dot plot. So you click on the plot and you make a plot whichever you want to choose. So here you go to the properties, and in the parameter you will have to change the forward scatter to, the side scatter to linear. And when you want to change the antibody which you want to see, you can again click on the x-axis and you can choose which antibody you want to analyze. For example, here I want to analyze CD19 with MPO. So on x-axis I have CD19, on y-axis I have MPO. Now my properties you can see, it is placed on all events. To make it cleaner, what you can do is again click on the plot, go to the properties. Here is the parent population which you can choose. So we have singlets, we have NDC, and then I have gated glands, blasts and limbs. So first I am marking my lymphocytes. So here you can clearly see there are two population of cells, which are, one is 19 positive, one is 19 negative, and both of them are MPO negative. So the ones which are 19 positive are mature B lymphocytes, and the ones which are CD19 negative are mature T lymphocytes. So, and both of them are obviously MPO negative.
Dr. Shilpi, you can tell them here that when do we call a population positive and when negative, and what is dim, moderate, bright?
Okay. So the positive and negative is always in respect to the internal control. So your mature lymphocytes, so here this is my negative control, as in anything which will be beyond this population would be positive. But anything which would be here would be dim positive. And anything which would be coming near the bright CD19 or the mature B lymphocytes would be bright positive for that marker.
Can you put a quadrant gate?
Yeah. Yeah. So for this, you can also use the quadrant gate. So this population is your negative for both MPO as well as CD19. And a population which would fall here would be dim positive for CD19, and this would be bright CD19. So this helps us in analyzing and differentiating the cells as dim or bright positive. Ma'am, you want to add anything? Okay, sir. Okay.
From
MPO.
And she has gated lymphocytes. So can anybody tell me which population is seen in the LL quadrant? What there is only T?
So now quickly go and select the
Yeah. So now again I'm going to the properties, and now I want to include my blast in the population. So can anybody tell me what is the positivity or the expression of the blast for these markers? Bright positive for? And MPO is? So which kind of blast are you expecting these to be? Yeah. So what is the next marker you want to analyze now? Which one? Okay. Yeah. सर मोस्ट लाइकली या तो कुछ वो डेब्री होगा या वो ग्रैंड्स थोड़े से उसमें आ गए होंगे.
Can be backgated and maybe they are falling from here.
So what we can do is, like sir has correctly pointed out, that some cells are coming as bright MPO positive. So to see what is their expression or what are they, you remove your limbs, because sometimes when you put a further gate, sometimes they pick it up as in limbs, so you're not able to see where they are going. So now I have selected my population as blast. Now I'll backgate this population.
You can find events.
Yes, ma'am.
Find events.
So more, yeah.
But it's coming from the neutrophils.
Some of them which are overlapping in the blast gate, sir.
So that is basically a small population of brands. Further, your internal control that MPO is working. Because MPO positive आया तो MPO आपका काम.
So यहीं पे आ रहे हैं sir, but they are 0.69%. They are less than 1% of the event.
Sir, so another, if you are think that they may be part of blast, you can have a added marker 11B in this. If they are showing 11B, then they are neutrophils and not blasts.
Or maybe you have overgated the blast, you can put a tight grease and remove that. Yeah.
Remove them on a plot with 11B in the tube.
They constitute.
Number, as you said, less than 1%.
It's less than 1%.
So what you do now is, you now gate the blast against CD34, so that you confirm it is a blast. So you are now replacing the anti-MPO in the y-axis with CD34. Am I right?
Yes, sir. So when you want to analyze further marker, what I have done is, in place of MPO, I have taken CD34. And you can clearly see that the blasts which were 19 positive are bright positive for CD34. Are you guys able to follow?
Are you all understanding? Yes. Each one of you? So now you got a 19 positive, 34 positive co-expression of blast. So we are very clear that they are blast. Right? So that's the first step.
To show where they are falling exactly. They will be pure blast.
Okay.
We are still in the first view, right? We are still in the first view. We have only used one cytoplasmic marker, which one is cytoplasmic MPO. Now we are going to use the other cytoplasmic markers, right?
And, sir, like ma'am said, when you want to confirm your blast population, here I am backgating my blast.
Backgating is finding out further. So there, you see the same color, purple color is coming there. So that confirms further that you went the blast. Backgating is, उसको दोबारा gate करके you are seeing your original form.
Is that concept clear?
Perfect. So carry on. We got another case.
Yes, sir. Now I want to see the expression of cytoplasmic CD79A. Again, you can clearly see that CD19 positive blasts, which were CD34 also positive, are bright positive for cytoplasmic CD79A. And your mature B lymphocytes are falling at the same place. Alright? Now coming for the expression of CD10. So again, this is, what does it become?
Ask someone, wait. Yeah, please stand up. Please answer the question. What has she gated now? What is there in the y-axis? What is there in the x-axis?
Y-axis there is CD10.
Yes.
CD19.
CD19. What does that show?
10 is Kala.
So there's a co-expression of the blast for both CD10 and CD19, right? You can explain.
So it has, it is a Kala positive BALL most likely, okay? So the blasts are positive for CD19, CD10, cytoplasmic CD79A, as well as 34 is bright positive, alright? Again,
Dr. Shan, can this be normal population, hematogone stage 1? The tube to confirm that these are blasts, okay?
More importantly, what was the smear? Peripheral smear or bone marrow?
Peripheral smear.
Do you get hematogones in peripheral smear? Yes. Autophagy overrules everything. Next.
And always you have to analyze your cytoplasmic CD3. So here these are your bright T positive and this would be considered negative, alright? So positive or negative is always in respect to your internal control populations.
I think so, fair enough. You want to show TDT?
Oh yes, yes sir.
How does TDT stain on IHC? Nuclear. So you got a very good TDT stain on the flow. Can anyone interpret the TDT in the y-axis, right?
Yes sir.
Again CD10 in the X?
CD19.
CD19, yeah. So CD19 is constant. So what does it show? Negative. Your internal controls कहाँ है? Quadrant gate. शिल्पी, quadrant gate. आप बताओ. So TDT positive, 34 positive, 10 positive, 19 positive, cytoplasmic 79 is positive, cyto MPO negative. What's your diagnosis? So now we can call Dr. Sujata for the explain the case. Because we will now move to the second case. Sujata quickly explain the case and you will see a fascinating finding in this. So that's why Dr. Sujata is explaining this. This is a fascinating finding in this. Yeah, carry on.
Okay.
So Sujata, you're going to show us the flow report. This is how the flow report comes from our department. So the Harit slide is PowerPoint. So you can show the gist of the report in the next slide.
Yeah, this is how the flow report comes, as sir is pointing out rightly. So it's not very clear because we have tried to take a screenshot of that. Anyhow, so you can see all the markers are there, then the intensity, whether it is bright or moderate like that, the next column, and the final column says whether it is positive or negative. And finally, we give the impression and if any advice like cytogenetics, etc. If it is not readable, so I'm just trying to read it out for all of you. So moving to the next slide, the flow gist. Now, as Dr. Shilpi has rightly explained, and sir and Dr. Richa has intervened and tried to explain to you about this case, and all the markers are discussed. So let us quickly recapitulate what all positivity we saw. So before that, we saw that 70% of the cells were gated on CD45 versus side scatter. So these were the blast. These cells showed dim to moderate CD45 positivity and dim side scatter as expected for a blast, as we have read since morning. Cells were bright positive for CD10, CD19, 20, TDT and HLA-DR. And also the cells were bright positive for cytoplasmic CD79A. So all these are confirming that the lineage is B cell. We can also see that there is no positivity for cytoplasmic MPO, ruling out myeloid differentiation, and also there is no positivity for cytoplasmic CD3, ruling out our T cells. So
very importantly, she has put one marker, CD22.
Yeah, CD20.
Anyone? This has been explained by Dr. Richa in our
Yeah.
first class.
Targeted therapy.
Which is the targeted therapy?
And
Twister. Whoever says is correct.
sir only will tell us. So what is it? Okay.
It's a sum web.
Okay. Okay, so that was a nice humor. So cells were moderate positive for 38, 22 and CD123. So what are the importance of these? 22, sir already said it is a B cell marker, and now for targeted therapy we put it in our tube. 38 is an activation marker, and 123 again is a marker which is present in the stem cells and as well as all the precursor B and the T cells.
Richa, you'd like to comment on CD38 on the blast?
So moderate positivity of what? 38, 22, 123. So 123, can I see the plot? Is it overexpressed?
So it is moderate.
Okay. So 34 and 123 co-positivity is an aberrancy, helpful for MRD. And CD38 is positive, moderate. This will be a difficult one because 38 dimmer is more helpful in MRD detection. 38 will fall where your normal hematogons fall in this case.
So remember, you are not only diagnosing the case, you are also making a template for your future MRD with all these aberrant expressions. So your diagnostic panel is also helping you for your future MRD, right? Okay, next.
So now we also advise for cytogenetics because in this institute we have RT-PCR. Sir only has designed the lab.
So this is a beautiful slide for Varagas.
No sir, 1 second, I'll try.
Could you just
Zoom.
read this report? Yeah, that's it.
Is it okay, sir?
Yeah, perfect.
Okay. अच्छा slide show कर. अभी और छोटा हो
So it's pH like amplification signals in ABL1. So you'll have to check further with what is it? Is it really amplified or is it translocating, breaking apart?
Okay. So this is a very interesting thing. Though the ABL is a housekeeper gene, but when you talk about Philadelphia live acute lymphoblastic leukemia, which may be constituting 6% of the childhood ALL, so they are genetically and somatically similar to a Philadelphia positive ALL, except that they don't have the BCR ABL1 tyrosine kinase or the translocation 9. Would you like to tell something more about the?
Yeah, they may have this BCR ABL1. We have the typical translocation of 922, but the Philadelphia-like chromosome, they will not be fusing with the BCR part. ABL will not fuse with BCR, but maybe with some other genes, and so they may be producing a protein similar to tyrosine kinase, but not exactly the same.
So the Philadelphia-like ALL will respond to imatinib upfront, along with the BFM protocol. That is one. And if you have the JAK2 mutations, which are also very common in such cases, you can get solid anemia. So now we are going beyond the therapy. Now we are getting targeted therapy based on the flow cytometry and the molecular.
So in this particular case, as you can see, we have all these fusion genes negative, E2A, PBX1, TEL, AML1, MLL, different fusion genes, AF4, ENL and AF19.
Lineages, these days it's called KMT2A. पहले हम लोग बोलते हैं MLL gene, 11q23 बोलते थे. इसको अभी we have now divided into so many AF4, AF9, ENL, these are all monocytic genes, and they have got bad prognosis. Your flow and your PCR molecular mutations are giving you such a good diagnostic and a prognostic aspect of this case.
Yeah, one can go for the therapy based on these markers. Then yeah, sir. I don't think so,
This
sir.
was a wonderful slide. Which are your comments? So this is the childhood DALL. Remember, boys and girls, childhood DALL is a curable disease. We are now going to cure this disease with so much of therapy. In St. Jude's, 95% of childhood ALL with the age of 1 and 9 years, that's the case where I trained after my DA, is they cannot accept the death of a child within 1 and 9 years. These are called the standards, right? So we have to be very thorough in our flow, in our molecular, in our diagnostic panel, so that sooner we start the treatment. Correct? Case 2, Himani. Himani is senior resident, yeah. Yeah, this was 50. This is talking childhood. This is an objective case. But this case, this is your
Yes, sir.
category.
Okay, good afternoon, everyone. So, after such informative session and energetic session of acute lymphoblastic leukemia, let's start with our case 2. So, clinical presentation is 41-year-old male presented with complaints of hematuria and black-colored stools for 2 days, and on examination, patient is having moderate hepatosplenomegaly. So, lab investigation reveals hemoglobin of 10 gram per dl, WBC 25,900, and platelet count 21,000. So, when you look in the peripheral smear, as you can see, starting from the RBC morphology, it is predominantly normocytic normochromic RBCs, and fair number of macrocytes are seen with occasional polychromatophils. Occasional fully chromic teardrop cells are seen, and there are no schistocytes. So let's move on to the WBC series. As you can see, leukocytosis is there, and in the DLC, there are 90% hypogranular promyelocytes that are blast equivalents with moderate amount of cytoplasm. Large binucleated cells are seen on the PS along with occasional multinucleated cells. Some of them show prominent granules in the cytoplasm interspersed with occasional myeloblasts, and mature lymphoid and myeloid series are sparse. Platelet count was 20,000 on the spear. So final impression was acute leukemia, likely acute promyelocytic leukemia. So flow cytometry was advised on the peripheral blood. And now I would request Dr. Shilpi ma'am to demonstrate the live gating.
If anybody else want to come on the laptops, okay. So here again, when you have to start, which is the first gate you want to see? We just discussed singlets. So you will analyze your singlets on which parameters? Correct. So when you acquire any of the samples for the flow cytometry, this is how your population will look without any kind of gates. Always it is always better to have assays defined in your flow cytometry. So you can have an assay defined for acute leukemia, CLPD, PNH, or whatever you are doing in your institute. Once you have your assay defined, whenever you are putting or running the samples, you can just go to that assay and you can copy that. So you don't have to make the gates again and again. Alright? So here you have your first plot as forward scatter height versus forward scatter area. So these are my singlets and I have ruled out my doublets. You can clearly see that these are the ones which are having a higher area. So those are your doublets and you do not want to analyze the doublets. So once you have gated your singlets, what do you do? What is the next thing you want to remove? Yeah, you want to remove your debris. So what do? You click on the plot, you go to the properties, you go to drill down, because you want to do your next step of analysis on the singlets. So if you do not choose your singlets as your primary population, you will be analyzing your singlets for the NDCE as well. Alright? So once you have done that, you remove your debris from the forward scatter versus side scatter area. Is that clear? So here you can clearly see these are my debris, and what are these population of cells? They look like? So the ones which have very low forward scatter and very low side scatter are likely to be debris. So you don't gate those cells. Like here I am not gating these cells. So for example, so when I choose my population, these would be debris. So I select my gate on this population only, which has a higher forward scatter and a higher side scatter. And now when you want to analyze your population of interest, your gating plot would always be, you gate the cells with CD45. So CD45 versus side scatter. So here I have gated my cells and I can clearly see two population of cells. Can somebody tell me what are these cells? Lymphocytes. And what are these cells? Yes, sir. So what is the difference between the blast of this case vis-a-vis blast of the previous case? Comparatively, they have high side scatter. Yes, sir. So based on the morphology, you always have to analyze your flow cytometry in accordance with your morphology. So this is falling in collaboration with my morphological findings. So now
Do you
I
want
put
to
my
comment on whether this is teardrop pattern?
बहुत अच्छा teardrop नहीं है actually.
Say whether it is a maturing glands or not here actually.
मतलब it's like not very good teardrop.
So essentially your most important plot is side scatter versus CD45. ठीक है, आप जितना भी लाइनें ढूंढो जब तक side scatter CD45 आपको नहीं आता. So all you can say is these are complex granules because the side scatter is high. ठीक है. Now those complex granules you can speculate what they are.
So here I have gated my lymphocytes which are bright CD45 and dim side scatter, and these are my blasts which have dim CD45 but side scatter is dim to moderate. It's not very high but it is not as low as we see in cases of ALL. But yeah, not as high as we see in APML classical also. So
That's the hint she has given. Big hint. Next.
So going on to my individual markers, here I have gated the cells with cytoplasmic CD79A and NT-MPO. Can somebody tell me what is the interpretation of the blast for these markers? So regarding quadrants also there are two school of thoughts. So one thought is that you should always put a quadrant gate and analyze the cells in respect to those quadrants. But there are various theories and they say that quadrant gates are not mandatory. So what you can do instead is you can gate your internal controls or the lymphocytes as your gates which will be acting as your negative and the positive controls, and you gate your population of interest with respect to those. Alright. So here if I see, so some of the blasts are coming as MPO, they are MPO positive, but definitely cytoplasmic CD79A. Yes, ma'am. Yes.
The granularity of these cells is very high. I will not speak about background fluorescence much, but autofluorescence is one of the components of background fluorescence. And this CD79 which is coming, whether it is truly positive in this population or just a part of background fluorescence, you can make out only if you see the unstained tube, which is processed after, which is required after perm, because this is after permanentization that you're seeing this. Okay. So unstained tubes are important to check your positivity apart from what Dr. Shetty said in the earlier contents.
What is formalization? Madam has just said. You are breaking through the surface and going to the cytoplasm for the cytoplasmic stage. Formalizing stage, right?
And what ma'am said is very right, and you always, when you are starting your flow cytometry, you should always have a cytoplasmic unstained tube and a surface unstained tube. So unstained tube means what? Unstained tubes means what? Are you putting any antibody or no antibody? CD45 is there. Okay, so only CD45 with no other antibodies, and you will choose your positive or negative with respect to that tube, both for cytoplasmic as well as for surface markers. सर हमने इनिशियली 100 सैम्पल्स में हम अनस्टेन लगाते थे, अब हम सब में नहीं लगाते हैं. But when you start, you should ideally always put. And whenever you start having some problems, again start using unstained tubes. So
idea is this much fluorescence can be part
Yes.
of the internal complexity of the cells themselves. The CD79 may be part of autofluorescence.
The internal structure of the cell is creating the autofluorescence. And that is unique in some type of PPD also. Okay, next.
So now I am analyzing my CD19. We can clearly see the blasts are negative for CD19. The 10 is also clearly negative. Coming on to my immaturity markers. So 34, a population is dim positive. So I would like to see 34 in my other tubes as well. No, sir, x-axis. Yes, ma'am. It is almost, and they are almost 6%, and they are falling in the blast gate only.
So most of the blasts are negative, only 6% positive. So you have got a 34 negative blast. So what do you think it is? They are not blasts? No, you just can't say APML like that. Be very careful of your diagnosis. So they are 34 negative. We still don't know whether they are pro-myelocytes. So they are 34 negative.
TDT is clearly negative, 19 was negative. Cytoplasmic CD3 is also clearly negative. Now coming on to my second tube.
So before that, what are the leukemias? Obviously you have seen the morphology. You have seen the morphology, no? Nothing normal in the morphology. So you have got some sort of a diagnosis. When I was training under Dr. Pati, in 2 cells used to give a diagnosis of APML. There was no flow growth test. That used to start the treatment within 3 years. Okay? Now, what are 34 negative blasts? Name. Besides APML. Maybe pro-monocytic leukemia. Anything else? NPM1 positive AML, NPM1 FLT3 ITD AML. There are many of these entities which are 34 negative. Just don't think that खाली APML में 34 negative हो रहा है. The microgranular variant may be positive for 34 also. So सब mathematical चीज नहीं है. आप पहले morphology देखें, उसके बाद आप flow identify. But remember 34 negative you get an NPM1 mutation, NPM1 FLT3 ITD mutation, APML, AML, F5A, all those things you can take.
So in the second tube, we have the myeloid markers. What are the myeloid markers? Yes. Okay. Yes, sir. So in the second tube, we have CD13, CD33 as well as the monocytic markers with CD117. Plus we have HLA-DR. Alright. So here you can see the blasts are clearly brightly positive for CD33. CD13 is also positive, but there is heterogeneous expression of CD13.
So what is the difference in the expression of 13 and 33? Where do you get maximum expression of 13? Where do you get maximum expression of 33? Which you can explain to them.
So there was one comment that maturation
सकता है. So can somebody tell me, are these neutrophils or not? Are they maturing granulocytic series cells or not? Or they are just stuck at one position? So what is the pattern that is seen in 1333? 13 is brightest. You see 13 in normal myeloblast. After that, it is very bright in promyelocytes. Then at myelocytic stage, it dips.
Yes.
It is very less in myelocytes. And then it increases again on metamyelocytes. And neutrophils, again, it goes bright. Okay, so there are certain patterns on 1316, 1311B where you can see maturation. So here they are quite stuck at one place in CD33. They are not showing any mature. So 13, they are heterogeneous, but 33. So 33 similarly can have, they are not, so normally also granulocytic series cells gain some 33. Monocytic lineage gains further more 33. So there is always a pattern, but here it is stuck, homogeneous 33. This is abnormal.
And you are always supposed to see whether the expression is homogeneous and heterogeneous with respect to 13 and 33. Because that will help you in identifying, like ma'am said, whether they are the normal maturing granulocytes or maturing monocytes or there are some abnormal cells.
So at this stage, we still do not know what the diagnosis. Are we dealing with AML F3 variant? Are we dealing with AML F4? We don't know. Okay? So let's analyze further.
So this is 117 again, it is showing a heterogeneous expression.
So if they were neutrophils, then 117 should not
Have been there in such a large chunk of population, right? 34 117 positive cells and promyelocytes maximum would have been positive. But your majority of the chunk is, one is stuck at 33 expression and one it is showing good amount of 117.
Now, a very important marker, HLA-DR, which is?
Now, ask this question. HLA-DR is a very, very important graph. What is it? Positive, negative?
So the concept HLA-DR is not a maturity marker. It's a antigen presenting, it's present on antigen presenting cells. Okay, so B cells. But apart from that, myeloblasts shows DR. Stem cells can show DR. Neutrophils do not show DR. Promyelocytes also do not show DR. But in myeloblasts show DR.
It's not an immaturity. 34 TDC is one
HLA-DR?
Some monocytes will normally present with that, so it's not an immaturity.
So unlike 34 and DDT, this is not an immaturity. Okay? This is an antigen presenting cell. Now see the expression of HLA-DR in the dense population, the red population. What is the x-axis showing?
Sir, x-axis is HLA-DR, y-axis is CD33.
So you can even again put a quadrant and find out this is negative, right? So you got bulk of the population as 34 negative, few population positive, and HLA-DR negative. Anti-MPO strongly positive, 1333 there is an aberrant type of an expression, right? Okay.
And 64 also is clearly DIM positive.
So what is the 64 a marker of? So we are still stuck up. Okay, next.
34. So now we can go to the next tube.
So next tube is on T tube.
Yeah.
So now you tell very frankly, when you got this case, what diagnosis did you give on morphology and what diagnosis did you give on flow?
So on microscopy, we thought of acute leukemia, likely AML. On flow cytometry, we had HLA-DR negativity. We had 34 negativity with a small population which was DIM positive. It was MPO positive, CD117 was positive, and 33 was bright, 13 was heterogeneous. So we thought of APML, microgranular variant based on the morphology and the flow cytometry. And then we ordered RT-PCR.
Let's be very truthful. On the morphology, we said we cannot deny the start of treatment of APML. We started this patient on ETRA, it was Friday. We started this patient on ETRA, we said this is microgranular variant. Flow also we said, okay, no problem, morphology, ये है ETRA start कर दो, तो patient not over time. Okay, ETRA start कर दो. On Monday, we got them a very nice report. Who is showing that report? Himani. And with this, we'll end the first of this document. Himani, show the report. So this was what we said. We started the ETRA, right? Show the gist of the report.
Yes.
That is, now everyone knows the gist of the report, right? Now you come to the molecular mutation analysis. This is the gist of it, right? Clear? Now the molecular mutation analysis.
So next we have advised the molecular mutation by RT-PCR, and that's what we got. RNA markers AML1 ETO, rest are all negative, but PML RARA was positive. So final diagnosis was given as acute myelogenous leukemia positive for PML RARA mutational marker.
So once again I'm reiterating, you don't know anything by becoming old and so we gave this morphology report. Lot of confusion. Flow में क्या हो रहा है क्या हो रहा है हमने morphology में देखकर उसको start करा दिया and thank God we fast tracked a PML RARA report. In 2001 we never had these opportunities. Dr. Pati, Dr. Renu Saxena, Dr. Pati I remember 2 cell देखते थे pancytopenia. APML classical has pancytopenia. उसमें 2 या 3 सेल हमारा hypergranular देखकर हम लोग APML का treatment start करते थे. ठीक है? So we have come from that and we still stick to the morphology. This report came after 4 days. That is also a very fast report for a government hospital. हमारे hospital. Thank you very much. We have finished the first session. Yeah, there is a question from a young lady. Yeah, take a
मैम इसमें CD14 and 64 पॉजिटिव क्यों आया?
CD14 and 64 were DIM positive.
Why?
Why?
Because these are.
Chromocytes normally express 64, so that is not a problem. 14, I'm not seeing the plot, but
They're dim.
Dim positive.
positive it might again be due to autofluorescence.
Likely autofluorescence.
Putting in the chromocytic region level like teardrop. And one very helpful simple cytometry
To help you differentiate the case of monocytic leukemia versus APL.
Because in monocytic, it will be dim positive or negative. That is very important. That's why cytochemistry is so important. What is chloroacetate esterase? Have you ever heard of CAE? And what would the report of this be in APML microgranular variant? Please read up. You must read DESI. ठीक है? सीधा flow में मत आओ. Morphology, cytochemistry from DESI and then flow. Follow the sequence. There is no shortcut. What is the difference between NSC with F in AML M5 versus NSC with F in APML? ये सब MD में मैं पूछूंगा तुम लोग का एग्जामिनेशन. कान पकड़ूंगा. NSC with F, fluoride sensitive monocytic, fluoride resistant APML. Remember these things. We have been taught like that by Dr. Pati. Okay, thank you so much. We have finished the first session.
So we can wait for some time. MRD session is not over.
You know, MRT session will never get over. Go and tell them. जल्दी करो. Please go.
Sir, आप बोल दो.
मैं तो बोल कर आया, मेरा बात नहीं सुन रहे. अभी डॉक्टर पति को हम भेजेंगे. डॉक्टर पति बोल. No, it can't. We just can't.
They are in between the session.
No. See, we have to. You get the next batch. 4 is free. ठीक है? This one. Mukta. We have to abort the session. ऐसा नहीं चलेगा. We have to finish. Because we have to keep time. अच्छा wait. डॉक्टर पति जब चले जाएंगे हाँ तो बाकी गाड़ी जी वो या डीटी की गाड़ी
Any questions? Anybody has any questions? Abhi wait for 5 minutes because you have to go to Shishmas.
So wait a second. You all are lucky that the next batch has not come. Just hear some pearls from Dr. Pati because he's leaving. So please listen to him.
See, just now we heard the flow of 2 actual cases of acute lymphoblastic leukemia and a representative case of promyelocytic leukemia. See, the difference one is because as already Dr. Tathagar said that morphology, already you have seen the case and that differential count also know, that you know the blast count. That's important because straight away you are, so that's one comparison you can see that the blast count is suppose you have seen that 80-90% and in flow also it should be, even if it is dilute, still it will be around 50%. So the major population you are seeing, so that clarity one is there. And also the technical point of view, all that you have to know, have to understand how really to interpret each individual cell with your internal control, like lymphocytes, how they are behaving, so that whether it is the brightness is there or they are dim positive or negative, that clarity one has to be there. Second, like the APML, one is the morphology that you have seen and the percentage, that's one aspect. But another thing also one should know that as per the knowledge that the final diagnosis will not come from flow. So there are few conditions one should be aware where molecular diagnosis is a must. Many places people not even, yes, as you say that we take the advantage or we say that we don't doubt about it. I don't think that's actually wrong. That's, I think, I believe also that. But literature-wise that you have to, that you can say, always say suggestive. So flow, one is the AML, the majority cases, flow is the final lineage. Whereas APML is the one exclusion where the lineage will again final the molecular, which already Tathagat has done and confirmed.
Thank you, sir. Thank you all. Now you all go and replace the other people and tell them to come back quickly. Otherwise you will be here for dinner. And we have not catered for dinner. They're laughing.
चलो be seated, all of you. Come. आज यहाँ से सीधा हम लोग डिनर देंगे अगर मौका मिले ललिता हाँ अरे ये तो हो गया है अच्छा ये वाला अच्छा अभी नेक्स्ट
It was over, but it was over.
Yeah, but where are they?
मैं देख के आती
आ गया होगा हाँ इसमें ना सेल्फी सबसे कम से करने वाला हमारा बेस्ट टीचर द स्पीकर सबसे कम स्पीकर सो गुड so we are about to start Please hurry up. Shall we start?
Okay, good afternoon, ladies and gentlemen, boys and girls. Hey, Hasi Band. So, we have just finished the first session. Jaldi aao andar. Please come inside. We are starting. Please come, jaldi aao. यहाँ इसके पीछे आ जाओ सीधा लाइव गेटिंग मोनिका क्या बोलते हैं वंदना इनके पीछे आ जाओ सीधा लाइव गेटिंग Are we ready? We are starting. So, good afternoon, boys and girls, and ladies and gentlemen, of course. So, Dr. Shilpi, please come here. She is going to do the live gating of 2 cases and simultaneously you all will be doing the live gating, right? Professor Sujata, please start your first case.
So good afternoon. We start with the second round of the acute leukemia session. We start with the first case. This is a case of a 58-year-old male complaining of generalized weakness and fever for 10 days. The lab investigation shows hemoglobin is 4.5 gram per deciliter. Total count is 27,000 per millimeter cube and the platelet count is 5,000 per millimeter cube. The peripheral smear finding, as you can see, the RBCs are predominantly microcytic hypochromic with few occasional polychromatophils. Occasional nucleated RBCs are also noted. Can you appreciate? However, there is no evidence of any schistocytes or fragmented RBCs, that is, there is no hemolysis as far. The total count, the differential count, the WBC counts are raised, and on the differential we see most of the cells are large.
Just focus it here. Yeah. Now don't move it, just focus it here. Yeah.
These cells are large, which are about 3 to 4 times the size of a lymphocyte, having amphiphilic cytoplasm with cytoplasmic vacuolation in some, of course not in this one. However, there are no OR rods. The nuclei, as you can see, these are irregular and the chromatin is clumped, showing prominent nucleoli in few of them. The platelet count is decreased. So the impression or morphology is acute leukemia, likely lymphoid based on the morphology. Now I would request Dr. Shilpi to come and show you the live gating of this case for flow cytometry.
Hi, good afternoon everyone. So I'm assuming now you are aware of what are singlets, what are NDC. Okay, so can you tell me how to get singlets? Yes, that is correct. And why do you want to see your singlets? Yeah, correct. And yes, and once you have identified your singlets, what do you want to do next? You want to remove your debris. So, following the same things. Here you can see in the forward scatter height versus forward scatter area, I have removed. नहीं स्क्रीन में नहीं आ रहा अच्छा अब आ रहा है okay. So here you can see in my forward scatter height versus area, I have removed my doublets. So once you have gated your signal, you can replicate what is being shown. Okay. So this also you must have been told that you can click on the plot on this population of events. When you click on drill down, so you are selecting your singlets and these singlets you are removing your debris. So here I am selecting, for example, forward scatter area versus side scatter area. So the thing which you will click first will go on x-axis and the one which you will click on the second time, that will go on your y-axis. Here you can see everything is going here. So for correction of that, you again go to the properties of the plot, you go to the parameters and you select your scale to linear for both side scatter as well as your forward scatter. So here now I have removed my debris and these are my NDCE. Is that clear? So once that is done, again when you want to analyze your population, you will drill down and then what is your gating marker? Yes. So now when I drill down, I will select my parameters with the CD45 versus high scatter area. So here you can see two populations. Now to confirm that these are two population of cells, again I go to my plot properties and I select contour plot. Here we can clearly see that these are two population of cells. One is having a brighter CD45 with dim side scatter. The other population is having a dimmer CD45 with dim side scatter. So what are these populations? Which is the one which is having a higher CD45? Yes, and the other one is likely to be blast, which is in accordance with your morphology. So you gate them on the polygonal gate, which are these three are the gates which you can select. So first is the rectangular gate, then is the circular gate, and then is the polygonal gate. In the gating, the most useful gate which you can employ is the polygonal gate. Now, which markers do you want to analyze first, considering the morphology which you have already seen? Pardon me? Okay. So, usually we have the A-law tube as the first tube. What is A-law tube? Like Richa ma'am has already told you in the class, is the tube which has the cytoplasmic markers and the lineage markers. So what is the lineage marker for myeloid cells? Cytoplasmic MPO. For B cells it is CD19, and the cytoplasmic marker cytoplasmic CD79A. For T cells it is? Huh? Yeah, which CD3? Cytoplasmic CD3. Okay, and we also have CD34 in this tube. Okay, so on my gated blast, I have gated this with, I want to analyze my MPO with cytoplasmic CD79A. So first you select, when you go to the plot properties, you can select the parent population. Here I am selecting my limbs. So what are these cells? So what is this population? What is the population in pink? It is gated on MPO versus cytoplasmic CD79A. I am asking you what is P1?
Positive for 79. Where is it coming from? So what is B cell?
And what is this population? Is it MPO positive? What is the basis of you making it myeloid? You can be wrong, just keep on guessing. See, abhi mera population kya hai? What is the parent population?
Lymphocytes.
Limbs, right? And these are my B lymphocytes. What are these likely to be?
T lymphocytes.
So these are mature T lymphocytes. Okay. Now I select my properties to include the blast. So the blast are cytoplasmic CD79A positive, heterogeneous positive for cytoplasmic CD79A, and MPO is negative. All right.
So for the students, what is dim, moderate, bright positivity?
Yeah.
Here what will you label the positivity as? It is reaching, starting from ahead of negative population and reaching up to the normal intensity of the B lymphocytes. So it is covering at least 2 logs, more than 2 logs. It's a heterogeneous expression. Okay, moderate would have been equivalent to the internal control, and bright would be brighter than the internal control. Dim would be anything between these. Okay.
So now I am gating these cells with the CD19. So what do you think is the expression of blast for CD19? It's bright positive because you can see the mature B lymphocytes are falling over there. In fact, my blasts are brighter than the mature B lymphocytes. Now, which marker do you want to analyze next? Okay, CD10. Anybody before CD10 you want to analyze something? Okay, that would be to rule out whether it is B or T. CD10 is bright positive. So there is bright expression of both 19 and 10. So it becomes, what is CD10? Kala positive, common ALL antigen. But we have not analyzed CD34. So if you have not analyzed CD34, can you be sure whether they are mature or immature? So here you can see that CD34 is also bright positive. And when you see the T markers, so these are my mature T lymphocytes, these are my B lymphocytes, mature B lymphocytes, and my blasts are falling here, which are clearly cytoplasmic CD3 negative. Some of the channels have more fluorescence, so the negative and positive, you have to also see the fluorescence of the channel or the autofluorescence. Okay, but the bright positivity is your of the internal population, that is of the lymphocytes, will be your internal control for the positive and the negative population. So TDT also you can clearly see it is positive. So second tube which we use is for the myeloid markers. So 13 and 33 both are clearly negative. 34 we have already seen is quite bright positive. HLA-DR is also bright positive. CD38 is heterogeneous. These are the similar markers. Richa ma'am, do you want to add on anything? Any take home messages for BLS? I'm almost over.
For BAL, this population in CD45 site starter, you saw it's in CD45 positive. So where else can B blast fall in this plot? Move on to the row. This is still where else? So sometimes, thinking that these could be deadly, B blast could be missed. If they are not this population, if there is nothing here and you have some cell here, this diluted manner and a few blasts, you should always check this population for 1910 possibility so that you don't miss out on B blast. Normal, so you have to know the normal intensity of positivity in normal population. So for example, CD19 is moderate on B lymphoid blast, moderate intensity. It can be brighter in some lymphomas or leukemias. For example, CD38 is moderate intensity in T cells, NK cells, but it is bright, normally it is bright on plasma cells. So you know normal. From that you say comment on abnormal. So if the intensity of 19 is more than the normal B cells, it is brighter. If this intensity of 38 is more than the normal, near the plasma cells, it is bright 38. Otherwise it is moderate or dim. Okay? You have to know the normal intensity of antigens on your normal cells in the marrow or blood.
And just to reiterate what ma'am has already told you, this is another case of BALL only. Here you can see there are three populations in the site scatter versus CD45. So you are not supposed to just rule out these cells as debris. When we gated these cells, these were my blasts. So these are the blasts. So the negativity of the CD45 does not make them debris. You have to always interpret flow cytometry as per your morphology. Now coming on to the next case, I would request Dr. Himani to present the next case.
So what is the full form of ICC?
International Consensus Classification.
Yeah. What is the controversy? नाम तो क्या है बोल दिया? No, I am still
Main controversy.
Main controversy है. नाम तो जान लिया तुमने impression market. but है क्या? हैं? चलो.
Okay, so
Which group wrote 2008 and who are the authors in ICC? You go and find out, you know the controversy.
Okay, so after having the learning session for the acute lymphoblastic leukemia, let's come towards our second case. Clinical presentation is 41-year-old male presented with the complaints of hematuria and black colored stool for 2 days. That means he is having some bleeding tendencies. On examination, patient is having moderate hepatosplenomegaly. So lab investigation reveals hemoglobin of 10 gram per dl, WBC 25,900 cells per cubic mm, and platelet 21,000 cells per cubic mm. So when we saw the peripheral smear, first we comment on the RBCs that were predominantly normocytic, normochromic, and fair number of macrocytes were seen with occasional polychromatophils. And occasional fully chromic teardrop cells were seen, and there were no schistocytes or no evidence of hemolysis. In WBC series, there was leukocytosis, and on DLC, 90% of the hypogranular promyelocytes were seen. These are blast equivalents with moderate cytoplasm, large binucleated cells along with occasional multinucleated cells are seen. As you can see on the peripheral smear, some of them show the prominent granules in the cytoplasm interspersed with occasional myeloblast. Mature lymphoid and myeloid cells are sparse, and platelet count was 20,000 on smear. So impression given was acute leukemia, likely acute promyelocytic leukemia. So we advise flow cytometry on peripheral blood, and that will be demonstrated by Dr. Shilpi ma'am. She will demonstrate the live gating on this case.
Okay. So coming on to the next case. So like we have already seen, first you want to get your singlets, then your NDC. Are you able to follow? If anybody else wants to come on the laptops, you can shift. Do you want to come? No? Okay, you can carry on. Okay. So once you have gated your singlet, then you will gate your NDCE, and then you will have your gating channel. That would be side scatter versus CD45. So here you are having primarily two population of cells. So can somebody comment on this population? Okay. On morphology, how many neutrophils do you remember seeing?
So the question is whether there are any neutrophils or not in this. There are cells which have granules but not as much.
So how can you say if there are any neutrophils in this plot? You can't. Abhi you cannot comment.
And another thing which you can do is you can again go to the properties. You can see the contour. Sometimes it will show two different populations, sometimes it will not show. But that is some way of delineating if any mixture of population exists within a dense population. So at present, I would gate both of these cells as both of these populations as a single population. And later on, you can do backgating, and you can see whether the population of interest are a single population or it is a mixture of two populations. So once you have done that, you analyze the individual markers. So here you can clearly see that this population of cells are So they are mature B cells. What about this population? Yeah, so they are T cells. And what are this? What is this at this point of time, can you say they are blast? They are just MPO positive cells which are having dim CD45 and dim to moderate side scatter. Now you want to analyze probably CD34. So what do you think CD34 is? More or less negative, a small population which is coming out to be a little positive. So we gated these cells just to confirm where they are falling. Okay. So in the color bar you can change the color. So they are falling in the blast window only. Okay.
How much percentage of these? Whole population, how much is this 34 positive population? Only 5%. Okay, so very small population of 34 positive cells. Rest all, this population, abnormal population is CD34 negative. Dealing with dysplastic neutrophils or blast equivalents, right?
So next I want to see CD19 we have seen, cytoplasmic CD79A. I am including my lymphs in my plot so that I know where are my internal controls. So cytoplasmic CD79A is clearly positive or negative? Negative. T marker clearly?
So this much is actually due to autofluorescence. This shift from the negative that you are seeing, the abnormal cells are quite granular. Okay, that is why they are showing some signals in other channels, spillover because of autofluorescence.
So T marker is negative, B marker is negative, MPO is positive. So as of now we are just sure that they are MPO positive cells, but 34 is negative. TTT is also negative. And so I go on to my second tube, CD7 is also negative. In my second tube I have my myeloid markers and I know that I am dealing with MPO positive cells. So my second tube will be quite important. Okay. So I want to analyze my 13 versus 33 first. Here, what is the problem with this plot? Can anybody point out? So what are the populations of interest here? My plot is on, what is this? All events. And that is why you are seeing so many colors. So you go to the properties and you select your lymphs and blast. Now is it cleaner? Now what is the expression of CD33 on my blast? It is bright positive. And what about CD13? It is heterogeneous. So what is your interpretation of CD3 and 13 on these cells? Positive, but anything else you want to comment on? Homogeneous versus heterogeneous expression of 13 and 30 carries some significance? So what is the significance of homogeneous or heterogeneous expression of these two markers?
Whether those abnormal cells are dysplastic neutrophils or blast equivalents. You have a small population of CD34 blasts. That is not an issue. What are those cells? So those events are showing not a normal maturation pattern. This is not the normal maturation pattern shown by neutrophils. They will have CD33 will show some pattern, this is all stuck at one place. Homogeneous positivity, this is abnormal. Then 13 is also heterogeneous. So what is the expression of 13 normally in promyelocytes? Normal promyelocytes, it's bright. It's a small part of the marrow cells, but then it starts dipping down in myelocytes. And in metamyelocytes, neutrophils, it will again be gained. If you see pattern with 11B, 13, 11B, you will find a U-shaped pattern or a Nike pattern, okay, in 13, 16. So all these patterns you have to see to check whether these are all dysplastic neutrophils or they are stuck. This is maturation arrest. This plot here is showing maturation arrest. Normal neutrophils are not there, okay?
So normal ontogeny is very important for analyzing any kind of flow. So if you are not aware of the ontogeny, it will be difficult because just positive and negative will not give you answers all the time.
And one more thing is that this population, majority of them is showing CD117 positivity. So neutrophils won't be showing CD117 positivity, right? So they are clearly abnormal. And what could be these be if not neutrophils? Be louder. They are bright MPO positive. So they are promyelocytes.
So which is the next marker you want to assess? You have seen 1333, you have seen MPO, you have seen the B cells, T markers. Which is one more marker which you need to see? You can be louder. You are right. Yes. So HLA-DR is? HLA-DR is a marker of? No.
Presenting cells. Markers of immaturity are 34, TDT, 1A, CD99. Okay, DR is normally present on B cells and monocytes. In fact, normal monocytes will have bright DR. They are not blast, they are not immature. Okay.
So now you have a population which is MPO positive, 34 negative, HLA-DR negative. 33 is homogeneous bright positive, 13 is heterogeneous positive, 117 is also heterogeneous positive. So what is your likely impression?
13 किसमें पॉजिटिव आता है?
अभी मैम ने बताया. अभी मैम ने बोला. ब्राइट होता है, माइलो में डिप करता है. So here we gave it as APML microgranular variant, and then we did RT-PCR.
Wait a sec. She had a lot of doubts. Okay. So what is the diagnosis you give till P? She thought of a AML with an NPM1 mutation.
नहीं सर, that's different case. That's a different case.
But even this, a 34 negative.
सर ये अशोक है.
I know. Chromocyte na microgranular. So whatever it is, a 34 negative, HLA-DR negative does not mean APML only. So what are the other conditions which are 34 negative HLA-DR negative? AML with NPM1 mutation, AML with FFT3 ITD, AML with monocytic APL. So many things. हट से ये HLA-DR negative है, APML दे दी. ऐसा नहीं होता है. Hematology is never like this. This sequential Boolean, that is from the morphology to the clinical presentation to the flow to the molecule. Everything.
Sir, should I show them more cases because we have time?
They are busy. They are tired.
नहीं, but the other sessions are not free. एक मिनट please check. Please check.
So you have finished all the 3 sessions?
Second session है sir.
So let's have a quiz. Anyone for a quiz? Imani, कौन है quiz में?
हाँ सर, मैं बुला रही हूँ.
Imani चली गई क्या?
सर वो पूछने गई है.
We got 5 minutes. HR says 6. So, we got time, so we'll show you a quiz. ये तो वही वाला दिखा रहा हूँ.
नहीं सर जा रही हूँ मैं. So we had a 28 years old female who complained of fever, cough and continuous pervaginal bleeding since 10 days. On hemogram, her hemoglobin was 9.3 gram per deciliter. TLC was 1,11,000 per millimeter cube. And she had thrombocytopenia with a platelet count of 45,000.
Was she bleeding from the cervix or uterus? You said pre-vaginal.
Sir, yeah. So on morphology, patient had, there were more than 80.
This is not changing.
Okay. सर ये मैम ये इस पे जाएगा क्योंकि ये लैपटॉप से कनेक्टेड है, वो सिलिका से कनेक्टेड है.
So you concentrate on the screen. Okay? All of you concentrate on the screen. ये slide show करते हैं.
हाँ सर. So on microscopy, there were more than 80% blast, which had which were 2 to 2.5 times the size of a small lymphocyte, with scan to moderate cytoplasm, and the chromatin was quite coarse, and many of the cells had apple core appearance. Many of the cells showed nuclear convolution, and nuclear irregularity was visible in many of them. So there were 95% blasts, which were 3 to 4 times the size of a mature lymphocyte, with nuclear convolution, which I have already shown, and there was cup-shaped nuclei in many of them.
So what's your morphological diagnosis? ALL or AML? First level है ALL है कि AML? पहले acute में तो है ना? ALL है कि AML है? ALL.
And okay.
ALL. Which type? Why did you say ALL? Why did you say ALL? Go back to the morphology. Yeah. So you can justify. You can say, yeah, the chromatin is still clumped. It is not open like a myeloblast chromatin. Yes. See, at the end of the day, no one is wrong. Okay. So you can justify. Don't feel shy. Workshop में कोई wrong or right नहीं होता जैसे justify नहीं होता. Workshop is for clarification. If you have given ALL and son is in AML, ऐसा नहीं कि 12th का math नहीं हो. So you have kept that in mind. Cup shaped nuclei.
So the impression given was acute leukemia. So next, what will you advise? That straight away. Before flow cytometry? डायरेक्टली फ्लो साइटोमेट्री. अभी आपने सिर्फ पीएस देखा है.
ये सीधा इनके में बता दिया है. I think he is Lord Winthrop.
So genetic testing also you have to tell the molecular and pathologist the tentative diagnosis, whether they should run the DNA or the RNA, because panels will depend on your primary diagnosis. You have to determine the lineage. This won't send for directly.
Also, as of now, you have just seen the hemogram with the peripheral smear. What would be your next investigation? Before immunophenotyping? Yeah, special stains. You will advise? Yeah, so MPO, PAS, SBB. Okay. Here I am not showing you the bone marrow and the cytochemistry slides, but we did a flow cytometry, and these are the singlet, these are the NDC. Can you tell me which is the population termed as P1 in the CD45 side scatter grade? Likely to be blast. So
Now you have already seen the microgranular blast. Now see the difference in this blast.
Can you tell me any lineage marker which you can see in this slide, which is positive or negative? What are the lineage markers you can see on this? 19. Is 19 positive, negative?
19 is negative, yes.
Any other lineage marker? MPO. Is it positive or negative?
Where are the B lymphocytes? Where are the B lymphocytes? Then you can look at what B lymphocytes. population blast है clear cut 19 but cyto and PO.
So MPO was dim positive. Okay, this is a quiz. So these are the other markers.
See the other markers. See. Now see the.
So this is 13 with 38. Yes. Yes, ma'am. HLDR is negative. 64 is dim positive. 34 is also negative.
What is the difference from the previous case? 13 to 13, 33.
Hey, next class ready है? Next class को भेज दो. बंद करो यार, बहुत हो गया. चलो.
So primarily the 92% of the cells were gated on 45 versus side scatter, which were moderately positive for CD117, MPO 13 and 38. 33 and 123 were bright positive, while other markers such as HLA-DR, 34, B lymphoid markers, T lymphoid markers, they were all negative. So what is your diagnosis? Okay, you want to plus 1333 का एक्सप्रेशन कैसा था? So what are HLA-DR negative non-APML AML, which Richa ma'am has already shown in the lecture today? So one is NPM1, the other one is yes, and the other one is KMT2A rearranged. So we gave three differentials which were NPM1 mutated AML with FLT3-ITD, KMT2A rearranged non-APML AML, or microgranular APML, which you have to put as differential.
So
and
we
then you
communicated to the physicians something because it has come as an APML from outside. We told the physician this is not right. That is what we said in the morphology and flow. And we said the final diagnosis, Dr. Palti said, obviously it's molecule, isn't it? Like here. So just show the final diagnosis. See.
So on the RT-PCR for AML, when we did it, so we found that the patient had positive for NPM1 mutation while the other markers were negative.
So you got RNA markers and DNA markers. The DNA markers are based on the CT value, and the RNA markers are either positive or negative. So the entire, what is the AML 1 ETO? Translocation 821, right? Rungs 1, products 1. CBF, MIH 111, what is it? 4 binding, MLM4 E0.
सारा molecular आता है.
15 NKL1, acute, megakaryocytic leukemia. TEC-CAN, SET-CAN, monocytic lineage. BCR-ABL1, BCR-ABL minor, PML RARA, sub-negative. Then we come to DNA markers. CKIT, NPM1, FLT3, E835. FLT3 ITD is done by conventional PCR. But E835 is done by RT-PCR. NPM1, the DNA, CT value, see here. CT of the individual is calculated as CT. Less than 5 is considered positive. In CKIT and FLT3, and less than 6.2 is positive in NPM1. Less than 6.2, this is 0.92. So this is AML with NPM1 mutation positive. What is the meaning of this? Good prognosis, bad or intermediate? Good. Suppose the NPM1 is mixed with the FLT3 ITD. I well understood. Thank you. और कुछ बोलना है? Thank you बोलते हैं. चलो. Now go to where? All the best. हाँ. Last batch. 4, 3.
तक देख के आ रही हूँ.
लास्ट बैच अभी तक अभी तो बता दिया कहाँ है लास्ट बैच कहाँ है I think they must be dead after a while. Most boring session. Yeah, चलो settle down. हिमानी का है come to the front. There is a laptop here. Quickly, laptop. Live meeting करोगे. तुमने ना ऑफिस के लिए तुम क्या बोलो? 3 बैठ से बोलो. वाइट होता है. Please, इतना जगह तो ट्रेन से प्लेन में ना मिलता है. Request भी है सब चीजें. It's like what that premier. इसमें आ जाओ free here live meeting. Live meeting और इसके पीछे भी active involve yourself. Like all of you to involve yourself with this. Okay. One more can come here. Anyone? Take it. Yeah, you. सुजाता जो तो फर्स्ट केस कम अच्छा स्टार्ट करेंगे कहाँ से ऐसे लिखा हुआ you are coming from the MRT or from the CLPD which is one of the classical markers of hairy cell leukemia. Don't miss out on white CD22 level is 25 SLBL what percentage of SLBL is CD22 25% cases of SLBL can be CD22 just around it. Be careful. Clinic lymphoma is prominently visualized. NTD now doesn't. There are a lot of things. PLL is no longer done. So it comes under this. When you see the vessel scan of PLL, they all look like lymphomas, but they actually not. So you do some is different hemoglobin and the 90. Side scatter 90, side. अभी ब्लास्ट होना ब्राइट होगा the moment you see side scatter 19 or side scatter 45 with the cells so bright your blast is off and we're dealing with CL आजाओ ज़्यादा पीछे मत जाओ आगे आ जाओ प्लीज कम से शिल्पी why are we delaying हाँ तो I'm waiting for you please start Professor Sujata, Professor of Pathology, ESIC Masai, earlier ESIC FARIDABAD. Professor is going to present you shortly. Start. Yeah, this is a good focus, Sujata. This is a good focus. Right, let's start. Jaldi आओ, So you can describe. Yeah.
So we start with the first case of the acute leukemia. So we have a case with a 58-year-old male presenting with complaints of fever and generalized weakness for 10 days. The lab investigations show the hemoglobin is 4.5 gram per deciliter, total count is 27,000 and the platelet count is 5,000. So what is your impression? That the TLC is increased, but the other two parameters are decreased, the platelet count and the hemoglobin. So on the peripheral smear, if you focus, the cells are predominantly microcytic hypochromic and occasional polychromatophils are noted. Occasional NRBCs are also noted. However, there is no any evidence of schistocytes or fragmented RBCs. That is, there is no evidence of hemolysis. The TLC, as we saw that there was leukocytosis, and most of the cells are blast-like morphology, having 3 to 4 times the size of a lymphocyte, having amphiphilic cytoplasm. If you see the cytoplasm of these cells, while some are having the vacuolations. Can you appreciate the vacuolation in the cytoplasm? The nuclei are irregular and there is a clumping of the chromatin with prominent nucleoli in few of them, and there is perichromatin condensation. The platelet count is markedly decreased, and our impression on morphology is acute leukemia and likely lymphoid type. So now I invite Dr. Shilpi to come and explain you the live gating of this case.
Good afternoon, everyone. So you can replicate what is being shown here, as you've already seen. Okay. So now you are already aware that the first plot which you have to see is, time plot is very important when you are analyzing MRD. In leukemias and lymphomas, you have to choose singlets because you want to remove your doublets. So singlets you choose on forward scatter area versus forward scatter height. So here you can clearly see these are my doublets. Okay, and ma'am 3698. खुल गया मैम? Somebody help, विशाली वहाँ पे help करा पासवर्ड. मैम पासवर्ड खुल गया उसका? Okay. Okay. So once you have gated your singlets, next what do you want to do? You want to remove your debris. So how can you do that? Huh? Yeah, on forward scatter versus side scatter, which population would you choose? No. So the population which is having low side scatter as well as low forward scatter are your debris. And the rest of the population is your NDC. But what I wanted to ask was, when you want to make, you want to draw your NDCE gate, that is forward scatter versus side scatter, how would you proceed to that gate? You would make a separate plot, or you will do something else with the first plot? See, abhi they are pre-formed gates. Because we were taking a workshop, it is easier for us to demonstrate this way. But if, suppose there is no gate. So when you are analyzing the flow cytometry, when you have a new setup, what would you have? You won't have any kind of plots. So for making the plots, what you do is, you go to the create dot plot, icons from the top, you choose that and you draw the plot. And on this you can specify what you want on the x-axis, what you want on the y-axis. Here, to make it simpler, we have this procedure for you. And once you have the singlets, what are you going to do with that population? So these are my singlets. You can click on the gate. So this whole thing is a plot and this is a gate. You click on the gate, you go to the properties, you click on the gate and you drill down. Drill down means you are selecting your singlets to choose from for making a gate for NDC. Because if you choose all events, you will not be ruling out your doublets. Okay? So it is basically Boolean gating or the sequential gating you do. So from one population, you are selecting the further down population which you want to identify and which you want to choose and gate. Alright? So these are my singlets. Then this is my NDC. So these are the ones which is having low side scatter and low forward scatter. Those are my debris. I have ruled out that population. I have made the gate on NDCE. And the main gating plot is with CD45. That is correct. Okay. So can anybody tell me what are these two populations? Yeah. But are you sure those are lymphocytes and blasts? How can you confirm?
So you guys did MRD and platelet plasma,
Yeah.
right? So was there a large population or they were more concentrated here or here? Here you have a large chunk of population. Here, right. So what is the possible lymphocytes? This is the lymphocyte. This is the population of the lymphocyte blast. And what are these?
So like ma'am has said, so the blue population are your lymphocytes which serve as internal control, and the red population are your blast. So now going on to my individual plots, which are the markers you want to analyze. In the first tube you usually have lineage markers, yes. So the A-lot tube which ma'am was talking about, so it will have cytoplasmic markers which includes your cytoplasmic MPO, cytoplasmic CD79A as well as cytoplasmic CD3 and your TDT plus your lineage markers, that is CD19, and immaturity marker which is CD34. Okay. So here I have put my gate on, I have put my population with CD19 and cytoplasmic MPO. So what is this population of cells? Which lymphocytes? Yes? Yeah. So basically they are negative for CD19 and MPO, so they are T lymphocytes. And what is this population? And the red population? Which are positive for, no, this 19, correct. And now I want to confirm whether these are mature B lymphocytes or something else. So now I have gated these cells with 19 versus 34. So what are these cells? Okay. So my blast population are CD19 as well as CD34 bright positive. Is that clear? Then coming on to, 1 second, I'll take 19 here and I'll take, so 19 is on the x-axis and y-axis has now CD10. So CD10 is again bright positive. Cytoplasmic CD79A is bright positive. So bright and dim positive we say according to, this is decided as per, yes. So here your internal control is, in most of the plots your internal controls are lymphocytes and if granulocytes are there, granulocytes. So your positivity or negativity will be dim, bright in respect to whether your lymphocyte, where your lymphocytes are and where your grans are. So you will decide it on the basis of your internal controls. So here this is your, for example, if you put a quadrant gate. So these are my population which is completely negative. And this is the population which is bright positive for CD19 as well as cytoplasmic CD79A. And your blasts are also falling in the same area. And to confirm, you can always change your plot type and see the contour type. So the main population is just near about to the internal control positivity. Alright? So you have seen CD19, you have seen CD34, NDMPO is clearly see the log difference.
There is no internal control, right? That is why. So this is peripheral blood specimen that has been acquired. If there was bone marrow, there was no issue. Internally, blasts, myeloid blasts would be there. So positive, negative, when do you say a population is positive and when do you say a population is negative? If there is no internal control. Positive internal control. And you are seeing lymphocytes. Lymphocytes won't have 34. So if any population is ahead on that axis of 34, then it is positive. Whether dim, moderate or bright, log scale can decide. 1 log, 2 log, 3 log, if there is no internal positive. Other way is to see the unstained surface tube. No marker, only 45, or for unstained surface, they may not be 45 also. So where the background fluorescence is falling, okay, ahead of that would be positive.
So, cytoplasmic CD79A we have already seen. Okay, nuclear TDT. So, what is TDT here?
Again, TDT has no internal positive control. So, what do you think TDT here is? So it's very, so when no internal positive control is there, then we see the log scale. So your negative population is still this log, right? And this is just half log ahead of the negative. Dim. Dim. Or maybe moderate. Dim. It's not crossing 1 log.
Also, you have to see your T markers. So, like here, you can see the mature T lymphocytes are here, and your blasts are going above that. But you would not make it positive because there are certain channels which have more autofluorescence. Okay, and here you can clearly see that your T lymphocytes are much above the negative population. Plus, to confirm, you can have other T markers such as surface CD3 and your CD7. So these are your B lymphocytes, and your blasts are falling exactly with your mature B lymphocytes. And the ones which are T lymphocytes are clearly CD7 positive. The blasts are negative for CD7 or positive? Okay. So this was the A-lot tube. In the second tube, we have the myeloid markers. You can clearly see that 13 is negative, 33 is also negative. It is some autofluorescence which is coming out like this.
Subset is positive. That can be seen in part of cross lineage expression in abnormal B blasts, right?
So some contour is showing, yeah.
Fluorescence in B lymphoid blast that much. BALL, normal, abnormal, B blasts or B cells. Is DR expression normal or abnormal on B cells? Normal. So is DR a marker of immaturity? No, it's not. Okay. So basically, myeloid blasts can have 34 DR, but in general, we do not call DR as marker of immaturity, right? DR is present on antigen presenting cells. It is present in monocytes. Normal blast also show DR. It's not a marker of immaturity, okay?
So this case had bright 19, bright 10, bright cytoplasmic CD79A with MPO negativity and TDT positivity. So it was basically a Kala positive BALL. Okay. So now coming on to the next case, ma'am, do you want to add on anything?
That you should always check the 45 site scatter plot for the CD45 negative region. So many a times this BL is clearly showing CD45 dim positivity, but many a times you find, especially if there is fibrosis and diluted marrow, you find very few blasts even in the bone marrow, then you can miss out on B blast if you gate out these cells as debris. So remember to check this area also for B blasts. Okay?
Just to give example of what ma'am has already said, so this was another case in which in the NDCE, when we gated the population, we had 3 population of cells. In morphology, we had blasts. These were clearly lymphocytes, these were glands. And we were not sure whether this is debris or these were blasts. But when we backgated, all these cells were 1910 positive with 34 positivity. So, like ma'am said, a negative or a very dim CD45 can be B lymphoid blast.
So one more point here, if this was not peripheral blood and marrow of a child who is recovering from some illness. So in that previous case plot, can you show? Yes. CD45 dim positive cluster, it's a cluster, it's abnormal. But based on 1910 positivity, can there be another differential? If you're not seen the morphology, ideally you should have seen the morphology and clinical scenario. But what are the other cells which can be here in CD45 dim region 1910 and 34 positive? Hematogones. So second tube in BL for BLL helps in differentiating hematogones from B blast, and based on those pattern of positivity, we give further clues as to how the patient can be tracked for those abnormal blasts, differentiating them from the hematogones. When later on the patient recovers from chemotherapy, this B-ALL patient will have hematogones. Then you will have to differentiate them from hematogones. So you should always put a second tube to know how to differentiate these from hematogones.
Now I would like to invite Dr. Sujata to give a gist of the case.
Thank you, Shilpi, and Dr. Richa for nicely explaining the gating. I hope all of you followed. So, to continue, as Dr. Shilpi said, she showed you the live gating on this case. So this is our flow report. This is how we get the report. So you can see on your first column is the marker. So we have enumerated the markers here. Is it visible? Can you now see? इस स्लाइड शो तो नहीं होता इसमें ना, तो क्या होता है? So you can see these are the markers on first column. The second column gives you the intensity of these markers. So we mentioned like is it dim or moderate, like CD45 usually it is dim or moderate, or it is bright. In this case the bright markers are CD34, TDT, CD79A and CD10. Okay. So, and the last column shows you whether the markers are positive or negative. So, we have actually gated CD45 versus site scatter and found that 70% of the cells were blast, which on morphology we saw it was 82%. These cells were dim to moderate CD5 positivity with dim site scatter. The cells were brightly positive for CD10, 19, 20, TDT and HLA-DR, while there was moderate positivity for CD38, CD22 and 123, and again CD79 was bright to moderate positive. So based on the morphology and the immunophenotypic features, the final diagnosis was Scala positive B-ALL. So now I call Dr. Himani to come and present the second case.
Sujata, you can present the second case.
Okay, so after a quite learning and interactive session of acute lymphoblastic leukemia, let's come towards our second case.
So just before that, so this is a 58-year-old elderly male who has presented with acute lymphoblastic leukemia. What is standard risk? What is intermediate risk? What is high risk? NCI. Correct. Age 1 to 9 years. TLC less than 50,000. response to prednisolone therapy within 7 days with the blast count reduces. So these are all standard risk. Okay? Now, molecular mutations have come in a very, very big way, right? So, did we show the molecular mutation analysis in PALL? Right? So you see this. What is E2A PBX1? What translocation is E2A PBX1? So you should read all that. Okay? Read childhood. Yes? Yes. Translocation 119, which is an adverse prognosis. So childhood, whatever ALL you read, whether childhood, elderly, you must know what is Philadelphia positive ALL, what is Philadelphia like ALL. You know, we have got a marker called CXCR4 in flow. If the CXCR4 is positive, we can say that this patient is likely to have a Philadelphia like ALL, which is somatically and genotypically like Philadelphia positive, except that they don't have the translocation or the BCR ABL1, both major and minor. But they respond well to upfront imatinib therapy. So the flow is giving you so many informations. So just don't do flow mechanically. Follow your morphology, follow the biology of the case, do the flow, and each and every marker has got its significance. Maybe the future MRD will depend on your diagnostic panel. So take flow very seriously. Okay?
Okay, so coming towards the second case of the session. 41-year-old male presented with complaints of hematuria and black colored stools for 2 days, and on examination, moderate hepatosplenomegaly was there. Lab investigation reveals hemoglobin of 10 gram per dl, WBC 25,900 cells per cubic mm, and platelet 21,000 per cubic mm. When we saw the peripheral smear, RBC picture shows predominantly normocytic normochromic RBCs and fair number of macrocytes with occasional polychromatophils, and occasional fully chromic teardrop cells are seen, and no schistocytes were there. WBC series
What is the significance of fully chromic teardrop cells? Every word she says is important. What are hypochromic teardrop cells? What condition gives you hypochromic teardrop cells? Microcytic hypochromic anemia of iron deficiency and thalassemia. Fully chromic teardrop cells? Say again? Correct. Myelofibrosis or any myelopathic anemia which is involving the bone marrow. Okay.
On TLC, we saw 90% hypogranular promyelocytes, which were blast equivalent, with moderate cytoplasm, large binucleated cells, along with occasional multinucleated cells. As you can see on the peripheral smear, some of them shows prominent granules in the cytoplasm interspersed with occasional myeloblasts. Mature lymphoid and myeloid cells were sparse. Platelet count was 20,000 per cubic mm on smear. So, final provisional diagnosis was given as acute leukemia, likely acute promyelocytic leukemia. So, we advised flow cytometry on the peripheral blood. And now I would request Dr. Shilpi ma'am to demonstrate the live gating of this case.
So, before you come to the live gating, this impression was given on morphology. Why? Because we didn't want to deny the patient ATRA therapy. What is the full form of ATRA? Yeah. So, it was a weekend on Friday. And we gave this diagnosis on morphology. Flow we are doing. We gave the flow report also in the evening. But in the morning, we already initiated the ATRA therapy on morphology, right?
So here, like we have already discussed, the first plot will be of the singlets. So once you have gated your singlets and removed the debris, removed the doublets, you want your NDCE. So this is my NDCE where I have removed my debris. The cells with very low side scatter and very low forward scatter are your debris. Once you have removed that, what do you want to do? On NDCE you want to do what? Huh? Yes. So what will you do for that? Yeah, dot plot is the type of plot. How do you make that plot? You can choose this gate, you go to this gate and you drill down. Okay? On drill down, you choose your CD45 with side scatter. So here my parameters are going haywire. So you check the parameters by going again on the plot properties and you choose linear for your side scatter and you will have the plot like the way you want to see it. Okay? So your basically what is flow cytometry? what you are analyzing. They are usually which kind of plots? Bi-exponential. What do you understand by bi-exponential plots? So either they are in the linear mode or the log mode. Okay? So for the side scatter, you have the linear expression. For the fluorochromes or the specific antibodies, it is log. Alright? So once you have done that, so this is my side scatter versus CD45. So any comments on the population which I have? So what is this population? That is limbs. And any specific thing you want to say about the, yeah, not a fully teardrop shell but a half teardrop, because it is not high side scatter but it is dim to moderate. But yeah, some teardrop can be appreciated. So once you have population of interest, you see the individual markers. So here I have, this is cytoplasmic CD3 with MPO. So these are, what is this population? So these are mature T cells, correct. And what about this population? Is that clear? These are mature B cells. And what about these cells? So MPO positive and negative for cytoplasmic CD3. CD19 is clearly on the blast. CD19 is positive or negative? Negative. So first is your ALOR tube. The ALOR tube is comprising of the cytoplasmic markers as well as the lineage markers. So lineage markers will include your myeloid, B lymphoid as well as T lymphoid markers. And the cytoplasmic markers are MPO and MPO and what did you? And TDT. Okay? So TDT, cytoplasmic CD3, cytoplasmic CD79A and cytoplasmic MPO. Plus we have the immaturity marker, that is CD34, and the lineage marker 19, correct. So my 19 is clearly negative on the blast, MPO is clearly positive. Then I want to see my 34. So 34 is? Yeah, it is negative. And for checking what about this population, what you can do is you select, yeah, you backgate. So it is falling in my blast, and this is just 5% of the cells. Okay? This was, no ma'am, peripheral blood.
So peripheral blood, mostly they were giving circulating blood. So internal control difficult point. If it was bone marrow, easy to say because some myeloid blast would be falling in 34 positive region, which will be negative for NGO. Normal blast are negative for NGO. They will be falling here. We can compare the positivity in myeloid to myeloid. But if there is peripheral blood, then we check the negative population. How much negative population is from a negative control and how much log it is covering. So
आप B lymphocyte ले लीजिए ना, mature B lymphocyte ले लीजिए, आपके internal control है. You don't expect 34 to be positive as that. Is that? You now see the blue cells. What are these blue cells? I already said that.
Then moderate, right, based on how much log positive. Less than 1 log, then positive. 1 to 2 logs, then moderate. Brighter than 2, bright positive.
इसमें एक lymphocyte का just select lymphocyte.
So the blue population is lymphocytes. So there were no grans in the morphology only.
Okay, there were two populations. So you have no choice but to have the lymphocytes only. Based on that, negative. अगर
कि थोड़ा पॉजिटिव है.
It is autofluorescence also, sir. These blasts are granular of morphology. So we expect some autofluorescence, which might be causing some symptoms. But the negative population only, the blasts are negative, but they are falling still 10 to the power 3 because of autofluorescence.
What Dr. Vijay is telling, if you put a quadrant here, this portion is positive.
So, that's what. If you do autofluorescence of the property of the blast.
So at this stage, you can say majority of the blasts are 34 negative, but some are 34 positive. You are not wrong in that. Instead of telling that bulk are 34 negative, बोले कुछ positive है, कुछ और आपने backdate करके वो जो खाली उसको दिया, बाकी सब नेगलेक्ट किया. अगर B lymphocyte आपको internal control है, तो कुछ तो positive आ रहा है. And microgranular variant, nothing to do with what here. Bad. A microgranular variant can have a 34 positive and a 34. Unlike a hypergranular variant, which is majority 34.
So, my
APL gating especially is difficult because of autofluorescence. Many of the markers appear to be positive. But they are not. So they come as rocketing in most of the plots.
तो अगर क्लासिकली TA ड्रॉप मिल गया तब तो लाइफ इज इजी अदरवाइज
So what Patty sir said, that you can't diagnose APL on flow, you have to demonstrate PNL or
Anyone is going to take this question, what is autofluorescence? We are continuously talking about autofluorescence. And you will see this in various conditions which will. So what is autofluorescence? So Chilpi, case, all are out.
So basically, it is the inherent property of the fluorochromes to have some fluorescence. So that inherent property, which is manifested, that is the autofluorescence, and you have to remove that. And I think Richa ma'am will be able to better explain.
Fluorescence is a broad term.
Yes.
And we have fluorescence due to the,
Inherent
everything that
property.
we use for flow. The fluorochromes, the sheath fluid, the filters, the optics, everything that is being used will have some background, contribute to the background fluorescence. Plus the cells have granules, okay? So what are fluorochrome molecules anyway? Some are natural enough. So cells can also have natural components that can fluoresce right in the visible spectrum.
Both the components.
So that can also contribute to your background fluorescence.
Vinay, can you just, yeah.
Because of urine and pyrimidine, all biological samples will have some level of autofluorescence in all these channels.
Any way of reducing it?
No.
There is no way. This is the internal property.
There is
You
no
have to
reason
face it.
that we have to see
Unstained.
Got it. So this has to be actioned. This is a limiting factor. Okay, carry on. Yeah. So what are you getting? Are you the gate here?
So we have seen CD19, we have seen cytoplasmic CD3, anti-MPO, CD34 we have seen, and TDT is also negative. So we go to the second tube, because in the first tube, basically all my cytoplasmic or the lineage markers, only the MPO is positive and 34 is also negative. So in the second tube, that is my myeloid tube. So I want to see my CD13 versus CD33. So what is the expression of CD33? Positive. And vis-a-vis 13? Vis-a-vis 13. So 13 is also positive, 33 is also positive. What is the difference? You want to comment on anything else also in their positivity? Yes, 13 is heterogeneous positive, 33 is homogeneously positive. So what do you interpret by that? In the ontogeny, which cells are homogeneous positive for 33 and which cells are heterogeneous? So based on this, what do you interpret them to be? A normal population of cell or abnormal population of cell?
So what is the expression of CD13 on the blast, on the promyelocytes, on the myelocytes? You must know that. Where is it maximum? Where is it minimum? What is dipping of CD13 expression? So it comes in the myeloblast. It comes in the myeloblast. It is maximum in the promyelocyte and then dips again. So that sort of expression is should go. So what is the expression here? 13.
33.
33 and 13.
So,
we have just a 13 expression, and can you replace 33 in y with something else?
so when you see a
Yeah, now see this.
So you can clearly see that 13 is heterogeneously positive. And the same thing when I put with 33, it is bright positive. Okay? So 33 is bright positive in your promyelocytes, but it dips in myelocytes. And again, the expression is increased in your metamyelocytes and neutrophils. So now I can clearly see that the 33 population, the positivity, it is stuck. That means there is some kind of maturation arrest. Because in case of a maturing population, you will have a heterogeneous expression of CD33, which we are not able to see. Is that clear? And
So what is classical 13 expression you would have expected here? 13, we are not talking 33 now. 33 आपने बता दिया. 13.
13 has to be bright positive if it is classical APML. So based on 13, 33, you can interpret whether the cells are mature or they are immature. Okay? Then coming to CD117, here you can clearly see CD117 is positive. So which gives us assurance that these are not neutrophils, because neutrophil will not have such expression of CD117. Okay? So when you have to differentiate between a mature population vis-a-vis immature population, 13, 33, 117 are very important myeloid markers. So you have to be aware of ontogeny to have an interpretation correctly. हाँ sir, next I am coming to HLA-DR only. So what is HLA-DR? Negative. What is HLA-DR? Not in this case. What is it? It is a marker of? Yes. So basically it is an activation marker or antigen presenting cell, and DR is present on B cells, T cells, monocytes. Okay? So it is not an immaturity marker. Okay? It can be present on your B lymphocytes, B lymphoid blasts also. That is a normal presentation of HLA-DR. Alright? So here you have a case where MPO is positive, HLA-DR is negative. 33 is bright positive, homogeneous. 13 is heterogeneous positive. 117 is again heterogeneous positive. So, and 34 was negative. So what is your likely diagnosis?
So what is the 34 negatives?
HLA-DR negative.
You've already seen the variant expression of person. Yes, we know there is a maturation there. But just by saying HLA-DR negative, 34 negative, APM is the wrong statement. So what are the other conditions which are 30 and 33 HLA-DR negative and 34 negative? Even monocytic leukemia sometimes can present like that. AML with NPM1 mutation can present. AML with FACT3 ITT can present. And HLA-DR negative 34. There are a lot of conditions. So now you have seen the we have already given a diagnosis so that the patient is not denied at the on the Friday night, isn't it? And we get the flow, and yet we do not know what it is except that there is a maturation. Then Ivani?
Also 64 was DIM positive in this case. So I would like to invite Dr. Hemani to present the gist of the flow.
Last 5 minutes. Come on, let's finish it. Because we got another long way to go. I hope you all are enjoying. Very informative. We are also enjoying your presence and your lovely answers.
So after learning the live gating, this is the final report which we got. 89% cells were gated on CD45 versus side scatter, and these cells show moderate to bright CD45 positivity and dim side scatter. CMPO, cytoplasmic MPO, CD13 and CD33 were bright positive. CD117, CD14, 64, 11C were dim positive. CD34, HLA-DR were negative, and CD38, CD56 were moderate positive. So final impression was given on the basis of morphology and immunophenotyping impression was acute promyelocytic leukemia, APML hypogranular variant was given. And further, we advised molecular by RT-PCR, and this is what we got. On RT-PCR, we got the DNA marker, we got the RNA marker PML RARA positive. So final impression on the RT-PCR report was given as acute myelogenous leukemia positive for PML RARA mutation marker.
Okay? Excellent. So we have come to the end of the session. I thank Professor Sujata, I thank Dr. Shilpi, Himani, of course Richa Veshi. Come here. Give her a great applause. From a busy schedule from AIMS, she has come. And before we end, we would like to felicitate our senior two chairpersons, Dr. Vijay Tilak and Dr. Deepa Rani, who have come all the way from Banaras in the university to grace the occasion. We are extremely grateful for both of you. And may we request Dr. Richa to present a memento to Vijay Tilak. And Jasmeeta, I would request you to present a memento to Deepa Rani. And may I request the cameraman who is never here to take a photograph. Just when we want to photograph, he's just a bit. Could you pass a message to him? Because he takes a click and says eyes closed. So I think one of you can take in your mobile and share with us. That will be better. So, Vijay ji. Come. Please take a photo from the front because he's looking there and he's clicking here. Take some. Right there.
इनको दिया भाई
कैमरा में नहीं है ना
मैम ये डोंगल भी है संभालने के लिए ना डोंगल भी है
And also before we end, though we always consider Professor Sujata as part of our team, but now she's in ESI Basai, and it is my pleasure to present this memento to Professor Sujata, who has been a very, very dear friend of mine also. Himani?
Yes, sir.
Well done.
Thank you so much, sir.
बहुत अच्छा. You are very bright people.
Thank you, ma'am. मैं फोटो ले लेती हूँ. ऐश्वर्या बैग देना, इसका भी डाल देते हैं, ये वाले लैपटॉप का बैग। फिर हम अपने लैपटॉप्स का वो कर लेते हैं कि ये कहाँ जाएंगे।
यहाँ से ना सब
दीदी के जो लैपटॉप्स है वो तो अभी तो
सिलिका का जो भी स्टफ है, जो भी सामान है ना, अभी वहाँ भी शिफ्ट करना है और वहाँ पे लॉक करना है। मैम ने कहा है।
तो ये उसको बोलना है नरेंद्र को, नरेंद्र एंड यश।
सब कुछ ना शिफ्ट करना है, जो भी यहाँ से यहाँ पे जो निकालना है माइक्रोस्कोप सिलिका का
ये भी साथ में जाएगा, ये भी साथ में जाएगा। तुमने दो माइक्रोस्कोप, दो लैपटॉप रख लिया, दो लैपटॉप रख लिए ना, 3 नंबर। हाँ, दोनों लैपटॉप साथ में रखे ना। ये ऐश्वर्या के पास है, ये वाला 3 नंबर का देखिए, इसके साथ
ये सुश्रुदा पे रखो।
एक माउस मेरा है इसमें पर्सनल।
अभी ले रहे हैं
कल इसके अंदर ही है
माउस इधर 8 और 9 है।
हाँ, अब ये बैग
ये किसका फ़ोन है हमारे में, आपका है?
हाँ मेरा रखा हुआ है, ये वाला विशाली, ये भी उधर ही जाएगा। कहाँ रख रहे हैं?
सुश्रुत में।
किसी से माइक्स का पूछना, माइक्स रखे हैं यहाँ पर।
वन के लिए तो नेट को तो अपने ऑफिस वाले देख रहे हैं, सच्ची में प्राइवेट लैब में ऐसी है। प्राइवेट लैब का सच्ची में एक मैं सच्ची में गई थी। आप ही जो टेक्निक, अच्छा मैं गई हूँ पोडियम पे ज़्यादा हो जाएगा, लगता नहीं मुझे, पोडियम पे रहना। हाँ ठीक है, बिल्कुल ऐसा लगता है, बिल्कुल ऐसा पोस्ट जैसा पोडियम पे खड़ा हो
काजी लॉक
ठीक है अभी कर
मैडम मैम नहीं कोई बात नहीं, कल के दिन स्लाइड चेंजेस करवाना तो इस काम ही नहीं करेगा।
हो गया भाई जी?
अच्छा इसमें बस नेक्स्ट टाइम लगेगा।
कोई बात नहीं। और तेरे वहाँ में कोई रूम तो ना है? तेरे वाले रूम में?
रखना है सामान।